Good morning, everyone. I hope everyone can have a seat. I want to welcome all of you for joining today's workshop. I'm Lee Hellman, the director of the Osteosarcoma Institute, a nonprofit dedicated to improving the outcomes of patients with osteosarcoma through a number of means, including funding preclinical and clinical trials, among other things. It was founded by Mac and Lisa Tishner and by the Quad W Foundation in memory of their son, Willie, who lost his life to osteosarcoma. And Lisa and Mac have dedicated themselves to honoring Willie, honoring Willie's wishes by working to improve outcomes in patients with osteosarcoma everywhere. And Mac and Lisa are here. So thank you again for everything you guys do. So solidifying partnerships is the driving force behind today's FDA OSI workshop entitled Advancing Osteosarcoma Drug Development, Connecting Research and Regulatory Pathways for Improved Outcomes. In May of 23, I can't believe it was this long ago, in May of 23, the FDA Oncology Center of Excellence hosted a symposium entitled Current Challenges in Clinical Trial Design for Relapsed and Refractory Osteosarcoma. Participants included advocates, FDA experts, and academic leaders in the field of osteosarcoma. It became obvious to me, and I think to all of us at OSI after that meeting, that FDA members are our knowledgeable partners in helping to provide promising new therapies to our patients. That's their mission as well. But we also know that we need to partner with pharma. And they weren't at the meeting for technical reasons. But we need pharma to be successful in our quest to improve outcomes for all patients. We need new drugs active against osteosarcoma, and we also need access to existing drugs that were developed for other purposes but have activity in osteosarcoma. And we need the drug development expertise of pharma. Osteosarcoma is a rare tumor, obviously, and it will never be a blockbuster business opportunity. And that's a problem. We all recognize that. It is also a highly complex, biologically challenging tumor, hard to fit into existing regulatory frameworks. But there are champions in all of these areas eager to help meet these challenges. And the more we all know about each other's challenges and capabilities and roadblocks, the more effective our collaborations can be. We're very happy to have our critical stakeholders here today. As I said, we didn't have pharma at our last meeting. I look forward to today's discussions and anticipate that we will find ways to work together to accelerate progress that is so desperately needed in this disease. Once again, I thank you all for taking time out of your busy schedules. I know some of you are flying in and flying out, and we really appreciate that. And we look forward to a very interesting discussion. Can I get the slide? Okay, so I just want to remind people that our speakers won't be able to address individual, because I know we have lots of advocates here, and of course we're all here for the same purpose, to help our patients. But we won't have time to address individual patient questions regarding treatment. But the OSI does have the OSI Connect. It's a free, easy to use resource dedicated to supporting osteosarcoma patients and their family. And if anyone is interested, you can use that code, and we'd be happy to take anyone that's interested in talking to us in the future. Thank you very much, and I will turn it over to our next introductory speaker. Thank you.
Good morning, everybody. And thank you, Lee. It's truly an honor to be here. And on behalf of the FDA Oncology Center of Excellence, it's my great privilege to second Lee's welcome to today's workshop. It has been a very long time in coming, and we're deeply grateful to the Osteosarcoma Institute and their supporters for their hard work and commitment for making this meeting come to fruition. During the many months of preparation for today's meeting, I've been really humbled and struck by OSI's unwavering dedication to creating a program that will foster collaborative and constructive discussion and identification of approaches that will enable meaningful advances in the treatment of our patients with osteosarcoma. I'm joined today by some of my colleagues at the FDA, Drs. Nicole Dresner, Dr. Jasmine Smith, Dr. Katie Barnett, Dr. Kristen Wessel, and I think I saw Dr. Jeff Summers here as well as Erica Haradnachanu, who was really instrumental in helping put this meeting together. And I know that we have many other FDA staff online. And I hope this signals to you all the level of dedication that we have to doing everything that we can possibly do to foster development of new, safe, and effective treatments for patients with osteosarcoma. Many of us have treated patients with osteosarcoma. Some of us still do, and those patients really remain in our hearts and in our minds in all of the work that we do every day. And we understand the urgency and need for new and better treatments. I know that I speak not only for myself, but for all of my colleagues when I say that we are truly excited and humbled to be here and to take part in what we know will be very stimulating and collaborative and constructive discussions with all of you today. I have to figure out how to switch the slide. Okay. This slide summarizes the goals for the workshop, which Lee really touched upon quite eloquently already. Our objectives are pretty wide in scope and threefold. The first is, which I think is the primary goal, which is to bring all of us here today, representatives from every important stakeholder community involved in product development for osteosarcoma. So we've already achieved at least part of that goal. And the rest of what we hope to accomplish really will be accomplished through the rest of today's discussion, and I hope the discussions that will come later. The second goal is to identify opportunities to increase efficiency in product development, to learn from what we've done in the past, how we can address the challenges that we've encountered, correct any mistakes that we've made along the way, and really establish the foundation for future kind of improvement and discussions going forward. This is quite a lot for us to accomplish in one day, and I think we will definitely have our work cut out for us, not only today, but in the days to come. But I'm confident that the discussions that we have will strengthen our community and foster improved and open multi-stakeholder engagement and lay this foundation for a better future for our patients. Lastly, I just want to express my sincere thanks to all of you. As Lee said, we know how busy everybody is. We're grateful that you guys came to DC to have this meeting so that so many of us at the FDA could be here today. And this workshop really could not be a success without each and every one of you and for your openness and collaboration today. I'll now pass the microphone on to a dear friend and colleague, pediatric oncologist, who's the Deputy Division Director of the Division of Oncology 2 at the FDA, and that's the division that helps oversee product development for osteosarcoma. Go ahead, Nicole.
Hey, good morning. I'm Nicole Dresner. I'm really happy to be here. I'm going to quickly go over the agenda for the day and some housekeeping items. So the agenda is shown here. In the first 4 sessions, you're going to hear insights from patients and advocates, followed by presentations in session 2 on osteosarcoma's clinical landscape to date and the regulatory requirements for rare disease drug approval, and in session 3 on some promising therapeutic targets for osteosarcoma. After a break, we will hear presentations and discussion on regulatory, clinical, and industry perspectives on endpoints and innovative approaches to trial design in osteosarcoma. In session 5, you will all have the chance to ask questions to any of the speakers from sessions 1 to 4. And then we are planning for a lunch break at 1:30. Right after lunch, we'll have the wonderful opportunity to hear from Nancy Goodman of Kids v. Cancer in session 6's Fireside Chat. In session 7, we are going to focus on the path forward for osteosarcoma drug development during a moderated panel. And in session 8, you'll have the opportunity to ask questions of the session 6 and 7 panelists, followed by closing remarks and an ending at 5 p.m. In this very packed day. So some additional important housekeeping items. For questions, if you're here in person, please state your name and organization and the intended speaker for your question, if applicable. For virtual attendees, please submit questions by using the Q&A tab at the bottom of your Zoom screen, and we will try to get to those as well. We note that we may not be able to respond to every question due to time or confidentiality constraints. For myself and my FDA colleagues in particular, we will not be able to discuss specific products and their development plans or the off-label and investigational use of specific drugs. We also will not be providing formal FDA advice during this meeting, and the views that are expressed are those of the individual speakers and not official FDA positions. Please feel free to take photos during the workshop and notify the OSI staff if you prefer not to be photographed. Finally, importantly, slides and a recording will be available after the event. So I just want to really thank you all again for being here, both in person and virtually. I know I speak especially for my FDA colleagues that we are really thrilled to be here and join you. And we hope that the discussions today strengthen the collaborative bonds that we share toward our common goal of developing effective therapies for children and young adults with osteosarcoma. I'm now going to turn over to Mac Titchener of OSI, who is going to moderate our first session on patient and advocate insights. Thank you so much.
Well, we'll go ahead and start, and we'll have the roster put up in a moment. We're going to begin this session by hearing directly from patients whose experiences remind us of the stakes that are involved in what we're here for today, osteosarcoma treatment and research. First is Mackenzie Madry. And by the way, I'm Mac Titchener with the Osteosarcoma Institute. Should have said that first. But our first patient who's going to speak with us is Mackenzie Madry. Mackenzie is a survivor of osteosarcoma whose journey through diagnosis, treatment, and survivorship captures both the toll and the resilience that define the disease and the people who face it. Mackenzie's been generous with sharing her story to help all of us researchers and industry understand what it really means to live through osteosarcoma. And we're very pleased, Mackenzie, that you could join us today.
Thank you all for having me. It's really nice to be here. Hi. Hi, my name is Mackenzie Madry. I'm 19 and I'm from Northwest Arkansas. My journey with osteosarcoma started in 2020. At the end of my sports season, I was in soccer and cross country, and we were just ending our season, and I noticed this bump on my leg. It started out nothing, and it started to get a lot more painful. I think I started noticing a problem when I went out for a run with my team, and I came back having to walk the second half of it, or walk more than I normally do because running is hard. That was one part, and I also did orchestra in the morning. This was COVID times and towards the end of 2020, so I did part online school and mostly sports and music in person. So in the morning, I'd be in orchestra for 90 minutes sitting there. And by the end of it, I would have trouble standing up and straightening my leg and getting to the door. And I noticed I was taking a lot more time than my classmates to get out the door. We were eventually able to see a doctor. And we thought it was a sports injury because it was just the end of sports. So I went to PT, and they kindly directed me to somebody that could help, and got x-rays, and went to Arkansas Children's in Little Rock, where they diagnosed me with osteosarcoma in my left femur. We started chemo like right away, about 2 weeks after my diagnosis, 2 days before Christmas. So I spent Christmas Eve in the hospital and was discharged Christmas Day. And then I was able to drive home, 3 hours home. I don't remember a lot from that Christmas. I slept the car ride home, was able to stay up long enough to do presents, and I was right back asleep. I had 4 more months of chemo before I had surgery. In my left femur, I had part of my femur taken out and had a cadaver bone to replace that. Then I had the rest of my chemo, thankfully in Northwest Arkansas, when they had enough nurses at that hospital certified to give chemo. MAP therapy was really hard on me. I've had hearing loss from cisplatin, and I had cardiomyopathy from doxorubicin. After my treatment ending in September, I was cancer-free. And then about a year later, my leg got infected. So we had to take the cadaver bone out and replace it over a span of 3 surgeries, 2 to clean and debride, and then 1 that happened around a month later. I went home from that surgery, getting another cadaver bone placed. And after about 2 or 3 days being home, I was right back in the hospital and I ended up at end-stage heart failure. Between the chemo and those surgeries, there was a lot of stress on my heart. And so I got flown to Arkansas Children's in Little Rock and they treated me there. I was not eligible for a heart transplant because I was not 2 years cancer-free. So we didn't have a lot of options and I ended up getting a heart pump placed in my heart. And I wasn't eligible for a transplant for another 11 months. And then those 11 months came around and we started the evaluation to get a transplant and we noticed some spots on my lungs. So we had to wait another 3 months to see if those would grow. Thankfully, they didn't, and I was put on the heart transplant list. But then screws broke in my leg and I had to be put on hold to have that surgery. And then I was back on the list. Thankfully, I had surgery, a transplant in 2024, in June 2024. And I had a new heart. It's really hard to think that the chemo that saved my life also damaged my heart so much that I had to go basically through another set of treatment just to survive again. I had a heart pump for almost 2 years before I had a transplant. So I was living with something else sustaining my life for a really long time. And it was really hard for me to deal with that. When I had first came in with heart failure, they weren't sure if I was going to make it to my next birthday because I wasn't even on the list. There wasn't end of the tunnel anywhere in sight. So that LVAD was, or the heart pump was really only thing keeping me alive. I am now about a year out from transplant and 4 years cancer-free. And just one last thing I wanted to say, I would like to thank everybody for coming here today to make a difference. Most patients don't really think about how much the odds are against them when they go through treatment. Once they get out of it and they start doing research on their own, they realize that some of the drugs they were treated with are over 50 years old. And then they start to think of the what ifs. What if we had newer drugs, kinder drugs? Would I have lost some of my hearing? Would my heart have ever failed? This is some things we carry with us through everyday life. When we can't keep up with our friends walking the mall, when it takes us a long time to go upstairs because our knees don't work like they used to anymore. When we have to ask people to repeat themselves over and over again because we can't hear what they said. And when we have trouble standing on the beach because the neuropathy in our feet is making us feel like we're standing on the core of the earth. These are things we carry with us every day. And we also carry with us the people that are no longer here. So I wanted to thank you guys again for being here and wanting to make a difference and change things for people that are coming after us. Thank you.
Thank you, Mackenzie. Now joining us by Zoom is Michaela Nyland. Make sure that's true. Hi, I'm Michaela. Michaela has been through years of treatment for osteosarcoma and is now facing a recurrence. Despite that, she continues to attend school and finds time to lend her voice to efforts like this one, to make sure future patients have better outcomes and options and fewer burdens. Michaela?
Thank you so much. Um, I was getting ready to go on a trip with my family, but my ankle had started hurting a lot. Much to my dismay, I thought I was going to the doctor for a sprained ankle. Um, I went in for an x-ray and my doctor immediately said that he was calling Children's Hospital to set up an appointment for me. He never said exactly what he thought it could be. Myself and my parents went to the appointment at Children's for a biopsy, and the doctor that was going to handle the procedure came in and said that he was 99.9% sure that it was cancer, osteosarcoma to be exact. He was going to do the biopsy and place my port, and I immediately just began to cry. I didn't know how I was going to get through this. And that was in the beginning of July in 2020. So COVID was pretty rampant. Since COVID had the country shut down and I just learned that I had cancer, I was terrified. Everything that I was doing before I got sick was taken away from me and everyone else due to the global pandemic. I wasn't able to meet other patients. Everyone was wearing a mask. I would have liked to see their whole face for some type of reassurance. At my first treatment, I had no idea what was going to happen. How was it going to affect me? How soon was my hair going to fall out? Normal questions for anyone, but all we had was a 3-ring binder with the drugs that I was going to be receiving. Sure, we could ask questions, but I would have liked to talk to other kids and see how they were handling the chemo. Maybe I was worried that I wouldn't be able to handle it. I started the standard MAP treatment for osteosarcoma in August. By my 11th birthday, I had no hair. I learned that I was going to have to make some really tough decisions regarding my health. My parents held my hand, but not once did they tell me what I had to do. I did all the rounds of chemo that they said I needed before I had to decide to amputate or receive a limb salvage. After the PowerPoint from my orthopedic doctor, lots more tears because because of the choices that I had. And I had a day or so to decide. I made my choice to amputate. Either way, I wouldn't be able to move my ankle. I felt like if I had amputated and removed this tumor, I would have a better chance at beating this disease. October 5th, I had my amputation. I also knew that I had nodules in my lungs, and so in November, I had a double thoracotomy, and we spent Thanksgiving in the hospital. I finished all remaining rounds of chemo in March of 2021 and was in remission for 9 months. Fast forward to relapse after relapse, surgery after surgery, moving treatment from Colorado to Texas and to Ohio. I have had a total of 4 thoracotomies after my amputation, radiation on my lungs, since surgery was no longer an option, 2 brain surgeries to remove tumors, more radiation on my lung, arm, and my good leg. This last radiation was so hard on me that I just went back to school last week. I have gone to a total of 8 days of my junior year. I had to rearrange my school schedule and give up one of my college classes. The thought of making it to the end of the day isn't in the cards for me right now. I'm still taking chemo in hopes that it will kill this thing that keeps taking everything away from me. I'm 16. I have dreams and aspirations, and I want to make sure that others receive a better standard of care.
Thank you.
you.
Thank you, Michaela. These young ladies' perspectives bring us back to the purpose of this workshop, to connect policy, science, and humanity so that stories like theirs lead directly to change. We're now going to turn from the patient perspective to the family perspective, because osteosarcoma doesn't just hit the patient, it hits families. Oftentimes, it hits a family in the middle of other dynamic changes. It's often a teenage patient that is working toward autonomy, and parents and other siblings who are thinking about them soon being out of the house. And many of us know those dynamics are enough. And into that is introduced for the first time for most people, the word osteosarcoma. We'll now hear from Mike Egge, whom, by the way, I want to thank for providing this facility for this meeting. But Mike's going to talk to us about what it's like for a family to face an osteosarcoma diagnosis and make treatment decisions in real time. Mike, your daughter Olivia is now a survivor and a first-year medical student. But take us back to the days when you first heard her diagnosis. What was that like, and what decisions did you have to make fast? Just take us through that.
Yeah, so thank you, Mac, and thank you, Mackenzie and Michaela. What I have to say is pales in comparison, although you do bring back some very, very strong memories, because my daughter Olivia, who like you both, was a very active teenager. She was 16 years old, a sophomore up here at National Cathedral School in Washington. An athlete, a competitive gymnast. It just seems it's uncanny how much this disease affects very active children in sports, especially, at least in experience we've had meeting people since. And so when February 3rd, 2017, I'll never forget that day. Like you, Olivia had been having pain in her knee and had complained about it over the course of now many months. As parents, we feel intense guilt because we didn't do more about it, but it was, gee, it must be gymnastics. It must be, do some PT. And when it persisted, we discovered it this very same way that you did, Mackenzie. And so when we learned about it, from a parent's perspective, at least my perspective, it was, hey, there's a protocol, and you follow the protocol. You looked at what Dr. Google had to say, or some of the early doctors. It's what you do these days, and you say, hey, it's 2 out of 3. The survival rates. If you're like me, you're a cup half full guy, and you figure, let's be disciplined about this. We've got great advice. Let's do the protocol. And then about a week in, the fallacy of statistics started to kind of meld in, because that 60% isn't, or 2 out of 3 isn't really 2 out of 3. If you have metastatic disease, it changes dramatically. We learned a lot about the fact that life would never be the same for Olivia, whether it's intense chemotherapy, as Mackenzie well described, or amputation or limb-sparing surgery. It's about 100% odds that you're going to have a disability for life. And Olivia's no exception in that. She has the same hearing problem. She was spared the heart problem so far, but it is the toll of this protocol is intense. The dosing of the chemotherapy here, it's because it's children. They're resilient. They can handle it. This is dosing that most people in this room, at least above the age of 50, my age, could not take, could not survive. It's dosing that requires you to go to the hospital to be monitored. It is dosing that will result in many midnight runs to the hospital, as many here know well. The protocol, then you learn, is 40 years old. That was a really tough one because at first, you know, it's what do I have to do? And they tell you what you got to do. It's going to be chemotherapy, surgery, resection, and then more chemotherapy, the MAP protocol. And you figure, okay, we'll do that. But then you find out that there has been no advancement in God knows how long. That was a punch in the gut that came within about a week. The other dark truth is genetic testing is not standard of care. We all hear about these wonderful developments. We got ads on TV every night about you know, this or that precision medicine that attacks things that are derived from mutations and the like. Genetics is the core. You know, you learn pretty quickly. It's the path to cure for cancer. This is just this dumb lawyer's rendition of this, but the fact then that I had, you know, to learn not through doctors but through other parents that, gee, you should get that tumor, you should get her cancer sequenced, and that it's not standard of care in a world where data is king is enraging. Then we went, but we still follow the protocol, and the purpose of the protocol is to do resection a few months in, is to see, we were told, how well your tumor reacted to the chemotherapy. Part of it is also because just the way surgical science works, the early days with limb sparing surgery, you couldn't have an endoprosthetic in place in time. So we did that. Well, then we learned 30% necrosis, which is a terrible result. At least that's what we had been told, and that the prognosis for Olivia was bad. So we felt like we wasted. We didn't waste, but 2 months doing this instead of maybe getting the tumor out of her body immediately, and felt pretty frustrated by that. So, but then the question is, okay, what do we do now? You just told me there's 30% necrosis. We need to do something different. Well, the answer was, sorry. There's the protocol. The protocol to me was an incredibly dark place at that time. And they'll tell Olivia, hey, suck it up, follow through with this terrible experience with chemotherapy, because that's all we have right now. Was not very satisfying. We did end up importing MEPACT from Europe. Again, something that doctors here did not tell me about. I had to find on my own. In terms of research, they don't do it because it's not FDA approved, and they don't push options that are not FDA approved. As a parent, that's unacceptable. I needed to know everything because I'd go to any end of the world to save my daughter's life, and I was frustrated by that. So, Mac, that's a long answer to your question. It was shock, bewilderment, and then later, frankly, rage, and it's I think you're sensing that in this panel.
Thank you, Mike. Our third speaker is Ann Graham, who will help us think about what matters to patients themselves in the context of clinical trials, and especially adolescents and young adults navigating these trials. And with your own diagnosis at age 43 of osteosarcoma, and as founder of the MIB Agents and Osteosarcoma Collaborative, you probably got the broadest experience with osteosarcoma patients of anyone in the country. What does that experience tell you that matters most to kids facing this disease?
Going after Michaela and Mackenzie, like, I'm not sure exactly how much I have to add because they've really told you how it is. And after listening to Michael from a parent perspective, it's awful. It's, I'm going to go with bloody awful. It's really, it's a street fight all the way through. And what you can see from Michaela and Mackenzie as well is we, as a community of patients, are desperate to help improve outcomes. We want them to be improved from our perspective as patients, but we really want those outcomes to be improved for our friends, because we all get together, we all know each other, and we all feel loss greatly. Whether that's a heart transplant, whether that's somebody who went from limb salvage to amputation, our friends are suffering all the time. So I think the biggest thing is, and it's something, I'm just going to put this out there up front. It's something that everybody who's here and online is here for. We are all here to reduce suffering in the world. If there is a greater cause, I don't know what it is. You all are doing this, and we're doing a good job. We just have to do it better. That's, I think, the message from our junior advisory board members, from doctors and researchers. We all want better, and that's the bottom line.
Thanks, Ann. So that brings our discussion to research and the funding landscape, and how that landscape today is part of the barrier to progress. Justin Ingram of Alex's Lemonade Stand is with us, and Justin has a remarkably broad and unusual vantage point from having worked in biotech and pharma on drug development to now leading scientific programs for Alex's Lemonade Stand Foundation. And Alex's is, for those who may not know, funds research across the gamut of pediatric cancers. So Justin, from that perspective, what do you see as the main challenges and barriers that make progress in osteo so difficult?
Yeah, thank you, Mac, and thank you everybody for attending. And it's amazing to be able to speak with all of you today. I think what's really challenging about osteosarcoma is how complex biologically osteosarcoma is, especially compared to other types of tumors. Osteosarcoma is very heterogeneous, meaning many cells within the same tumor actually aren't necessarily exactly alike, meaning if you target many of these cells, other cells within the same tumor may grow out, leading to treatment with other types of inhibitors and other types of treatment options. Further, the development of targeted therapies within osteosarcoma has been challenging just due to how biologically complex these tumors are as well. And there's been great progress made in identifying targets that could potentially be brought forward. And I think a lot of drug discovery efforts could continue in this space to help develop targeted therapies. At Alex's Lemonade Stand, we did fund 2 large projects helping to help answer these questions, called our Crazy 8 initiative, which are $3 to $5 million projects helping to help answer these questions. But I think the complexity biologically of osteosarcoma has really been challenging.
Reaching back to your industry days, what do you think would move the needle in terms of companies' willingness to invest in cures for osteosarcoma?
Yeah, I think it's finding widespread targets that could be applicable to other types of cancers as well, or looking into adult therapies, seeing if they could be essentially also applied to osteosarcoma would be really beneficial. So along with developing new therapies, also looking at what is existing and seeing if that could apply to osteosarcoma would be very helpful as well.
Okay. For the panel generally, if with this assembled group and particularly the FDA, who's going to get their chance in a minute, what one thing would you like to see changed in this environment? Not necessarily how would you change it, but what one thing do you think is the most important to focus on for progress?
I think it's just continued collaboration. I think events like this where everyone can be in one space and communicate and work together would be really beneficial. I think partnerships between industry and academia are always helpful and could continue. And I think just more collaborations in general. So I would say just continuing to work together, continuing to exchange ideas, and continuing to foster ways to enhance drug development within osteosarcoma would be really beneficial.
Mac, I'd argue my one thing would just be where the conditions are right, faster paths to approval. It's as simple as that. This is a race against time, and we have to think out of the box in order to get treatments to the Mackenzies and the Michaelas of the world fast enough.
I'm gonna add a kind of a little bit of a different perspective. Yes to all of that, first of all. Yes to convening. Yes to being face to face with the people who are helping. I will add to that. I think meeting Mackenzie and Michaela, other kids who are suffering with osteosarcoma or who have come out the other side from osteosarcoma. I can't tell you at our own conference and conference like these, I'm sure I will hear the same thing. I hear researchers regularly say to me after meeting young adults on our junior advisory board, I knew the work I was doing was important. Now I know why. So I really feel strongly that having, in AI, there's a phrase, having a human in the loop. Right. So when you're anything you're undertaking with AI, you can't sort of download it and send it out in the world. You have to have a human in the loop. It's the same with this disease. Please find a human in the loop and we can help. We have a huge community here of experts in every possible way. I'll add that we have, of course, our junior advisory board, which is made up of 25 young adults who have experienced osteosarcoma. Some are in hospice care, some are in treatment, some are survivors. And then we have another group of 21 to 25-year-olds who are pursuing a career in medicine or biotech. And this group can tell you what it's like. They can help inform clinical trials. We have an actual group called Research Bee, where if you're undertaking a clinical trial, connect with us, get the patient perspective up front. Your trial will be more successful. It will accrue better if you have a patient in the loop. It can be the patient family in the loop. That's also important. Bereaved parents, really important. But the kids themselves, the young adults who are desperate for better for themselves and for their friends, are really all about telling you like it is. And they have two jobs. One is to be honest and the other is to be kind. So to share their experience and what would make them want to be on a trial, what would make them want to stay on a trial, what information do they need, how do they consume that information, what are the deal breakers, and getting that from them. Like I was just talking to the junior board a couple of days ago about this in preparation for this. And one of the funniest and truest things that I heard was from Walker, who's a first-year medical student at University of Kentucky. He said, here's the thing about clinical trials, or pretty much anytime you're in the doctor's office, you're really like Charlie Brown listening to the teacher, where you're nodding your head and you're trying to take it in, but all you're hearing is wah, wah, wah. So we need to create ways that young adults especially, but of course their parents, but that young adults can consume information in a way that's digestible for them. That's a way that they're used to consuming information. The 3-ring binder, helpful over the long term, but up front, that is really hard to take that in. You kind of, people are different. You need it in small bites. You need it maybe from a friend. You need somebody else who's been on that trial, preferably. If you don't have that, then let's have a video. Let's have, weekly webinar and podcast where we feature people who are undertaking clinical trials. There's so much that can be done to make a trial successful. And we are literally, as a community, all here to support that. And one last thing, thank you for convening this group. We are so grateful at MIB Agents to have partnership and love between our two organizations. It would be difficult to do this work without Osteosarcoma Institute. It's just great that we have these moments where we can all get together. We ask for collaboration from you, and I'm grateful that we get to collaborate together from our perspective. So thank you.
Thanks, Ann, and thanks all the speakers. We're now going to turn to Dr. Amanda Marinoff and Dr. Jasmine Smith to discuss the current clinical and regulatory landscape.
Good morning, everyone. It is really such an honor to be here with you all. I'm Amanda Marinoff, a pediatric oncologist at UCSF. And wow, what a wonderful panel and way to start off this session, and a hard act to follow. But thank you for the opportunity to present an overview of current therapy and ongoing clinical trials for osteosarcoma. I have no disclosures. I think our panel really beautifully captured what the current treatment is like for osteosarcoma, but we'll formalize it a little bit here. As you heard, patients with newly diagnosed osteosarcoma undergo multi-agent intensive chemotherapy and surgery. And in North America, the standard you heard is MAP, or high-dose methotrexate, doxorubicin, and cisplatin. You can see what this roadmap looks like at the bottom of the slide. Patients typically receive 10 weeks of preoperative MAP chemotherapy, followed by local control surgery, and another 19 weeks of MAP. And I think you heard from Michaela and Mackenzie so eloquently, and as Anne summed it up, this regimen is bloody awful. This regimen does not incorporate any molecular or clinical factors to guide risk or biology-tailored therapy. So this means that all patients essentially receive the same therapy regardless of their clinical presentation or underlying biology. And using this approach, we know that some patients are likely overtreated and exposed to sometimes lifelong toxicities. And on the other side of the spectrum, we know that we're still failing too many patients. Our 5-year overall survival is 65 to 70% with patients for localized disease, and under 30% for patients with metastatic disease. And usually cure in this context is only possible by resecting metastatic lesions, often in the lung via thoracotomies, and as you've heard today, often requiring multiple serial surgeries. For relapsed and refractory disease, there is no standard of care, and our outcomes are dismal. Beyond resecting all metastatic lesions when feasible, our salvage chemotherapy regimens that we commonly use include ifosfamide with or without etoposide, gemcitabine, and docetaxel. And while these sometimes achieve some disease stabilization, they're also toxic and rarely curative. Neither our approach to treatment nor our survival has improved in nearly 4 decades. On the left, we have the results from a landmark clinical trial that was published in the New England Journal of Medicine in 1986 that established that chemotherapy postoperatively is unequivocally beneficial with an event-free survival at 3 years of 66% compared to 17% among patients who did not receive chemotherapy. Then we fast forward to the Uramos trial that was published in 2019, and we see that the event-free survival is nearly identical. The URAMOS trial tried to move the needle using two interventions, one adding interferon to patients with good histologic response to chemotherapy and adding ifosfamide and etoposide to patients who had a poor histologic response, and neither improved outcomes. And so you've heard that we're still using the same regimen that we have been using for decades. We've highlighted a number of key barriers to progress that I'll just briefly run through here. Osteosarcoma is really a perfect storm of challenges. It occurs in a young population that's a rare disease with extreme clinical and biological heterogeneity. You've heard that the genome is highly complex, made of structural variants that are not readily targetable. We have limited access to novel agents, no validated prognostic or predictive biomarkers, and the fact that it's a bone tumor actually poses challenges to collecting good quality biospecimens, and that's a real hurdle to being able to really advance our understanding of biology. It also poses some challenges to interpreting the radiographic response to therapies and challenges our ability to use endpoints, which you'll hear more about from Dr. Janeway today. Before we jump into the ongoing clinical trials landscape, I wanted to just provide a snapshot. There's a recent review that nicely covered the evolution of phase 2 clinical trials over the last couple of decades and identified some areas of progress and a lot that still needs to change. On the positive side, we've seen that most trials are now including younger patients, some osteosarcoma-specific endpoints, including progression-free survival at 4 months, and a shift somewhat away from cytotoxic chemotherapy and toward targeted therapies and immunotherapy. What hasn't changed? We know that most trials are still failing to show benefit, with only 20% meeting their primary efficacy endpoint. Median objective response rates hover above around 5%, with a median progression-free survival of 3 months and median overall survival of only 10 months. And trial design is really geared towards signal finding and not regulatory approval, with most single-arm studies looking at monotherapy. These are small trials, and you'll see in the upcoming slides, there's a lot of variation in design, endpoints, and eligibility requirements. With that context, we'll turn to a review of the ongoing clinical trials in osteosarcoma, which I'll review based on their mechanism of action. This is not an entirely exhaustive review, but hopefully will give you a picture of the most salient trials that are going on, and some historical context to really help frame how we got here today. Most of the talk I'll spend on novel agents, but did just want to highlight that our old tools, chemotherapy and surgery, are still on the scene. There's some trials looking at new combinations of chemotherapy and certainly interest in combining it with novel agents, and looking into new surgical approaches. There's a trial, AOST 2031, through the Children's Oncology Group, looking at our standard open thoracotomies versus minimally invasive thoracoscopic approaches to removing pulmonary metastatic disease. We'll move on to our first set of novel therapies, the multi-tyrosine kinase inhibitors. And this is a class of agents that target receptor tyrosine kinase signaling involved in tumor growth, metastasis, and angiogenesis. And over the last decade or so, there's emerged a number of trials looking at these agents. And here's a review of those studying single agent multi-tyrosine kinase inhibitors. Sorafenib was the first to be published. Apatinib is available in China, regorafenib, and cabozantinib. And most of these were single arm studies, with the exception of regorafenib, where there were 2 parallel studies, one in the US and Europe, that were randomized placebo-controlled trials. And I'll draw your attention here to the bottom line of this table, where we can look at 4-month progression-free survival. And here we can see that it's above 40% for most of these trials, which is a meaningful improvement compared to historical controls. On the right, cabozantinib has drawn particular attention for its 4-month progression-free survival of 71%. But notably, when we look at progression-free survival with cabo at 6 months, it drops off to 33%. And indeed, this is true across the board where there is some modest benefit, but the duration is short-lived with median progression-free survival ranging from around 3.5 to 6.5 months, and objective responses are infrequent. But all of these studies met their primary efficacy endpoints, and in general, multi-tyrosine kinase inhibitors are really the first class of agents to show activity beyond chemotherapy in decades. There have been several studies looking at TKIs in combination with other types of therapies. Here I'm highlighting a few, excluding immunotherapy, because we'll discuss those later on. Sorafenib in combination with everolimus showed a 6-month progression-free survival of 45%, but did not meet its predefined efficacy threshold. There was a study of pazopanib and topotecan looking primarily in patients with soft tissue sarcomas, but there was an exploratory cohort of osteosarcoma patients comprised of 15 patients where the 3-month progression-free survival was 70%, and there were 2 partial responses. And then in the last row, we see a study looking at ifosfamide and etoposide with and without lenvatinib, and this is the randomized international OLI trial, where ultimately there was no statistically significant difference in the 2 arms with no benefit to lenvatinib. And so overall, we can see that there's some modest activity in some of these studies, but hard to know which patients are benefiting. There are several active multi-tyrosine kinase trials that are ongoing now, including upfront phase 2/3 study of MAP with and without cabozantinib that we'll discuss in a little more detail on the next slide. And then there are some ongoing early phase clinical studies, including high-dose ifosfamide and cabozantinib maintenance therapy in a couple of different contexts, looking at checkpoint inhibition in combination with cabozantinib, and a new multi-tyrosine kinase inhibitor called tanzalintinib that will open soon. And these are primarily for relapsed and refractory disease. The Children's Oncology Group study, AOST 2032, is the first frontline therapeutic study in a couple of decades. So I want to spend a couple of minutes just talking about what this is and how the study is designed. This is a feasibility and randomized phase 2/3 study of cabo with MAP for newly diagnosed patients with osteosarcoma. The feasibility portion of the study completed and showed that the combination of MAP given concomitantly with cabozantinib was feasible. And now we've moved on to the efficacy portion, which is randomized and randomizes patients to receive MAP with or without cabozantinib. Patients are stratified onto cohorts with standard risk or high risk, the latter of which is defined as metastatic, pelvic, or unresectable disease. And importantly, this standard and high risk is really for the trial design and not to guide risk-adapted therapy. And the primary endpoints here are feasibility and event-free survival. Here I wanted to bring up a few key points that came out of the Pediatric Strategy Forum on multi-tyrosine kinase inhibitors. This was a meeting that brought together multiple stakeholders through the Accelerate Consortium in collaboration with the FDA and the EMA. And they drew out several conclusions looking at tyrosine kinase inhibitors, but I think they're really applicable across the board to all of the classes of therapies that we'll discuss today. The first is that predictive biomarkers are poorly understood. The ideal characteristics of tyrosine kinase inhibitors are not defined. We often sort of lump them all together, but it's certainly possible that they work differently or better for some patients and can be better tailored. Trials should include children, adolescents, and adults when the biology is shared. Feasibility and combination treatment should be evaluated within the same protocol as a randomized trial when feasible, as we saw in the AOST 2032 schema. And trials should be designed to support regulatory and payer requirements. We'll next move on to immunotherapeutic approaches in osteosarcoma, which have not revolutionized the landscape here as they have in other cancers. And this is thought to be due to the fact that osteosarcoma is considered an immunologically cold tumor. There's not a lot of new epitopes that are presented and has the ability to exclude the immune system's ability to attack the tumor. And yet there is some evidence dating back to the early 1980s the immune system may indeed have a role. In 1981, William Coley famously injected osteosarcoma tumors with bacterial toxins and induced some regressions. And so there's ongoing interest in trying to leverage the immune system to try and induce responses. And so we'll talk about a few different categories, starting with immune checkpoint inhibition. We'll briefly review completed trials of single-agent immune checkpoint inhibitors. Easy bottom line here, if we look at the outcomes, there were no responses to any of these single-agent PD-1 and PD-L1 inhibitors. Phase 1 studies did show that they were safe. And so to try and address these limitations, there were combination studies that were tried, including dual checkpoint inhibition with nivolumab and ipilimumab, which also induced no responses on the top row. But in the bottom three, we can see that the combination of checkpoint inhibition and tyrosine kinase inhibitors seems to produce some responses in some patients, and some of which seem to be somewhat lasting. And so while the activity seems to be modest and these trials did not meet their pre-specified efficacy endpoints, it looks like there may be a subset of patients who may benefit from this therapy. And there's ongoing correlative biology work to try and tease out who are the right patients for these therapies. There are several active immune checkpoint inhibitor trials that are ongoing now, including combination of atezolizumab, a PD-1 plus cabozantinib, another PD-1 and another TKI, and then some novel strategies, including CD73 inhibition, looking at a demethylating agent, azacitidine, and combining PD-L1 inhibition with radiation for patients with pulmonary-only sarcoma. Beyond checkpoint inhibition, there are a lot of other strategies that are being employed to try and activate or enhance the immune system's innate and adaptive responses. And several have been completed that I'll review here. Interferon alpha, as we discussed in the Uremos trial, showed no benefit compared to MAP alone in patients with good response. There was a phase 2 study of aerosolized GM-CSF in patients with recurrent pulmonary osteosarcoma that also did not demonstrate efficacy. And the most extensively studied agent that was brought up in the last section is mefamotide or MEPACT. And I'll try and very briefly summarize some of the data from 4 clinical trials in newly diagnosed patients with osteosarcoma. INT 0133 was a study that used a 2x2 factorial design and randomized patients to receive MAP with and without mefamotide and or ifosfamide. And in this study, we found that the patients had an overall survival benefit who received mefamotide, but there was no statistically significant event-free survival benefit. Due to complexities with the way that the study was designed and analyzed and reported, there are difficulties in interpreting this data, and really there needs to be more work to definitively conclude whether MEPACT has a clinical benefit. But based on these data, it was approved by the EMA and is utilized in several other European countries. It was not approved by the FDA. More recently, there was a report that combined 2 European studies, one in Italy and one in Spain, to look at the impact of mefamotide in that context. And the study found that in these 2 trials, there was an improved event-free survival, but only in the univariate analysis, and this was not statistically significant in the multivariate analysis. And then there's an ongoing study called Sarcome 13 in France, looking at MEPACT for patients with metastatic disease or poor histologic response, and those results are still pending. There are several active trials of other immune modulatory agents, and there's a pretty wide range of the types of strategies that are being used that I'll highlight here. There's aerosolized IL-2 cytokine therapy for patients with lung metastases, a listeria-based HER2 vaccine, a TGF-beta inhibitor called vacdacertib, an RNA lipid particle vaccine, and a STING agonist to activate the innate immune system. These are early phase studies, but I think really kind of showcase the broad range of strategies that are being trialed. We'll next move to adoptive cellular therapy and antibody drug conjugates. These are distinct therapies mechanistically, but I'm grouping them here together temporarily because they share the commonality that they can both target proteins that live on the cell surface or the osteosarcoma cell surfaceome. This is from a nice review that highlights some of the key targets that are being investigated both preclinically and in the clinical setting, and we'll review some of them in detail in the context of the trials. Here are some of the recent ADC trials. There was an ADC of glembatumab vedotin and a HER2 targeting antibody, both of which showed minimal responses. But the bottom three trials showed some early signals of efficacy. There's an LRCC15 targeting ADC in patients with advanced solid tumors, all comers, and there were 4 PRs that were noted in the osteosarcoma and undifferentiated pleomorphic sarcoma cohort. This was phase 1 that was really meant to determine safety. There's a second trial of a CAB-AXL ADC, again in advanced sarcomas, but there were 11 patients with osteosarcoma, and these patients experienced a 3-month progression-free survival of about 46% with 2 partial responses. And finally, there's a B7H3 targeting ADC. This was from a trial that was conducted in China, in this should say osteosarcoma, where the response rates at the higher dose tested were somewhat impressive, with 10 out of 10 patients having disease control at a median follow-up time of 4 months and 2 partial responses that were induced. And so these are early days for ADCs, but given the high unmet need and the early signals of activity are of interest to the community and warrant continued investigation. Targeting some overlapping surfaceome proteins. We have chimeric antigen receptor therapy or CAR T cells. I've listed several targets here. There are completed trials targeting GD2 and B7H3, which demonstrated feasibility and safety, but limited responses to date. And certainly there's ongoing correlative biology to try and understand both mechanisms of response and resistance. There's an EGFR targeting antibody, and then one targeting the folate receptor, which is specifically overexpressed in osteosarcoma, and then a CAR CD70 with augmentation by IL-15 cord blood-derived natural killer cells. Beyond CAR T cell therapy, there are several other active cellular therapy trials. These are primarily being studied in basket trials across all pediatric advanced solid tumors, but I think a lot that we'll learn from this, including autologous tumor infiltrating lymphocytes, a T cell membrane anchored tumor targeted IL-2 therapy, donor NK cells in combination with chemotherapy, NK T cells in combination with checkpoint blockade, donor immune cells in combination with chemotherapy and dinutuximab, which is present on osteosarcoma and neuroblastoma, and afami-cel, which is a MAGE-A4 T cell receptor therapy in patients who have this marker on their solid tumors. Here's just a summary for an at a glance for your reference. And then we'll move forward to DNA damage repair inhibitors. At a very high level, this is a coordinated network that's really designed to help cells survive genomic instability. And what I want to just highlight here is that this is not a single pathway, but really a network with multiple druggable nodes. And traditionally, these therapies have been utilized in the context of cancers with BRCA mutations, which are not common in osteosarcoma, but there's emerging data suggesting that osteosarcoma has similar biology at multiple levels, and emerging preclinical data supporting these strategies in the lab and moving them toward the clinic, which you'll hear more about from Dr. Sweet Cordero. And there are 2 recent trials targeting DDR in osteosarcoma, one combining PARP inhibition with ATR inhibition. And here in the cohort of patients with measurable disease, the 4-month event-free survival was 13.5 with one partial response. This did not meet its predefined endpoint, but we see that there may be a subset of patients who may respond to this therapy. And the cohort of patients who had resectable pulmonary-only disease is still undergoing analysis. And then there was a recent phase 1 study combining gemcitabine with a WEE1 inhibitor, and this was well tolerated with early efficacy showing 18-week event-free survival of 39%, and this will move forward to a phase 2 study. And there are ongoing efforts to develop new trials targeting DDR and to explore predictive biomarkers. And as with all of our trial development, this is really going to be driven not only by the science, but also access to novel agents. And finally, we'll briefly discuss targeted inhibitors and do a deep dive on a trial that really showcases how solidifying partnerships can really make progress possible. Here we can see that there are several targeted agents, a basket trial with CDK4/6 inhibition, and then a Wind pathway inhibitor in all solid tumors, and one specifically in osteosarcoma. And what I really want to spend time on is the OMO 103 study. This is a MYC inhibitor that was really born out of amazing partnership, advocacy, and collaboration among the stakeholders here. This is a trial that is being supported by the Osteosarcoma Institute and in collaboration with the pharma company Peptomic, and is looking at drugging MYC for the first time in osteosarcoma. MYC-activated osteosarcoma is associated with a poor prognosis. This is becoming more and more evident with mounting studies, and here's a representative Kaplan-Meier curve with patients with MYC-amplified osteosarcoma, shown in blue, having a significantly worse overall survival. And so that makes it a rational target for osteosarcoma, and currently being conducted in Barcelona is this phase 2 pilot study looking at this mini protein that targets MYC's interaction with MAX, specifically in osteosarcoma. Again, highlighting how collaboration and partnerships can move novel agents forward into the clinic. And finally, I just want to say a word on ongoing efforts to define biomarkers in osteosarcoma. We talked about how current therapy is not really based on risk or biology, and that we're seeing some signals in some patients, but we don't quite know who they are. And to really match the right therapies with the right patients. And so there are many efforts to try and address these gaps. I'm showing an overview of one called the Clinical Biomarkers for Osteosarcoma Stratification Working Group that really focused on identifying prognostic markers at the time of diagnosis to classify risk, but these really apply broadly as potential markers to guide the selection of the right patients for the right treatments, and to inform novel targets and novel therapies. And these can include genomic alterations, transcriptomic epigenetic alterations, circulating tumor DNA, and cell surface protein expression. So to conclude, we know that there has been no meaningful survival gains or new treatments approved in approximately 40 years. There are multiple strategies in the pipeline, though definitive breakthroughs remain elusive. Though the landscape really is more dynamic, I think, than it has been in decades. The unmet needs really require collaborative solutions, which is why we're all here today to address the gaps in access to novel agents, biology-driven approaches, innovative trial designs that really balance the need for speed with rigor, and of course, multi-stakeholder partnerships. Thank you so much for being here today, and to the Osteosarcoma Institute and the FDA Oncology Center of Excellence for convening this absolutely needed meeting. Thanks to the leadership at OSI and Amy Loebner for making it possible, and specifically to Laura Davis and Katie Janeway for their mentorship and support of this presentation. And of course, the patients, families, and advocates who make progress possible. Thank you.
All right, good morning, everyone. Thank you, Amanda, for that great talk. It's good seeing you again. Sorry about your foot. My name is Jasmine Smith. I am a pediatric oncologist. I've been at the FDA for a little over a year now. And I work as a medical officer in the Division of Oncology 3, where we do review sarcoma applications, but we also review GI malignancies, melanoma, and superficial cutaneous malignancies. Today, I'll be providing a brief regulatory overview of drug approval pathways, in addition to considerations for rare cancers, such as osteosarcoma, as it relates to drug development. I have no financial conflicts to disclose. So let's begin by first reviewing the FDA's role in oncology drug development. There are a few guiding principles for FDA approval standards. The Code of Federal Regulations is legally binding, and it requires substantial evidence of safety and effectiveness for drug approval. Substantial refers to both quantity and quality of evidence. An important point is that FDA does not regulate the practice of medicine. One implication of that is just because we approve something does not mean that we're endorsing the drug to become the new standard. It only means that it met the regulatory standards of substantial evidence for safety and effectiveness. Additionally, we are not permitted to consider cost when making approval decisions, and we don't regulate off-label use of drugs. Development of oncology products is coordinated by the Oncology Center of Excellence, and this was founded in 2017 to facilitate interaction between the 3 product centers in the FDA. The Center for Biologics Evaluation and Research, or CBER, which is where the therapeutic proteins, vaccines, and cell therapies are evaluated. You have the Center for Devices and Radiological Health, or CDRH, where devices, including in vitro diagnostics and biomarkers, and therapeutic devices are regulated. And then you have the Center for Drug Evaluation and Research, or CDER, which is where small molecules and monoclonal antibodies are evaluated. Within CDER is a department called the Office of Oncologic Diseases, or OOD, which is where I work. We have the greatest number of oncologists at the FDA, and we're divided in disease-focused teams as depicted on the slide. Osteosarcoma studies, as Dr. Donahue mentioned earlier, are generally reviewed by the Division of Oncology 2, where applications for pediatric solid tumors are reviewed. However, as previously mentioned, in Division of Oncology 3, we also review sarcomas, and so there's sometimes overlap. Reviewing oncology trials and oncology drug products, as we all know, can be complex because they involve several different disciplines or people with different types of expertise. Some trials have biomarkers. Some biologic products have complex manufacturing processes that have unique considerations, and all require an understanding of the clinical context and changing treatment paradigms for the diseases they intend to treat. During the review process, we work in multidisciplinary teams, as pictured here, that facilitate collaborative discussion and thorough assessment of each drug application. The FDA is involved in the drug development process from bench to bedside, and sponsors can request to meet with the FDA at numerous time points, which we've outlined in purple. It's recommended that we meet with the FDA early and often. Of note, requesting a pre-investigational drug application meeting, or what we call a pre-IND meeting, is often helpful, as it gives sponsors and investigators time to discuss study design, even before submitting the actual drug application. And even before this, investigators and small companies can reach out to the FDA through Project Catalyst. And so Project Catalyst is an OCE initiative that helps to provide guidance and educational resources to small pharmaceutical companies and academic oncology innovators to help navigate the drug development process. It offers the Oncology Regulatory Expertise and Early Guidance Program, or OREG, which includes some discussions that can be had to provide insight and engagement with OREG's regulatory experts and access to frequently asked questions when it comes to oncology drug development. This is especially beneficial for drug development programs focused on rare diseases like osteosarcoma, as it aims to help avoid any unnecessary challenges in order to facilitate expedited development and availability of new treatments. Next, I will provide a brief overview of the regulatory framework for drug approval and the approval pathways. The requirement for that new drugs show effectiveness is based on the 1962 amendment of the Federal Food, Drug, and Cosmetic Act. And this law requires substantial evidence of effectiveness and specifies that this evidence must be derived from adequate and well-controlled clinical investigations. Under certain circumstances, the FDA can conclude that one adequate and well-controlled clinical investigation plus confirmatory evidence is sufficient to establish effectiveness. It's important to note that the evidentiary standard, though, it's the same for both common and rare cancers. So there are two approval pathways for drugs and biologics. We have accelerated approval and regular or traditional approval. Accelerated approval allows for earlier approval of drugs for serious or life-threatening conditions that address an unmet medical need. The guiding principle for approval is the same for both pathways, but there are, however, a few differences to highlight between the two. So for regular approval, while there's no requirement for the treatment to be better than therapy that is already available, to be granted accelerated approval, there must be a meaningful therapeutic benefit over available therapies. Accelerated approval is based on an intermediate clinical endpoint that is likely to predict a clinical benefit, while regular or traditional approval is based on an endpoint that demonstrates that direct clinical benefit. In oncology, we often use intermediate clinical endpoints that directly measure the tumor itself, like overall response rate or progression-free survival. And these endpoints can be used for accelerated or regular approval, depending on the context. Oncology has by far the most accelerated approvals among the medical specialties, accounting for about 80% in the last decade. And I think this reflects the urgent nature of cancer treatment, the high unmet medical needs, especially in rare cancers like osteosarcoma, and the field's advancement in identifying meaningful biomarkers and intermediate clinical endpoints. In a later talk, my colleague, Dr. Kristen Wassel, will further discuss endpoints as it relates to potential trial design in osteosarcoma. Of note, there are additional considerations as it relates to accelerated approval. Eligibility for this should be discussed during development and prior to submission of the application. At a minimum, the proposed endpoints and design and conduct of confirmatory trials should be discussed. Randomized control trials are commonly required to verify clinical benefit of a drug granted accelerated approval and may be challenging to conduct, especially placebo-controlled studies, if the trial is not underway when the drug is approved. So the FDA generally intends to require that the confirmatory trial be underway prior to accelerated approval being granted, with few exceptions. For certain rare diseases, the clinically relevant endpoints and disease natural history may enable post-marketing studies that do not require randomization to verify the clinical benefit. And this design could reduce the challenge of enrolling and completing the study if it's not underway prior to approval. FDA also recognizes that sponsors seeking approval for drugs intended to treat some rare diseases, especially those with very small populations like osteosarcoma, may face unique challenges with initiating a post-approval confirmatory trial prior to approval. In these circumstances, if appropriate, justification is provided, the FDA may not require that the confirmatory trial be underway prior to accelerated approval. And an important note is that accelerated approval can be withdrawn if the said confirmatory trial is not conducted or it does not verify that clinical benefit. So next, I will discuss some considerations for rare cancers as it relates to drug development. So development programs for rare cancers face many challenges, which may include any number of the listed bullets on the slide. Again, early and careful planning is important for all drug development programs, but especially for rare cancer drug development for these reasons. As noted earlier in the discussion, we recommend that sponsors or investigators meet with us early and often during the drug development process. We also encourage interactions with stakeholders in collaborative settings such as today. This engagement, as we all know, is important. It helps to ensure the research and interventions are designed to meet the real needs and perspectives of patients, which we heard from earlier today, facilitates the adoption and successful implementation of new evidence-based interventions by building that trust and shared understanding, and leverages diverse insights to identify those barriers and really develop effective solutions. You'll recall that I mentioned earlier that drug approval requires this demonstration of substantial evidence of effectiveness, and the risk-benefit assessment must also be favorable. It is important to note that the statutory standards of safety and effectiveness apply to all products, irrespective of rarity of the disease and which approval pathway is pursued. Having said that, I'd like to describe how our regulations to also mandate that the FDA exert some flexibility in our assessment of whether the regulatory standards of approval are met, taking into account things such as rarity and unmet medical need. As you can see, based on the first regulatory citation, we are required to exercise our judgment to determine the type and quantity of information that the applicant should provide in their application to support approval. Not only that, but under the Code of Federal Regulations for drugs intended to treat life-threatening and severely debilitating illnesses such as osteosarcoma, we acknowledge that it's appropriate to exercise broad flexibility in applying those statutory standards while preserving, of course, guarantees of safety and effectiveness. So there are several ways that the FDA can potentially exert this flexibility, as listed on this slide. So FDA can and has exerted flexibility in terms of the number of trials that are required, the design of these trials, the endpoints, as well as statistical considerations of trials supporting approval when and as appropriate. However, identifying when flexibility is appropriate and the type of regulatory flexibility that's warranted is highly contextual and complex. So again, another reason for sponsors to developing products for rare cancers like osteosarcoma to seek FDA guidance early on in development, and particularly at key decision points prior to the initiation of trials that are intended to provide the primary basis of support for their new product. And so expedited programs are intended to facilitate and expedite, as it says, development and review of new drugs to address unmet medical need in the treatment of serious or life-threatening conditions. Drugs intended to treat osteosarcoma may qualify for one or more of these expedited programs. Accelerated approval, as previously discussed, is one type of expedited program. Fast track designation, breakthrough therapy designation, and priority review are other available expedited programs that will be discussed later in a talk by my colleague, Dr. Katie Barnett. Similarly, expedited programs exist for other biologics and for devices as well. And so we encourage sponsors to consider these programs. Lastly, I'll briefly discuss pediatric regulations that have aided drug development for pediatric patients with cancer. And so early pediatric legislation reflected a response to products that caused harm. But more recent pediatric legislation encourages pediatric investigations to inform product labeling. The Best Pharmaceuticals for Children's Act, or BPCA, and Pediatric Research Equity Act, or PREA, are US laws that are designed to improve pediatric drug development and labeling. In 2017, the FDA Reauthorization Act, or FDARA, provided an amendment to PREA that made it a requirement for sponsors to complete pediatric investigations of drugs directed at molecular targets that are considered relevant in pediatric cancer, regardless of what the adult indication is. This slide shows a high-level comparison of the differences between the BPCA and PREA programs. In general, BPCA is voluntary and it provides an incentive of patent exclusivity, while PREA is mandatory and it requires sponsors to assess safety and effectiveness of new drugs and biologics in pediatric patients prior to the submission of a drug application or supplement. The results of any studies, pediatric studies that are performed in response to either a PREA requirement or a BPCA written request must be incorporated in labeling regardless of the results of the study, whether positive, negative, or inconclusive. As noted with FDARA, PREA was amended to give FDA the authority to require certain drugs and biologics with relevant molecular targets in pediatric cancer be investigated in pediatric patients. And this can be met by early studies that are not intended to support a marketing application. And it applies, again, if the adult cancer indication does not occur in the pediatric population. Additionally, orphan drug designation no longer provides exemption from PREA for pediatric studies. And my colleague, Dr. Katie Barnett, she'll provide a brief overview of orphan drug designation in a later talk. Lastly, investigations are required which provide clinically meaningful study data using appropriate formulations regarding pediatric dosing and also safety and preliminary efficacy to inform a potential pediatric labeling. So in summary, I've provided a very broad, brief overview of FDA regulatory considerations for rare cancer product development and with the intent of providing a little bit of context at the beginning of today for osteosarcoma drug development. So we at the FDA, we acknowledge the difficulties associated with drug development for rare cancers such as osteosarcoma, and we're vested in working with stakeholders to minimize and overcome these challenges. The OCE has multiple programs and initiatives that can be leveraged through collaboration to help with overcoming these challenges. Regulatory approval standards requiring substantial evidence of effectiveness are the same, whether a cancer is common or rare. However, the FDA can exercise flexibility in our assessment of whether these standards are met, taking into account things relevant to osteosarcoma, such as rarity and unmet medical need. So we aim to work as collaborative partners as we are today with sponsors, investigators, and other stakeholders by encouraging early and frequent discussions and interactions to support osteosarcoma drug development programs. So on this slide, I've provided a few relevant FDA resources for your review, including some guidances and then OCE programs and initiatives that we have. And these can be found on the FDA.gov webpage as well. So I would like to thank all of my colleagues at the FDA for their support and guidance with this presentation, and as well as everyone at the Osteosarcoma Institute for their collaboration for this very important workshop. Thank you all for being here. Special shout out to the patients. I love hearing patients talk. So thank you guys for sharing your stories. And thank you everyone for your attention. I'll now pass it over to Dr. Alejandro Sweet Cordero for the next presentation.
Great. Well, first I wanted to thank the organizers of this event, OSI. Oh, sorry. Can you hear me better now? Is that good? For this amazing event. I think it was really special, obviously, to hear from the patients and really lay out what we need to do to change the current situation. Grateful to be here. My name is Alejandro Sweet Cordero. I'm a professor at the University of California, San Francisco. Some of what I was going to talk about today was already touched on by a wonderful colleague at UCSF, Amanda Marinoff, so I can skip through that and hopefully just have this be a little bit more of a dialogue and my thoughts about kind of where we are in the preclinical space, since that's mostly what I focus on and the things that I think we can try to bring forward. So going to talk, piggyback a little bit on what Dr. Marinoff talked about, stepping back a little bit to kind of where we are in the lab, trying to bring new things forward. Obviously, I don't need to remind everybody, but there might be a few people here who don't think about this every day. The disease that we're focused on, it's a rare cancer. We struggle with some of the kind of clinical characteristics of the disease that make it hard to study. The rarity, the fact that it's in the bone, the fact that it's quite heterogeneous in terms of its presentation and the localization of these tumors. And one of the things that's been a major focus of my lab's work is trying to access the clinical samples so that we can bring those to the lab and study them. And we've put a lot of effort into, like many other people in the room, into the logistics of doing that. And so that's one of the major challenges, is that patients come in, obviously they need a diagnosis. They don't want to wait. Research is not top of mind for most people. I think Mike Yagi really demonstrated that really clearly with his experiences with his daughter. It's a week later, or maybe 2 weeks later, or a month later, when you sort of are thinking back about what happened when the patient came into the hospital to be diagnosed. So you start to think, well, what happened to that tissue that was taken out of my child? And so that's where the space that we work in a lot, and we try to put a lot of effort into that, is coordinating the logistics of being at the operating room, talking to the surgeons, talking to the families. So obviously, the last thing they want to hear about is another consent that they have to sign, to get tissue, to do something with that. And so that's been a major effort for us. It's been a major effort in a lot of other centers. I think one of the major issues is that, at MSK, at UCSF, at Dana-Farber, there are the resources to do that. But we heard, a very poignant story from a patient who was treated at a smaller center. And a lot of patients are treated at these smaller centers who maybe don't have the resources to have somebody waiting outside of the operating room to get those tissues. So I think that's a major struggle, and something that I think we still need to work on quite a bit. But obviously, you also heard about what we really need to have an impact on is metastatic disease. I think we still can do better with the primary tumors and MAP chemotherapy. But if the patient doesn't relapse, we do, we obviously, even though MAP isn't great, we have survival. But when we have a metastatic disease, that's another, whole nother ballgame. And, getting access to these tissues has also been a major effort that we've put a lot of time into, and other centers have as well. That's has its own difficulties because, even if there's going to be a surgical resection of a metastatic lesion, these lesions are often very small. So the pathologist, even though we know it's osteosarcoma, they still want to confirm that it's a diagnosis. So oftentimes a lesion this size as the one you see here, we don't get any tissue to do research on. It just goes into the pathology lab, and there's limited access. So accessing biopsies, accessing metastatic lesions is still an issue. And obviously you heard about, the fact that there hasn't been a lot of progress. This is a complicated slide, but I put it up here because I think it illustrates a few really important points that we can highlight. So what this is basically what's called a heat map, and it's a summary of results from gene sequencing of a lot of individual patients. And so each column here is an individual sample from a patient, and the rows are genes that we selected from the data. And this is something called the whole genome sequencing. So this is taking the tumor and extracting the DNA. And now with technologies that allow us to do sequence the entire genome, we can see the copy number changes. So we can see the gains and the losses in the genome. And as you all know, and has been highlighted a little bit, osteosarcoma is unique among pediatric cancers in that it's incredibly genomically complicated, right? So usually you have 2 copies of every gene. In osteosarcoma, we have, so basically these are the dark red little blocks here are genes that have more than 12 copies, right? So there's only supposed to be 2 copies of every gene. These are massive amplifications and deletions, and how that happens is a subject of a lot of intense investigation, but it has to do with basically genomes being shattered and then being put back together in abnormal ways, and the machinery in the cell that copies genes and is supposed to copy things only twice basically goes crazy and starts copying many, many times. So we end up with these really aberrant genomes that have lots of copies of genes. So those are what you see up here. So we have samples that have lots of copies of MYC, lots of copies of cyclin E1, VEGF-A, various other genes. And then down here is the opposite. So these are genes that are deleted. So you can imagine when you shatter a genome and try to put it back together, you multiply things too many times, but you also lose a lot of pieces of the genome that should be there. The most famous deletion that we see in osteosarcoma is a gene called p53, which happens to be the most commonly deleted gene in all of human cancer. But there's a lot of other genes as well, some of which you heard about earlier. So this is actually genes that were selected because they're part of the DNA damage response pathway that you've heard of. So really the reason I put this up here is because I think it'll be obvious to everybody looking at this, even if you don't do, if you're not an expert in molecular biology, that there's no two columns in this panel that are the same. Every patient is different, right? Paul Meltzer, who's been studying osteosarcoma for a long time, says, these are like snowflakes, right? So every patient is different. That you can kind of respond to that two ways. You can say, well, you can sort of shrug your shoulders and say, well, how are we ever possibly going to find a path forward when every patient is unique? And so what we've been trying to do, and a lot of people have been trying to do, is to find patterns in this data, right? And I think the immediate thing you'll realize is that we don't have enough data. So this is my lab's data, Richard Gorlick's in the audience. He has his lab's data, and other researchers here have their own data. So one of the issues is putting the data together. But even if we put together the data from all the different labs that are working on this, it's still not enough data, right? So that's a limitation. So you hear about, machine learning and, how machine learning is going to solve all these problems. But, people who do machine learning, they like to have, 2,000 samples, 3,000 samples, maybe 10,000 samples, right? Well, there's no way we're ever going to get 10,000 samples, fortunately, because not that many patients get osteosarcoma, but we probably could pull together 500 samples or 600 samples. And so that's one of the things we need to do, is to find patterns to move away from this idea that everybody is a snowflake, because that's not going to get us anywhere. We need to find patterns. We need to find subtypes. And I think we're making a lot of progress in that, but there's still a lot more that needs to be done. So this is a slide that I borrowed from an effort that I think many of you in the audience are familiar with. It's a Breakthrough Cancer grant that was recently funded, which many of us are participating in. And really what we're trying to do in that grant is to move the field forward by putting together three things, right? The clinical trials, which you've heard about, the understanding of the biomarkers and the molecular targets that we need to identify through this kind of effort to find subtypes, and then the validation and the efforts to study these in model systems. And one of the things that I think is really exciting about this Breakthrough Cancer effort is that, at least as far as I'm aware, it's one of the first times that we've tried to bring together two models that are kind of out there, but again, studies have been kind of done in isolation. One is the mouse models that many of us work on, and we certainly work on in the lab, and then the canine patients. So one thing that hasn't been touched on at this meeting is that unlike humans, where osteosarcoma is actually a fairly rare disease, it's pretty common in dogs, and there's lots of dogs with osteosarcoma, many more dogs than humans, although there's obviously differences between dogs and humans, I think there's a huge potential to move the field forward by learning from each other. And and the dogs, obviously, they're not models; they're actually patients. They're just not human patients. And but there's a lot more flexibility with what you can do with a dog, and a lot more access to samples and whatnot. So we're really excited about this, and I think that this is really something that we need to think about more. How do we bring the clinical trials together with the models, and an effort to understand the complexity of the biology, so that we can move the field forward. So I had made a list of drugs. Amanda, who's much better with slides than I am, already did a beautiful job. So I don't need to go into all of these details. You already heard about all of this from Amanda. But just again, to briefly mention that, there are different categories of things that we're interested in moving forward. Some of them have to do with intrinsic targeting of the tumor cell itself. So this abnormal genome that I talked about. One of the things we're really excited about is the idea that, yes, you have this abnormal genome that's got lots of copies. Well, the machinery of a cell to be able to deal with that abnormal genome, it has to do lots of things, and many of the things we don't really understand. How does it deal with 12 copies of this gene, 12 copies of that gene, no copies of this other gene? That creates what we call vulnerabilities, right? So it's a sort of Frankenstein kind of cell. And so can we really target that abnormality in a way that's unique? So the normal cells in your body have 2 copies of every gene. They're doing things in a certain way. These Frankenstein cells are doing things in a different way. Can we target that sort of intrinsic vulnerability? So that's kind of one big category. And you heard from Amanda about ADCs, which are essentially a different way of getting drugs into the tumor cell. The other kind of big category is the immune system. So, we know that osteosarcomas, if you look under the microscope, they don't have a lot of the good types of immune cells that we like to see in tumors, the T cells, which are sort of designed to go in and kill tumor cells. They have a lot of what we call immunosuppressive cells. So basically cells in the immune system that are basically telling the T cells to go away and not attack the tumor. And so there's a lot of interest now in how can we change that immune microenvironment. And we know that there's got to be some signal there, because we know that things like MedPact, do have probably some effect, but they're not as strong immune modulators as we would like them to be. So that's another, really strong area of interest. And there's some other things you'll hear from here as well. So yeah, this is just touching on the immune modulators. We've been particularly interested in our lab and other labs as well, and there's some clinical trials in this pathway called the STING, the CGAS STING ENPP1 pathway, which is one of the ways that we know that the tumor cell basically signals to the immune microenvironment to either activate, or activate a T cell response or inhibit a T cell response. And we've done quite a bit of work in the lab indicating that this pathway is shut off in osteosarcoma. So can we reactivate it as a way to kind of wake up the immune system? TGF beta, you heard from. There's some exciting data from a clinical trial on a potential TGF beta inhibitor that's out there. And then checkpoint inhibitors so far have not really succeeded in osteosarcoma, but probably because we haven't figured out how to reprogram the immune microenvironment. There's also interest in directly targeting the metastatic cascade. And this is a slide from a paper that was recently published from Ryan Roberts' group. Most of you probably don't know what you're looking at here, but basically these are mouse tumors that were taken from mice that were either treated with this drug called nintedanib or not treated. This is an experimental metastasis model where you inject cells into the tail vein of a mouse, and then you treat them with a drug. And these dark purple spots are basically the tumors in the lung. And there's a couple of things to point out here. We do a lot of these kinds of studies in mice. Obviously, this isn't how metastasis happens in humans. Humans don't have tails, for one thing, and you don't obviously inject cells into their tails. So it's a very artificial model, but it does allow us to study, how tumors get to the lung, how do they survive in the lung, and can we do anything to block that treatment? So Dr. Roberts' group has done some really beautiful work showing that inhibiting this process called the fibrosis in the lung can really dramatically decrease the ability of tumors to survive in the lung. So here's the vehicle up here in one model, and here are the treated mice, and you can see that basically they don't have these kind of dark purple spots, suggesting that the tumor cells just are not able to survive in this in the presence of this drug called nintedanib. So this is, the kind of experiment that, those of us who focus on preclinical models, kind of love to see, even though we know that there's limitations in terms of the translation. And I just reached out to Ryan. I think it's pertinent to this meeting, and I hope he doesn't mind if I share this, that I was asking him, well, what's going on with this nintedanib? Is it moving towards clinical trials? Well, it turns out they had some meetings with the company, and company saw this data and was excited to move forward. And then at the last minute decided that, well, just kidding, we don't, this isn't really our priority. So we're not gonna provide the drug. So this is, really the challenge that we face. We put a lot of effort into doing these kinds of experiments, testing different drugs, lots of graduate students' time, lots of postdocs' time, lots of grants, something like this, you could get a grant to do. And then at the end of the day, it doesn't translate because, not because the FDA didn't want to approve it, but because the drug company didn't even want to do the trial because, they feel like it's not a priority for them. So I think hopefully one of the things we can really try to talk about at this meeting is how do we change that? How do we make these drugs? How do we make these clinical trials easier to translate? So as I mentioned, we there's a lot of preclinical models that we can use. I think the dog is a really exciting one, and that's going to be a major part of this breakthrough cancer effort. Mice is sort of our workhorse. There's also several types of ex vivo models, so the so-called PUMA assay, short-term tumor cultures, and in vitro models that are sort of being developed. We still do a lot of our work in kind of standard tissue models, although one thing that we've learned over the last several years is that there wasn't, a decade ago, there really weren't enough even cell line models. If you go to if you're studying lung cancer, if you're studying prostate cancer or pancreatic cancer, you can go to a resource called the ATCC and you can get, 50 or 100 cell lines for lung cancer or for pancreatic cancer. For osteosarcoma, there's really traditionally only been 3 or 4 lines that everybody uses. That probably don't reflect the full heterogeneity that I was talking about earlier. So lots of labs, including ours, have put a lot of effort into generating additional cell lines that hopefully reflect this heterogeneity. And I think that hopefully that will make things a little easier going forward. But I think we also still need additional models that more reflect this. One of the things that's really unique about osteosarcoma is this the fact that it's happening in a bone. And that has a lot of implications because these cells are growing in a very stiff matrix. And if you look under the microscope at osteosarcoma, it's not just the tumor cells, but they're surrounded by this kind of, or encased in this kind of almost like cement, right? And that's very unique. And then there's no other cancer that's like that. And so how do we model that in a dish, or how do we model that in some of these other ex vivo models, I think is a really important point. So this is just a slide kind of illustrating the process that we've developed in our lab over many years, which is similar to what's been done in other labs. As I mentioned earlier, getting access to these samples so that we can study these tumors and have more material. And how do we expand that? So the approach that we've taken in the lab is to put them directly into mice to generate these so-called patient-derived xenograft models, and then make cell lines from those models, and then use those cell lines or the mice themselves to do studies. And we have various ways to image. You can do CTs with mice, just like you can do with humans. You can label them with a fluorescent marker that allows you to detect light as a way to measure these tumors. This is from our lab, but this isn't unique to us. Lots of labs do the same kind of thing. And as I mentioned, I think the canine models are a really exciting new approach. They can give us the ability to rapidly study many iterations of different single drugs, but also drug combinations. I think that's going to be really exciting going forward. The genomics of osteosarcoma are almost identical to the human. They also have this very complex genome. And so I think that this is going to be, and obviously another thing I didn't really get into is that most of these mouse models that I'm talking about, when you take a human tumor and you put it into a mouse, you have to use what's called an immunocompromised mouse. You can't put it into a mouse with a normal immune system because the mouse will reject the human cell. It knows that it's not a mouse cell, so it gets rid of it. So you have to use these immunocompromised mice. So therefore, you can't study the role of the immune system. So one of the major advantages of the canine model is that it allows you to study tumors that are very similar to human in the context of an organism that has an intact immune system. So I think that's also going to be really exciting. So just wanted to end here with some important unanswered questions that I think we all need to be thinking about. So are there subtypes of osteosarcoma? We believe very firmly that there are, that not osteosarcomas are the same, but at the same time, not every patient is a snowflake. There are subtypes that are important, and understanding those subtypes, I think, is going to really drive the field forward. So are these subtypes epigenetic? We have some evidence that perhaps they are. There's also important genomic subtypes. This hot versus cold refers to the immune system. So if you look under the microscope, there are some osteosarcomas that have T cells in them and probably would respond to checkpoint inhibitors, but we need to better understand those. There's also a process called the ALT process, which I don't have time to get into, which is a way that how tumor cells, how the chromosomes are organized and replicated, which may also be important for osteosarcoma. And then that brings up important issues with regards to clinical trials. So should we be including this information about these subtypes in the design of the clinical trials? Or should we, at a minimum, design the trials in a way that will allow us to determine whether patients with these different subtypes responded or didn't respond? You heard a little bit about the seroassertive PARP trial that had some important signal, but what is that signal and what are the biomarkers that define that, I think, is really important. And then I think something we sometimes argue about is whether we should develop drugs that are specifically designed to evaluate the metastatic cascade itself, is understanding the ability of tumors to survive at the metastatic niche something that is not only biologically important, but also potentially important for the design of these trials. So just wanted to end there. I think I'm a little over on time, again, thank you for the opportunity to present here. I'm really excited to hear the speakers for the rest of this meeting, and I think it's going to be really impactful. So thank you.
All right, good morning, everybody. It's so good to be here with all of you today. We're kicking off session 4.
Everyone, please take their seats.
So my name is Kristen Wessel. I'm a pediatric oncologist. I've been at the FDA for a little over 3 years on the team that oversees drug development for pediatric and rare solid tumors. And I'll be speaking to you today about regulatory considerations for clinical trial design and endpoints in osteosarcoma. I have no disclosures. So this slide illustrates how, as regulators, we are always grappling with striking the balance between certainty and regulatory burden. And this is especially true when we're talking about rare diseases like osteosarcoma. When we require more data, that increases the certainty of the results we're looking at, but also increases the regulatory burden, which can lead to calls that we're being too cautious given the severity of the disease and the unmet need. On the other hand, if we require too little data, that leads to lower certainty and could potentially lead to the approval of unsafe or marginally effective therapies. So our goal is to strike the right balance, and to do so while remaining flexible, but also consistent, efficient, but also thorough, and interactive while remaining independent. Now, as Dr. Smith mentioned in her talk earlier, there is a federally mandated standard that must be met for a drug to be approved. and that is substantial evidence of effectiveness supported by clinical data from adequate and well-controlled trials. Now, what constitutes an adequate and well-controlled trial depends on the disease context. We don't follow a one-size-fits-all approach. Rather, we tailor our advice and assessment of the evidence to the specific disease and the approval pathway being sought. And we recognize the need for treating physicians to incorporate emerging data into clinical practice, especially in an area like this where there's such high unmet need. And that falls in the realm of practice of medicine, which we do not regulate at FDA. However, when the data from a trial are submitted to support a marketing application, we are still obligated to evaluate it based on the federally mandated standards. So the selection of an endpoint is crucial when designing a trial intended to support a marketing application. One endpoint we have that's unique to oncology is objective response rate, so whether a tumor gets smaller on imaging. An attractive feature of this endpoint is that it allows for single-arm trial designs. So since tumors don't shrink on their own typically, we can be confident that the effect we're seeing is due to the drug. Objective response rate also allows for earlier measurement of the drug effect than survival does. The downside of using ORR, particularly in osteosarcoma, is that imaging interpretation is required. And I know that Dr. Janeway is going to touch about this more in her talk, but because osteosarcoma tumors form a bony matrix, they don't always shrink on imaging. So just because a low ORR is observed may not mean that a drug is not active. So the other events on the slide, so progression-free survival, event-free survival, and overall survival, we refer to as time to event endpoints. And in general, studies using these endpoints require a control arm. Be more certain that the effect we're observing is due to the drug rather than due to some other factor. Progression-free survival, PFS, or event-free survival, EFS, are often used in oncology trials because they enable earlier readout than overall survival, and they occur prior to crossover if the trial allows for that. Overall survival is often considered the gold standard because it's a direct measure of clinical benefit. It's easy to interpret, and it incorporates safety information. On the other hand, it can take a long time to read out and may be confounded by crossover and receipt of subsequent therapies. So the appropriate trial design for a clinical trial in osteosarcoma may vary depending on the stage of development. So early on in development, the goal is to evaluate for a signal of activity and gather initial safety data in order to make a go or no-go decision about whether the drug merits further study. These signal finding studies in osteosarcoma are typically conducted in a relapsed or refractory setting. And the goal here is to determine whether there is a signal of activity as expeditiously as possible so that promising drugs can be moved forward and so that further resources are not invested into development of drugs that are not effective. Randomized trials are not often feasible at this stage of development because they take a long time to complete and require more patients. And while tumor response can be a useful tool in oncology to look for early signs of activity, we've kind of already touched on that ORR may underestimate drug activity in osteosarcoma. So the question is what are we left with? Osteosarcoma experts have suggested specific historical benchmarks to aid in this type of go, no-go decision-making. And we agree at FDA that these benchmarks are reasonable to use in this setting. I know in Dr. Janeway's talk, she's going to go into more detail about how these benchmarks were derived. But for studies including patients with measurable disease, it's reasonable to use an EFS benchmark of 4 months, and for 12 months in studies including patients with resected disease. So once a signal of activity has been detected and there's some initial safety data, at FDA, we're then looking for more information to make a determination whether a drug meets that safe and effective criteria that we're looking for, and to inform a full risk-benefit assessment for a marketing application, and to tell us whether the drug provides a meaningful benefit to patients. The appropriate endpoint for a registrational trial and other aspects of the trial design depend on several factors. One of these is the disease setting. We kind of heard about sort of the different scenarios that we're looking at for evaluating drugs in osteosarcoma, but for example, is the drug being studied in a newly diagnosed setting as part of upfront treatment? for patients with relapsed or refractory disease, or as a maintenance therapy for patients who have finished upfront therapy, may not have any residual disease, but who have a high risk of recurrence. Other factors may include the mechanism of action of the drug, whether measurable disease is present, any observation of tumor shrinkage on imaging in early studies, and the approval pathway being sought. Randomized controlled trials are needed for the interpretation of time to event primary endpoints in a trial supporting marketing application. And this is important in order to attribute the effect on a given endpoint to the investigational product and not other factors. As we heard about earlier today, osteosarcoma is a very heterogeneous disease, and there are many patient-specific factors that are associated with varying outcomes. And we're not sort of aware yet of what all of these may be. So for example, if a patient relapses early versus late, or whether a relapse is an isolated pulmonary lesion or with multifocal bone lesions. So it's very difficult to tease out the contribution of a drug versus these other factors when there's not a randomized control arm. Because of all these potential variables, we strongly recommend meeting with FDA early on in drug development. And by early, we mean before starting a trial intended to support a marketing application so that we can provide advice on the design. So with all that said, we acknowledge that randomized controlled trials are difficult to conduct in the rare disease space. And we're always willing to give feedback on innovative trial design approaches. In many scenarios in osteosarcoma, a trial with an active comparator arm may be appropriate. So one approach sometimes used in oncology trials is an investigator's choice control arm, where an investigator can choose from a list of pre-specified regimens already used in clinical practice. Another approach would be an add-on design. So this is, in the frontline setting, MAP versus MAP plus new drug. Or a new agent added on to a combination that's sometimes used in clinical practice. Alternative randomization ratios can be another strategy where there's a greater chance of randomization to an experimental arm versus a control arm. Crossover designs allow for patients randomized to a control arm to switch to an investigational arm after progression. And then platform trials are an opportunity potentially to more efficiently evaluate more than one new investigational agent or combination. So this type of trial would include one control arm and several investigational arms that would be either different products or combinations of products that are each compared back to that control arm. Seamless adaptive trial designs may also increase efficiency in drug development as they combine two or more clinical trial phases into a single study and offer flexibility in modifying the trial based on accumulating data. So while randomized controlled trials with time to event endpoints are the current standard, we at FDA share a common interest with the osteosarcoma community in development of novel endpoints that may enable earlier assessment of efficacy. So for example, ctDNA. To highlight a couple of recent initiatives, Project Endpoint is an Oncology Center of Excellence initiative started in 2023, which aims to enhance the development of endpoints in oncology drug development, both within different centers at the FDA and through engagement with external stakeholders. And then also last fall, FDA published a guidance on the use of ctDNA for early stage drug development that is available on our fda.gov website. Prior to use as a primary endpoint, though, these novel endpoints must be prospectively validated, and we welcome questions about incorporation of novel endpoints throughout drug development in sponsor meetings. So FDA also encourages use of patient-reported outcomes as endpoints in clinical trials in osteosarcoma. And a guidance on core patient-reported outcomes in cancer trials was published also last fall. PROs, when incorporated appropriately into a clinical trial, can be very helpful to our assessment of clinical benefit when we review marketing applications. Because osteosarcoma most commonly occurs in adolescents and young adults, I wanted to touch on the enrollment of pediatric patients in clinical trials. We generally encourage inclusion of adolescents or patients 12 and older early on in development, If the trial is first in human, we typically recommend obtaining some initial safety data in adults before moving to younger patients. There may be some exceptions to this rule. We take it on a case-by-case basis. We recommend expanding inclusion criteria to children younger than 12 as soon as feasible and appropriate based on safety data obtained in older age groups. And we also acknowledge that patients in this young age group will probably make up a small proportion of patients in most osteosarcoma clinical trials, particularly in the relapse refractory setting. So osteosarcoma is a disease where we can generally leverage extrapolation of efficacy from adult to pediatric patients because the disease process is similar in these age groups. And with this slide, I will again encourage you to come talk to us. Sponsors are strongly encouraged to meet with us early and often throughout the drug development process. At these meetings, we recommend inviting key academic investigators and patient advocacy representatives because we find these perspectives very helpful and informative when we are looking at a development program. For those sponsors also developing companion diagnostics, we encourage involving FDA's Center for Devices and Radiological Health early on. And finally, since many osteosarcoma trials require international collaboration, there are opportunities to engage FDA and other regulatory agencies such as the EMA together to reach alignment on key trial design issues prior to starting a registrational trial. So with that, I'd like to acknowledge my FDA colleagues, the OCE, Osteosarcoma Institute, and the Osteosarcoma patient and advocate community and those who spoke earlier this morning. Thank you all so much for being here. And I look forward to taking your questions during the Q&A. And at this time, I'll pass on the mic to Dr. Janeway.
Well, I just want to thank the Osteosarcoma Institute, Amy, the FDA, and organizing this meeting. I think it's incredibly important from my investigator and clinician perspective. So I'm Katie Janeway. I'm a associate professor of pediatrics at Harvard Medical School. I'm a practicing pediatric oncologist, and I do clinical investigation in osteosarcoma, wearing a couple of different hats. But most of my comments today are going to be based on my experiences as chair of the Bone Tumor Committee for the Children's Oncology Group, where I've really benefited from interaction with my colleagues in that setting and sort of developing these understandings. So I'm going to start by talking about trials to establish a new standard of care for newly diagnosed patients. Here, our primary endpoint is event-free survival, and our secondary endpoint is overall survival. The design is generally a randomized trial with our standard of care arm being MAP chemotherapy. You heard from Dr. Marinoff that has really been proven in a randomized trial to improve outcomes for patients with localized resectable disease. Some considerations in this setting are the patient populations, tumor resectability, and I want to make a very important point here, which is no clinical trials in osteosarcoma can be considered or designed without thinking about surgery, and more and more nowadays in the relapse setting, the possibility of things like radiation and interventional radiology. Soon, I hope that there will be biomarkers of outcome with MAP chemotherapy that need to be considered in trial design. The accrual and duration for trials like this are generally based on the patient population with localized disease with a resectable primary tumor, and usually require around in the high hundreds of patients. In the US cooperative group setting, where we have about 200 clinical trial sites, both in the US and in Canada and a couple of other countries, you end up enrolling about 14 patients per month, and this requires about 4 years of accrual. And given the sort of timeline for this patient population, that usually takes 6 to 7 years to have your trial results. What this really translates into in a practical manner is one trial every decade at best. And so what's really important is signal finding trials in other patient populations. And these are, from my perspective as a person who's interested in these sort of new standards of care, that we're really looking for signals of efficacy so that we have confidence moving into this sort of 10-year endeavor. But those signal finding trials are also really important for things like breakthrough designation for pharma. Those signal finding trials are typically performed in relapse patients. And I want to make a really important point here. For us, pediatric oncology and sarcoma doctors, we use the terminology relapse, but what we really mean is metastatic disease. Okay, so putting on your sort of GI cancer hat or breast cancer hat, what we're really talking about here is metastatic disease, okay? So we're really talking about sort of, for these patients, it's their first line therapy for metastatic disease. And I'm just showing you again our currently enrolling children's oncology group trial AOST 2032, which is assessing the addition of cabozantinib to MAP chemotherapy. So what's the problem with our signal finding trials? You already heard about this from the FDA, but osteosarcoma has a calcified stroma. It's a tumor arising from bone. And so what I'm showing you here is a very typical problem that we have where this is an X-ray. Maybe I can use this pointer. Hopefully you can see that. This is an X-ray of a patient prior to chemotherapy where you can see there's some soft tissue swelling and then this sort of moth-eaten bone. After the adjuvant chemotherapy, and this is that MAP chemotherapy that is correlated with improved overall survival, you can see a, quote, bigger tumor. The MRI here has no RECIST response. And if you want to read about this in more detail, we did actually study this in a very systematic way. And there are essentially no RECIST responses to MAP chemotherapy. So that is not a good surrogate endpoint for therapies that are going to improve cures in this disease. So we worked on an osteosarcoma specific endpoint. And just to give some additional background here, approximately 25% of patients at recurrence are rendered no evidence of disease. They have metastatic disease that's limited to the lung parenchyma. And because we have a goal to try to cure those patients, we are very focused on trying to resect those nodules when possible. So our objective in this study that we conducted was to determine a benchmark progression-free survival in recurrent patients enrolling on phase 2 trials to enable signal finding trials in patients with recurrence or metastatic disease. We really had to think about 2 cohorts of patients. Cohort 1, measurable disease, which in our sort of treatment context, because these trials generally say no other curative therapy available, means that these patients have unresectable metastatic disease. And cohort 2, which was a group of patients who had had their disease resected or resectable, limited to the lung parenchyma. And we had about 100 patients in the cohort 1 group and about 40 patients in the cohort 2 group who had enrolled on legacy trials. And we basically looked to establish EFS or PFS, which are really equivalent in this disease, benchmarks to enable these trials. So for cohort 1 or measurable disease, you've heard a couple of people talk about the 4-month progression-free survival. It really comes from this study, and it's 12% with a very narrow confidence interval. And in cohort 2, which are the resected patients, we chose a 12-month time point because that's when the curve sort of stabilizes, and that's 20% patients who are progression-free with a slightly wider confidence interval, which there's a typo in that. Sorry, I think that's supposed to be 14% to 43%. And there are really an important point here is that there's in this cohort 1 patient population in particular, there's no difference by number of lines of prior therapy. So that, and there are really no other sort of clinical features of these patients that take you from being really bad to worse. Okay. So the COG led a series of phase 2 trials using this single arm design in recurrent osteosarcoma in these 2 cohorts of patients. And I want to make a couple of points here. The first is that the FDA, the IRBs, site investigators, and patients seem to be accepting of this context, this type of trial, as the first trial in pediatrics. So all of the agents with an asterisk here, this was the first trial in pediatric population. The age of eligibility was 12 and above. And very importantly, these trials in relapsed osteosarcoma, the median age of enrollment is 17 years. So this is absolutely an adolescent young adult patient population. It is not children. It's not small people. It's larger people who have sort of grown drug metabolism and grown development, okay? The second point I want to make is that accrual is brisk. So in the measurable disease patient population, we enroll 4 patients per month. And so we accrue, like we often actually design these as a 2-stage design, even though we're using a progression-free endpoint in the measurable disease patient population, we sort of use it as a binary, progression, non-progressed, so that we can do 2-stage design. And then in the resected disease patient population, we have about 1.5 patients per month. So it enables us to do a single arm study for the measurable disease patient population using a large number of sites, somewhere between 6 months to a year, depending on whether we have 1 stage or 2 stages. And then in the resected disease patient population, about 2 years. The next point I want to make is something that you hear us talk about a lot. The bar is high. So for these trials, we chose, the people who designed these trials in the cooperative group, to ask for the drug to perform extremely well. So you can see the target change in progression-free at 4 months for measurable disease patients. We said we wanted to go from 20% to 40%. The 20% is the upper 95% upper limit of the 95% confidence interval. And we wanted to double that. That is asking a lot from these drugs, especially because they're single agents. But we did that intentionally because our goal was to find something that we could bring into that decade-long new standard of care therapy, okay? You don't have to choose a high bar. It's what we did intentionally. And then finally, oh, sorry, that's supposed to point that out. Okay, then one other point that I want to make is that glamitumomab is not as negative as some of the other trials. So there were 3 out of 19 patients who were progression-free, or 16%, compared to the other trials in this patient population of measurable disease, where it was like 1 or 0 patients. And there was actually a responder in that trial. What do we do with that information when we set this bar super high? I would say we have to iterate on that. So glamitumomab is an antibody drug conjugate, and you've heard that there's activity of other antibody drug conjugates. I think this points to that class of drugs as being of interest and needing additional trials. Finally, the EFS benchmark in the measurable disease patient population is accurate, reliable, and does not change over time. So this Kaplan-Meier curve of event-free survival that's shown here has the 7 older phase 2 trials, which were in that original publication in the dotted line, and the 3 newer phase 2 studies in the solid line. And they are essentially overlapping. So, but very important, the EFS benchmark is specific to the patient population. In other words, the eligibility criteria for the trials, okay? We have consistently used measurable disease at recurrence in the US standard of care context and the statement, no other curative therapy, which basically means unresectable metastatic disease, okay? So that's what that benchmark is about. It's not about other patient populations. And when you look at just some more proof that it's a very reliable benchmark, if you look at these 2 placebo-controlled trials of regorafenib, one conducted by Dr. Davis, who's here, you can see that the placebo arm has a progression-free survival of 10%. And it's just like our benchmark. And regorafenib actually hits that very high bar that we were looking for, over 40%, just like all of the other multi-tyrosine kinase inhibitors that you heard about from Dr. Marinoff. There are some limitations to this single-arm phase 2 signal-finding trial in the measurable disease patient population. Number 1, combination with agents active in recurrence like chemotherapy and the multi-targeted kinase inhibitors creates a problem. So here are 2 examples. Gemcitabine is thought to be an active drug at relapse and is been combined with 2 targeted agents, azenosertib, WEE1 inhibitor, and sirolimus, an mTOR inhibitor. In both of those settings, the 4-month event-free or progression-free survival is around that 40% target of sort of activity. But these results will need further investigation with a randomized trial because maybe you're seeing that just from the chemotherapy alone. That's okay. Using that kind of staged approach of doing a single arm to a randomized trial is useful if you need to know about the combination dosing or tolerability, if you want to know about biomarker assessment, or to really invest in a randomized trial, you want to see some early signal of like how active is that drug. That's a totally reasonable approach. You just have to know if you go into one of these single arm trials with a active drug, novel agent combination that you're going to need to do a follow-on study. Additional lessons learned. Moving to the other sort of cohort 2 patient population where there's been resection of pulmonary only, meaning pulmonary parenchyma only, resection of disease, this is a little bit more complicated. Interpreting results of these single-arm trials when event-free survival is greater than the benchmark, but does not achieve the pre-specified statistical goal is quite challenging. And so what you'll see here is that the blue line is from AOST 0221. That's the study that we used in our original publication about these EFS benchmarks. That is sorry, go back one. That is an older trial, was conducted in the 2000s. And then the more recent trials that we did in this series of COG trials are shown in the red dotted line and the black line. Possible explanations for this variability in event-free survival. 1, the agents have activity. Now, they did not achieve that high goal that we were looking for, but they're not the same as the historical benchmark. They're in between. So maybe the agents are somewhat active. 2, statistical chance. You just run the same experiment multiple times, you get slightly different results. Maybe there's improvement in outcome of this patient population over time. Maybe our surgeons are getting better at what they do or more aggressive. Or maybe it's just the enrolled patient population, which is sort of like statistical chance. Since we cannot account for numbers 2, 3, and 4, this is another reason to set the bar high for these trials. That if you're really seeing that big change in the single arm trial, it's most likely that it's due to efficacy of the drug and not these other factors. So let's turn now to a couple other approaches. Randomized trials in relapse and refractory disease. Here I want to take a case study of a trial that looked at chemotherapy in relapse versus chemotherapy plus the multi-kinase inhibitor lenvatinib. This is the OLI trial that was already mentioned by Dr. Marinoff. The key eligibility in this trial, patients could be 2 to 25 years of age. They could have measurable or evaluable disease. We're getting away from that benchmark cohort 1 population / cohort 2 population. We're actually mixing those. So 20% of the patients on this trial had resectable disease at baseline. The age here, just pointing it out, this trial was open in 21 countries, and it took 20 months of accrual. And I think the number of study sites is something around 80. This was a 1-to-1 randomization. There were 5 cycles of chemotherapy. Lenvatinib was continued after chemotherapy was completed. Crossover was allowed. And after cycle 5, patients could continue therapy and could have surgery. Here are the results of this trial. The lenvatinib IE has a slightly improved progression-free survival, as shown in the dark blue line on the left-hand graph. Whereas the IE only was in the orange. Overall survival was not different. The 4-month PFS, which I'm showing you under the left-hand graph, was above that benchmark, which is not surprising because we know ifosfamide and etoposide are active in this disease. So that does not tell you anything about IE plus lenvatinib. This did not meet the pre-specified difference in activity that they were looking for. 14 patients crossed over. I want to look at a couple of things about this trial because there's some lessons here. One of the big factors in osteosarcoma is this resection of disease. That has a big impact on outcome in the relapse setting. In fact, it really differentiates these patient populations. Survival, if you can't resect disease at recurrence, is really quite poor, where it's slightly higher if you are able to resect, around 20%. They asked at baseline, at enrollment, do we plan to resect? Do we think we can resect this patient? And that was actually about equal in the 2 arms, but surgery was imbalanced in the 2 arms. So twice as many people had resection in the ifosfamide etoposide only arm compared to the IE plus lenvatinib arm. And resection is terribly difficult to predict. So 5 out of the 18 resectable patients at baseline ended up having no surgery. And 15 of the non-resectable patients at baseline ended up having surgery. And some of that might be due to tumor response, but a lot of that has to do with other factors. We're just very bad at predicting what we are going to do in the future in terms of management of metastatic sites of disease. I want to say one more thing about assessing progression in recurrent osteosarcoma using RESIST, which is our sort of standard approach. This is relevant to all of the study designs I've talked about, except for in the newly diagnosed patients where usually they have no evidence of disease and then there's some very clear evidence of new disease. But any of these at relapse where we have disease present when we start treatment are subjected to these problems. So this is actually baseline scans from a patient enrolled on my investigator-initiated trial of olaparib plus ceralasertib at relapse, and this patient had measurable disease at baseline, which I'm showing you here. It's an inguinal lymph node, and there's just one of many lung nodules. And what you can see is even at baseline, these sort of white blobs are calcification in the lymph node. And you can see after 2 cycles, wow, that lymph node really looks bigger. It's like, and it was definitely measured as at least 20% bigger, and maybe the lung nodule looks a little bit bigger. Here's the PET scan. Tons of activity at baseline and not nearly as much activity in that follow-up study. And what's happened here is there's been calcification of the lymph node, and that is likely response. This patient actually had to come off study because of the way the protocol was written. We actually made a protocol amendment based on this to say that treatment could continue if we thought there was clinical benefit. The patient then was able to get the drugs by compassionate access and has been on those for a year. So other tools that we have at our disposal and are developing are circulating tumor DNA, which is detectable in 40% of patients who have localized disease at diagnosis. And I think my colleague, Dr. Crompton, who works a lot in this space, is still working on the proportion with relapse who have detectable ctDNA or metastatic disease who have detectable ctDNA. The presence is at baseline is associated with poor outcomes, and results of studies correlating ctDNA with disease activity are expected soon. So I just want to talk about randomization in relapsed and refractory disease in that cohort 2 group where it's sort of adjuvant therapy following resection of metastatic disease. This is a case study of a trial actually that Dr. Hellman was very important in developing. Patients were eligible if they had resected pulmonary metastases and were greater than or equal to 15 years of age. They accrued 88 patients in 4 years. It was a randomization between placebo and saracatinib, a SRC inhibitor. And they were looking for a 60% relative improvement from 33% to 53% in 2-year PFS. And they had an 80% power and a one-sided p-value of 0.1. 40 subjects were enrolled in 5 years and had a median age of 22 years. There was no statistically significant difference in EFS. So basically, this study was closed because the goal accrual was felt to be very difficult to reach. But in spite of this, I do think randomization in this patient population should be feasible. I think probably placebo is not the best approach here, especially for the younger patients. I think probably we would want the age to start at 12 years of age. I think both arms need some sort of similar treatment. So both agents being oral, for example, or both side effects being sort of equivalent, or 2 different immunotherapies. I think it's hard to do this if the 2 treatments are quite different. I actually had this study open at my site. I thought it was a great study because we really don't know whether people need treatment in this setting. But the reality is that the patient and the doctor have often decided that they either do or don't want treatment. And so when you randomize between treatment and not, people are often not willing to let chance make that decision. And I think you need many centers and community engagement, including, as Anne mentioned, engagement of the patients who may eventually be considering this. Are there other trials that we can do in newly diagnosed patients besides the one I started with, which is sort of determining new standard of care which is mostly based on localized resectable patients? I think novel approaches could be studied in patient populations with poor outcomes, even with MAP. Those are patients with metastatic disease at diagnosis. That's about 25% of our patient population, so it's a smaller patient population. 60% of those patients have lung-only disease, and their outcome is highly influenced by resectability. And you probably can get about 1 to 2 patients per month in a US cooperative group setting. 25% of those patients have extrapulmonary, usually bone metastases. they tend to have quite poor outcomes, but there's a small number of patients. But it is a favored population for studies of things like radiopharmaceuticals. Eventually, we hope to have biomarker-defined high-risk localized disease as well as a potential patient population for these trials. But we need additional data on biomarkers and outcomes in these patient populations. So we really need to do those kind of benchmarking studies in these different patient populations and have the data, like modern data, in order to do that. What interventions could you study? I think people like to study maintenance in this patient population, but there's a problem, which is that a lot of patients progress in the early part of treatment. And so you further reduce the patient population if you're looking at maintenance. You could do MAP plus a novel agent as sort of a single arm signal finding study, but again, you need that benchmarking work to be done. You need to really understand the outcomes in these patients. Or other novel osteosarcoma chemotherapy plus novel agents. And my colleague, Dr. Reed, is working on a study like this at Memorial Sloan Kettering. Those are just some benchmarking patient populations there, but you can see some of them are quite old. So some key takeaways. This patient population is adolescent young adults, not, quote, pediatric. Osteosarcoma trials can precede pediatric phase 1s and pediatric development plans. I'm not really in charge of the second part. That's really an FDA statement, so I'm stepping out of my lane. Event-free survival is a key endpoint in both relapsed refractory and newly diagnosed disease. Because of the tendency for calcification with response, measures of disease beyond cross-sectional imaging, such as PET and ctDNA, should be included. For rapid assessment of novel agents, single-arm signal finding trials and measurable unresectable disease are very helpful, but the eligibility must match the benchmark population. If combining with an active agent, you will need follow-on randomized trials, and most randomized trials require a very large number of study sites. And I just want to thank everybody for being here. This is such an important topic, and we need everyone in this room, I think, to work together to really get to a place where we're standing up these trials and completing them efficiently so we can bring new drugs. Thanks so much.
Thanks, Dr. Janeway and Dr. Wessel, for that background. Dr. Singh and myself are here as industry, non-academics, but people who have actually had that perspective. So I am Melinda Merchant. I am pulling it closer. We're happier. I am a physician scientist who spent the first 15 years of my career taking care of sarcoma patients in academia. And with that drive that we've heard so much about today, the urgency of timing, of need for more things, chose to go to industry where I've spent time in both pharma at AstraZeneca, and I'm currently the chief medical officer at a small biotech that's a spin out of Yale called Normunity. I'm here as my hat as being a part of the strategic advisory board for Osteosarcoma Institute, again, working with this large group, because it really does take a multifactorial group of people, multifunction. And that's one of the things that I think that industry does bring to the table quite well, is that group and that ability to get towards the approval. So that's a little bit about my background and my perspective. I'll pass it to Harpreet.
Thank you. Hello, I'm Harpreet Singh. I'm a medical oncologist by training. I trained at the National Cancer Institute and went directly from there to the FDA, where over time I had the honor and privilege of standing up and leading the newly created Division of Oncology 2, which was formed in 2019 as a result of a reorganization of the Office of Oncologic Diseases. And within that, I was able to preside over the team that included Martha and Nicole and Kristen and many others in regulating not only adult solid tumors, that included the thoracic cancers, the brain tumors like GBM, another challenging space, but also the pediatric solid tumors, as you've heard, including osteosarcoma, and rare tumors, so selumetinib and some of the plexiform neurofibroma. So many of those approvals and lessons I took with me. I spent the better part of 10 years at the FDA, and last May went into industry. I'm now the Chief Medical Officer of a global clinical research organization called Precision for Medicine. And we conduct clinical trials for oncology and rare diseases for small to mid-sized biotech companies. So we don't actually work with big pharma. We only work with biotech, only in oncology and rare. So it's been an incredibly steep learning curve, and I'm excited to be part of this conversation. And thank you so much for including me.
Thanks. We wanted to hit on some of the same themes that you've heard about. And I think one of the key things that you've heard from both our FDA colleagues in this business and also from the academics is engage early. That absolutely is important in industry as well. So there are a couple of inroads to having osteosarcoma early in the clinical trial population. And I want to say a couple of things about our perspectives on what's important, how to have those interactions, when to have those interactions, and how much power many of you in this room have because of your expertise, because of your personal experience, because of your advocacy. You are very, very welcome in this education component of engage early. So that's what we have to have, is an internal advocate to basically advocate for osteosarcoma to be included in those early trials. We've heard a couple of issues related to why that may not happen. In an all solid tumor phase 1 trial that we would be designing from new science, maybe it's not even publicly known yet, we are designing essentially that entire path to approval. So understanding where that path is, understanding the things, that we've just learned in the last couple of sessions about a mean or a EFS at 4 months can give you a signal of possible activity. That's important. That hits time and the bottom line. So having that vision mapped out for what it could look like is important. It's also important to know that osteosarcoma doesn't look like just everything else that can come on an all solid tumor trial in a phase 1. And especially in biotech and the funding of biotech in the last couple of years, it's really much more important that we have that signal finding in the phase 1A, in that first in human setting. And so I think it's a place where osteosarcoma, a little bit different than some of the other pediatric tumors, and very important to pull out, those patients, if they are resist evaluable, and sometimes that's only necessary later, but more and more that's necessary in the phase 1A, can be on that trial. And when you get that signal, you'll see experience of this is where we can do our phase 2, or you'll hear people this afternoon in our panel of multiple industry about how to take that signal finding and make it into your pivotal path toward approval. But getting back to the how do you get those patients into that first trial and think about endpoints that help people in that setting, I think is what we tried to ponder in some time and had some themes about how can you think about those endpoints in those early settings.
Yeah, I think one thing that in Melinda and I's discussion, I was saying that osteosarcoma is certainly not the only disease that does not lend itself to ORR and resist. If you take the adult tumor types, we have PDAC, pancreatic adenocarcinoma, GBM, head and neck cancer. If you look across the clinical trial landscape, you actually don't see any, if I'm correct, about any single arm approvals. There's one actually, Maris, for NRG1 fusion. This is incredibly rare percentage of pancreatic cancer where they were able to demonstrate a response rate. But so these are tumor types that do not generally lend themselves to response rate. And that's not necessarily because they're calcifications, but generally because of the issues around imaging, the anatomical constraints, and the limitations of our imaging today. So what do we do in these tumor types? And we walked this forward, but we actually needed to take a step back, because what Melinda pointed out to me was at least those tumor types are being put into kind of basket trials, all solid tumor trials, whereas osteosarcoma cohorts are traditionally not. I look at, dozens of protocols probably a week, and many of them are early phase first-in-human oncology assets. And I think sometimes because of the population for osteosarcoma being AYA, they kind of get caught in the middle where no one's, I don't want to say no one, but are they being thought about as adults in drug development? Just anecdotally, I don't think we've done enough ADC work for osteosarcoma or in adolescence period for other therapeutic areas. So that's one thing. And we talked about just that education piece. So if you're thinking about, and I'm seeing a lot of biotech companies use AI and other tools for signal seeking and signal detection and trying to identify, if there's a biologically plausible mechanism and rationale, just coming up with a list of tumor types that it makes sense to have cohorts in. And I think that education piece of what about osteosarcoma needs to be raised more so that there's a thoughtful conversation around adding these cohorts to classically adult oncology trials, if you will. And then knowledge about the landmark endpoints that are acceptable to FDA, at least for signal seeking and other registrational bodies, I think is a key first start.
Correct. I think also thinking about the patients, we've talked about how do you define a metastatic patient? In many of the adult indications, there is a first-line metastatic. That
Right.
patient could have had 5 lines of therapy before they became metastatic, but now they're a first line. And our randomization isn't against, something that it has to be had in the patient before you get to that trial. You can actually think about, okay, well, here's something that's approved in that setting. Can we do somewhat better? And I think we can have a robust discussion on how you define that somewhat better in your path too. But how do you start making those steps with that patient? It may start with having an NCCN guideline that says a young adult with osteosarcoma, first-line metastatic, should get this or a clinical trial. This is actually how we can often advocate with our internal stakeholders is we pull out, here's my screenshot of the NCCN guideline, and this says, I can't in this stage use a clinical trial. I don't have that tool right now for osteosarcoma. Could this room help make that available? Or not exactly that. I'm not telling you what it has to be. But can you put your layer on it to educate so that there are very easy, this would be the path to get there. This is the patient population we're talking about. And so maybe we're not so caught up in, does it matter how many lines you've had? It really matters what you have and where we're attaching it. Now, there was a mention of metastatic disease too. That is so hard in the first in human and understanding how to deal with that because you essentially, a biotech goes through Series A and Series B fundings. They have, this is my TRIM trial. This is what I need to get out of that early phase 1 that also happens to include the 1Bs that we put some of these disease specific expansions that the OSI has been able to support at times. In that setting, how are we defining the patient that's going on that and where they might be going is ultra important years before that's happening. And so that education from almost everybody you've heard in this room today and others, your voice as a key opinion leader, your voice as a patient, your voice as an advocate are actually really important to tell our stakeholders it's worth using this small amount of money to include this. And I think as a physician scientist, that's when your science, rationale match up for a win for the patient. And if you can describe that across the way in a relationship building time that keeps telling the stakeholders, quite frankly, sometimes shifting environments of pressure and different pressures in biotech than pharma, it will be very important to understand how you can get to that win-win for the patient and all the partners and stakeholders. And we'll try to, again, address that a little bit more with a variety of industry people who can talk to some of those different pressures and case studies in a panel this afternoon.
I think some of the, lessons that we can take or some of the flexibilities that we're seeing within the FDA could be appropriate for discussion to kind of borrow across CDER and CBER. And I'll give you an example of that. I'll give you two examples. So one is the notion of randomization. And is it appropriate or is it acceptable for patients to be, so I mean, there's many issues with this case study. The fact that you need so many sites adds time, adds cost. As someone who now works in a CRO, I can tell you that, you know, you will get to a point where the cost just makes things infeasible for biotechs, and those are often the companies developing assets for pediatric tumors and rare tumors. And so the idea that, is there any way that we can minimize this randomization either through trial design or through being flexible? So if you borrow from the lung cancer space there, and we've seen this unfortunately in second-line lung cancer, where again, no one wants to be randomized to single agent docetaxel. And what do we see? We see asymmetric dropout, and we see trials being rendered uninterpretable. So that helps no one. So what can we kind of do about this? So we can think about, and there was a thought, there was actually a project at FDA called Project Switch. And this was centered around second-line lung cancer. And which unfortunately today still remains the standard of care in second-line lung. But there was this idea that we have so much data on this particular asset, on how this drug performs and behaves. Can we kind of switch out, if you will, if we're meant, I'm just going to do simple math, but if we're meant to have a 400 patient trial, can 100 of those patients come from an external control, or patients that are either contemporaneously being treated in real life, so prospectively, pragmatically, or based on historical data, and then only 100 patients are randomized to that control arm? Is there crossover? That's another question. In an add-on design, that's questionable, but you could cross over to the single agent. And then therefore you have basically a 2-to-1 randomization. The problem, and I used to throw out 2-to-1 randomization a lot when I was at FDA, the problem with the 2-to-1 randomization is that it automatically increases the sample size. So if you got creative with your stats team, and this then incurs cost and time and more sites, and so we're back to like, this just may be infeasible, and we're going to not pursue this. And unfortunately, we are seeing that. We're in a capital constrained environment right now. We are seeing de-investment from oncology into autoimmune. And part of that is the global macro environment, but the more uncertainty you have from a regulatory and technical standpoint, the less risk companies are willing to take, and you have divestment. So that's one thing, is can we be flexible in the randomization schemas and really bringing in that external control, that real world data contemporaneously? There's some good examples of that. So that's one kind of food for thought. Another one is what CBER is doing with the RMAT designations, right? So RMAT designations are for the cell and gene therapies, the regenerative therapies, and by design, compared to breakthrough, which we have in CDER, which were for the TKIs, the monoclonal antibodies, and so forth, where breakthroughs are typically granted on overall response rate, objective response rate, which we've talked about here, not unlikely, right? So then you're going to see less breakthroughs if that is our criteria. On the other hand, for the regenerative therapies, they're utilizing more biomarkers in addition to the clinical safety and efficacy data. So that's really lending itself to more of the modality. But as we think about the disease itself, and I would encourage this, by the way, for head and neck, for GBM, and for PDAC, all these diseases in which we're not really garnering response rates, can we borrow a little bit from the concepts of CBER under the umbrella of OCE and say, you know, for certain diseases in which the biology of the disease or the restrictions of our imaging technologies don't allow really for RECIST to be a way for us to grant these designations, can we think a little bit more flexibly about what we need to grant a higher order designation, including a breakthrough on perhaps less response rate data, because it's just not going to be evident, maybe on that EFS data with some complementary biomarker data. I think those are real conversations that we could have. And I say this as someone who would be having them, or would, you know, so, you know, I think these aren't pie in the sky. Like, I think these are conversations we could have.
Do you want to address any of that comment? We'll kind of open it up to a little bit more of a
Sure.
panel discussion. here.
Right. I think you, Melinda and Harpreet brought up a lot of really great points about the sort of difficulties in conducting these trials from the industry perspective and the academic perspective, as we heard earlier from Dr. Janeway, and sort of brainstorm about what are the potential areas where we could exercise flexibility. On the subject of, externally controlled trials, in other oncology diseases, that's something that we have considered in the past. I think the caveat there is you need to talk to us about it like very, very early on, and because there is a lot of considerations that go into it, a lot of potential introduction of bias. For example, patient characteristics need to be matched. Often there's missing data. I don't know if Dr. Janeway, if there's been any efforts in this area in the osteosarcoma space, but from my understanding, because of the heterogeneity of this disease, it, I think, is a very tough space to incorporate external controls. I think that the development of other biomarkers and sort of using those to support what we're seeing with EFS benchmark data also really helps. The more pieces of information we can put together to assess drug activity, the better, especially when there's a little bit of uncertainty maybe around one of those areas. And I don't know, Dr. Dresner, if you wanted to add anything about the
No, you're doing a great job. You're talking about one of my favorite topics. So I wanted to come over and give my input, but that was a great response. Are you doing?
You can chime in about the external control issue if you like.
So no, I mean, I agree with everything Kristen is saying. And I just wanted to sort of add some perspective on that. As she said, it's vanishingly rare that we've been able to successfully utilize an external control as the primary evidence of efficacy in a marketing application for approval. We've done it one time in oncology in the approval of a eflornithine for high-risk neuroblastoma. And the external control in that case was a large children's oncology group study for which we had full patient level data, and the vast majority of the patients on the experimental arm in the eflornithine trial had directly come from that COG trial. So that reduced some of the uncertainty, knowing that we had a data set that we were able to apply numerous sensitivity analyses and propensity score matching to. But Harpreet did mention an interesting concept of a hybrid control arm, or borrowing outside data to augment a control arm in a disease that's A, rare, and B, you may not want to randomize as many patients to a control arm. And I think that is something that we are willing to consider. We've had relatively few programs actually you take that idea and sort of run with it and actually meet with their statisticians to determine, what that will entail. Because ultimately a hybrid control arm is still an external control. And so there's still comparability concerns with whatever you're taking as your external data. And so that still needs to be vetted through us, as Kristen was saying. But in general, when we consider external controls, the first thing we ask is randomization feasible? If randomization is feasible, then we would be happy to work with any of the other sort of regulatory flexibilities that were alluded to and discussed, but that's probably the direction we're going to go on. If we consider randomization to be infeasible, then our next question is going to be, what is your assumed effect size? Because one thing that I think often gets lost in the discussion of external control is that if the assumed effect size is not large, then an external control will not be a successful way to bring the drug to market, because we, in utilizing an external control, we need to account for multiple known and unknown factors. And as you are, because you're not randomizing concurrently, and as you're trying to line up all of those factors, you start chipping away at that effect. And so if your effect size isn't going to be large to begin with, we're going to most likely come to the conclusion that we can't tell the difference between the investigational arm and the external control.
Yeah, and I just just to add to this, I mean, it doesn't necessarily have to be like, there could also be some pragmatic prospective data collected, like Pragmatica Lung, which was this hugely successful, streamlined, pragmatic trial in second-line lung, which was open at over 100 sites, very minimal data collection. If it's a known therapy and we agree on the endpoint is EFS at 4 versus 12 months, and you only need to collect, I mean, we need to probably take the lessons from Pragmatica Lung, which were rapid enrollment, rapid site activation, and unfortunately it read out as a negative study with an overall survival endpoint. That being said, I think you can extrapolate from that and say it doesn't all have to be these historical, chart reviews. It can also be just data that's collected prospectively, but ultimately, you heard this, before patients are randomized, they know what they want to do. And that's in an adjuvant setting against placebo. Even against an active control, it's hard to salvage a trial where there's asymmetric dropout. Even if I think we need to go past, is randomization feasible based on patient numbers, and say patients vote with their feet. And if the company just can't run this study, they'll abort it and we won't make progress. So, it's a little bit more than a yes, no, patient numbers allow for randomization. And I also we struggled with this, Nicole, a lot while I was at FDA, is getting companies to work together on the common control. Osteosarcoma is not the only rare disease where we struggle to make headway. And in some cases, companies were fighting against each other to be first to market. And the RET example in medullary thyroid cancer was an unfortunate, if you followed that story, Blueprint ended up having to pull their indication because they could not conduct a randomized study in this rare population because Loxo kind of beat them to it and no patients were willing. So now patients with this disease only have one FDA-approved therapy. That was not the desired outcome, and they were competing for patient numbers. Had they worked together on a common control, and we, on full disclosure, I tried to talk to both companies separately. There's a whole process, and I think we attempted this 2 or 3 times for different indications over the years, and we were never successful there. So, when we talk about stakeholders, I don't like when I hear FDA didn't want to approve such and such. I think that's not the correct terminology to use, but it's more than just the FDA and a company. It's really the community, and for that, I'm very grateful that we're all here together.
Yeah, and I agree. And I think our assessment of whether randomization is feasible or not doesn't just account for patient numbers. I think in the eflornithine example, which is a public example and was discussed in an advisory committee publicly, we actually by patient numbers did think randomization was feasible, but there were a lot of other things that went into that determination. And so that isn't the only criterion we would use. It is an important one, but not the only. But yeah, it's your point is well taken about that. Okay, great. We're going to actually, since we're all here and having an interactive discussion, we're going to start session 5. And so, Dr. Davis and you want to go? Sure.
Amy, did you want any more folks up here? Okay. All right. So, am I just introducing myself? I'm Laura Davis. I am on the OSI advisory board, and I am also a medical oncologist, pediatric as well, at Oregon, and specializing in sarcomas. And I'm honored to be helping Nicole moderate this session. Do you want to reintroduce yourself since it's been a little while?
Yeah, sure. Sorry. If you couldn't tell, my name is Nicole Dresner. I'm the Deputy Director of the Division of Oncology 2, which is previously Harpreet's division when she was at the FDA, and I'm thrilled to be here. I think what we wanted to start out with was asking if there's anybody in the room that has questions that they'd like to ask. We have Amy on one side and Erica on the other with microphones. And please feel free to come up and ask your question. Dr. Hellman, maybe you can kick us off.
Great conversation so far. I have a question, Katie, about cohort 1 and cohort 2. Because if I remember right, the data from progression-free survival in cohort 2 was based on one study. Is that right?
It was based on one study.
Yeah.
In the original publication, we're working towards a subsequent publication that will aggregate or at least report the way that unpublished figure that I showed you, kind of our more recent experience with that group of patients.
So what is that? Does it look the same or is it change?
No,
Yeah,
that's the challenge with that group of
That's my
patients
worry,
is that
yeah.
each of the trials has a slightly different EFS with sort of, but none of the trials achieving that bar that we set for activity. So they're sort of in no man's land.
So,
and so there are multiple possibilities
okay,
for
but
that.
let me ask you my question then. I understand it's much more complicated. So it seems to me, wouldn't it be better to just focus on cohort 1 and get signals quicker than to try to use one that we can't quite figure out because they're so more heterogeneous? I just wonder about that.
Yeah, so it's a really good question that you ask, but I would say that there are people who believe that if we just focus on the sort of metastatic unresectable patient population, let's call them that, many of whom have had prior lines of therapy, that we may miss a signal of activity of a drug that might be more efficacious in a sort of minimal residual disease setting. And there's a real focus in osteosarcoma, sort of biology research, large around kind of the what's special about the pulmonary microenvironment for hosting micrometastatic disease. And so that cohort 1 patient population doesn't really address that potential biology. How important that unique biology is versus just finding drugs that are active is a matter of debate, and I have my own opinions about it, but I'm not going to pollute the room with them. But so that is one of the reasons why that cohort 2 study population remains, I think, an important area. What we have done in our own investigator-initiated trials is to have cohort 2 be a little bit more of an exploratory endpoint where we can get biology samples because, and I think actually I credit Melinda with this idea, you can get paired pre and post treatment samples, for example, in that patient population. You can allow them to enroll and get access to a drug, but without having to power your study based on that patient population, because it is a smaller patient population.
I like that much better because samples are important, but
Katie, or maybe somebody else, can you speak a little bit to, this other topic that we've been engaged in a lot is the use of real world data and how you might be able to use real world data to influence the design of clinical trials, particularly linking the real world data to the genomic biomarkers, which I think is something that is increasingly possible to do.
Yeah, maybe I think like the whole discussion about these synthetic controls or I forget what you called them, the additional what did you call it, Harpreet, when you have an additional population of patients? That's sort of about this real world data. It's the same conversation. I agree in general with what my colleagues here have said, which is part of the problem in osteosarcoma. It's not like neuroblastoma. In neuroblastoma, we know what the groups of patients are. We know you have MICA amplified disease. We know that age matters. We know that Shimada classification, whatever that is, matters in terms of outcome in a particular patient population. We're able to match patients based on those characteristics. In osteosarcoma, other than this like kind of like just observational cohort 1, cohort 2 in this metastatic patient population where we're doing signal finding studies, we understand very little about the biomarkers, the biology, the clinical features that actually impact how those patients are expected to, what's expected to happen to their disease with any given treatment. And so that makes it very difficult to leverage real world data. What we need to do with that real world data and with our current set of trials is to make sure that we're getting that biology data in conjunction with our either therapy at our own institutions for the real world data or in conjunction with our clinical trials for the clinical trials that we're conducting. So we can create that data set. So that we know, this is this type of patient and that's that type of patient. For our current generation of trials right now, we don't have that robust genomic characterization. There was a comment in the chat, a question in the chat about can we make molecular characterization standard of care? There actually is an effort underway that really is a pediatric effort to say that we should be getting that molecular characterization on every patient. So hopefully that will help. But the problem is that clinical molecular characterization doesn't go into a database that's easily accessible and linked to the patient outcome. Okay, that's enough about that.
Hi, I'm Robert Pettit, Chief Medical Officer of OS Therapies. And I wanted to really go back to the cohort 2 discussion a little bit because we happen to be the highest 12-month EFS in that cohort 2 at 32.5% versus a historical data set that represented a 20% expected rate. But yet my question really focuses around is the success criteria in that design perhaps a little too high or maybe not necessarily suited to all mechanisms of action? Because we just found out literally yesterday that our 2-year, 24-month overall survival in that study is 75%. And, in these resected pulmonary, osteosarcomas. And so something that was just not quite able to meet the cutoff in the beginning is looking like it's going to be associated with a good survival. And that may have something to do with the mechanism of action of the way some drugs work versus others work. And I'm encouraged as I listen to the discussions, hearing people talk about flexibility and collaboration, because if you don't have something that fits, into a standard box, it requires a lot of extra thought and flexibility and consideration. And so I'd just like to, ask for your advice to really how a company like us, which is dedicated to osteosarcoma and literally has this many employees, how we negotiate the expense of conducting these trials, how we please our patient population who are going to be calling me tomorrow morning asking for compassionate use. I guarantee that, with the regulatory requirements to get this out there for the patient and not jeopardize public health, but make it available, because it took us, we put 9% of the patients with that disease setting on our trial while we conducted it. And so, it takes a long time to get these patients in these ultra rare diseases. And I'm all for subdividing diseases based on molecular biomarkers. But if you've already got a teeny tiny population and you've shaved that down to just a subgroup, how are we ever going to get enough data points to make a regulatory decision in these teeny tiny nano diseases? So I guess it's a rambling question, but really what I'm looking for is guidance on the best way to collaborate between patient advocacy, the agency, academic institutions to get these drugs out to patients in a disease that's frankly been underserved for 30 years by big pharma.
Sure. So I can take that question. I think, well, it's hard to give a specific advice on a very specific development program and situation. I think these are the scenarios where we really want to talk to companies one-on-one and think about maybe creative ways where you can create sort of supportive evidence. Are there other, data points we can pull in and sort of create more support if, maybe you're not meeting an established benchmark, what other data can be generated or leveraged to help bolster the development program and provide additional data so we can get to that substantial evidence of effectiveness.
I just want to add to this a little bit because I think a lot of times we come together to these meetings and they're strategic in nature, which is great, but it's going to be very hard to get us all in a room at once. And I think sometimes we need to get down to tactical discussions. And what I have found is a tactical challenge with my biotech clients. You are not a client of mine, but you very much fit the profile. We could talk later. But I think what happens is they do go to FDA and they do prepare a briefing document and they have the list of questions, and then they get feedback, which is go think about this and go work up this study design. And then they do that and they work through every single possibility on this, everything that FDA throws out at them. And again, this is not a critique. I did the exact same thing. And then they find that they're hitting walls at every step. It's not that easy to do a 2-to-1, et cetera. And so I think a tactical potential, strategy would be, can we, and then they don't have the time to wait another 60, 90, 120 days. They have to move. They have to talk to investors. They have to say, we have a plan, or they get no funding and they don't have a plan. So I think it'd be interesting to harness things like Project Catalyst and others to actually say, can we have some breakout session where we actually take more than these 50 minutes that's such precious time with FDA, and have more, yes, product specific, development specific discussions with companies in short turnaround, with longer time, where we are actually pressure testing the ideas that FDA is kind of throwing out to get more quickly to where we know that there are natural roadblocks, and then try to work through them. I'm not asking Nicole or Kristen to answer this right now, but I find this in my day-to-day work to be where the strategy and the tactics aren't fully aligned.
Thanks for that comment. So I don't know if you want to say anything, Martha. I'll just say, I agree. It's very easy to sit in the meeting and for us to say, oh, consider 2-to-1 randomization, consider hybrid design. I will push back a little bit in that I think that on the rare disease team, we very much consider things in a product specific way. And we spend a lot of time internally trying to ascertain what the realities of what we're asking for. If we're saying, oh, it's a rare disease, you can utilize a hybrid control arm, we spend a lot of time on the back end with our statisticians trying to figure out what that actually means for the company. How, if we say things like you can use a relaxed alpha, we spend a lot of time running the analyses with our statisticians to say, how many more patients does that get you? And we look at that number of patients in the context of the disease and the incidence in the United States. So I think we do that. Some of what you may call roadblocks may just be that the drug product is not performing to the degree that
I will give you that. Yes. And I just want to say the NPR team, which is Neuro Peds Rare, which sits in Division of Oncology too, is I think one of the crown jewels of all of the Office of Oncologic Diseases, because all the work but I just don't know that companies see that. And I think that's agreed. That's fair. Okay.
If I could just make one point. I do think that collaboration around messaging and around what data we have and around uncertainty, describing uncertainty, is incredibly important. So for example, with the specific question that you asked and the specific data that you cited, we actually don't have overall survival data for that cohort 2 patient population that we have. I've never looked at it, to be honest. We probably can get it. And so I don't know how to interpret an overall survival of 75% at 2 years. I have no idea what that means in that patient population. And so something like a collaboration around trying to find out that data in order to help you with the interpretation of those results is an example of an academic industry collaboration that would be incredibly important. So that's one example of where sort of early collaboration and communication is really important, because it it matters, I think, what words we use and how we use them and when. So I think we want to be sure that we can put that into context, and we're happy to help you do that.
And I think I just want to pick up on that theme is that in those early experiments, the biomarkers and their role in understanding proof of mechanism. Are you hitting your target? Is it doing what you expected to do? Are you Project Optimus in the right dose range to be doing what you were hoping to do? Those are ultra important. And to put that in as part of the collaboration with the science that's able to say, this gives me confidence in that bit that we're seeing that's still in a confidence overlap. That won't work for a pivotal approval trial, but that will work to gain confidence within your stakeholders early in the trial. And I will say, I very rarely have it has to be this or this. It's a plus or a negative or a minus. There is this gray area that basically, do I need a subgroup analysis? So is that where Alejandro, our gene signature, is going to help me figure out there's a proportion of these patients that are going to respond much more? Now I'm able to design my next trial specifically for that. And so those, I think, are the things that we need to be doing very intentionally and be working at putting that money in early to find those things out. Because you get down the line and you go, oh, shoot, I didn't have that before. It is really important to show the worth of that early on. And I think that gets back to the educate and engage early. If I'm talking with FDA that this is gaining me some sort of confidence in my data, is there a way to design that trial better that utilizes that in the context of what we're doing?
Hello, thanks very much for this really interesting and amazing get together of the different disciplines. I think it's really, really great. My name's Amos Het. I'm from Perspective Therapeutics. We're developing radiopharmaceuticals. We have a product that addresses many different tumors, and I think that's the case for a lot of drug developers. Sorry. Usually drug developers want to treat as many patients as they can with their drug. And so the opportunity in rare diseases like osteosarcoma really for us is more rapid approval than the large indications, because you are hopefully smaller trials, possibly a much better signal. So the idea is that we could rapidly go to an osteosarcoma indication it's cheaper, it could be quicker, et cetera, et cetera. That's the attraction here. I guess what I'm hearing is that that's not necessarily the case by the sounds of the strategic input from both the clinical side, where you're talking about several hundred patients, which is a several $100 million trial for a addressable market of maybe 800 patients a year in the US. I'd love to hear the panel's thoughts on that, because I think that might go to the key of why we haven't seen drugs developed in this space, and we haven't seen breakthroughs like we have in some other indications.
Yeah, I agree. You know, both the rarity combined with sort of maybe carving up different patient populations, combined with sort of a lack of sort of intermediate endpoints we can use in terms of, you know, for actually regulatory approval, not just signal finding, makes it really difficult. It's sort of, you know, it's hard to sort of even brainstorm like how to approve drugs faster without the data that you need. And I think that's the challenge. We still have to determine that a drug is effective in a rigorous way. I think that a key here will be developing and interacting with us regarding maybe some of these novel endpoints that I know are still in development, like circulating tumor DNA. I don't know if there's any consideration to PET as as a response endpoint that may be more informative than ORR, and if that is being incorporated into trials. But I think sort of those novel endpoints, I think, are really, really needed in order to enable us to outline a clearer pathway for an expedited type of approval.
And I'll just say, I think your question is outstanding. I think that's the work that this group has to do together, because if we don't answer your question, so I think we've figured out how to do signal finding studies. I don't think as a community, or we've partway to it, let's put it that way. I think there are a lot more designs that could be employed that are maybe not being fully utilized. I think your question about how do we translate that signal finding into a path for regulatory approval is why this meeting is so incredibly important, and why it's so important for all these people to be in this room together, because that we have to figure that out, or else we're not going to serve the needs of the people that we started the day off with, or the people who are making the drugs. We've got to figure it out.
Hi, thanks for the conversation. This is Martha Donahue with FDA. I do want to ask a question related to something that somebody has asked in the chat, but I wanted to touch a little bit upon the last question as well. And I think this is where we need to have creative approaches, where we're thinking very prospectively at the beginning of a development program. How do we set ourselves up for success? And acknowledging that we'll likely be in a scenario in the absence of a complete blockbuster drug that we're going to be dealing with uncertainty down the road. So how do we design a trial that's flexible enough to for us to be able to make it feasible, but also has the capacity to adjust over time and collect the data that we need at the end to make a good assessment of activity initially, and then eventually safety and effectiveness. And I think that needs to take place generally before a trial starts. And we should be thinking, as you said, Katie and others, about collection of biomarkers that we think are likely to be good measures of activity, even if they're not what's standard. We do have examples of approval of drugs in very rare cancers that don't, which I think Harpreet also mentioned, but giant cell tumor of the bone is one example where we used an atypical radiologic endpoint. So we should be thinking about that, and we can certainly think about that. ctDNA is another thing that Rick Pazder mentioned at a recent AACR meeting, where we were talking about early endpoints, and he very much pushed the idea of ctDNA in the rare cancer space. So, does that mean we could approve based upon ctDNA changes? I don't know that we can say that at this point, but you can envision a scenario where you have an active agent, you're collecting that, you're also looking at PET data. If the trial is adaptable enough, you could consider an accelerated approval endpoint that's then followed by the additional data to confirm benefit. So I think we can get there. I think it just takes creativity, a lot of discussion, and making sure that we're kind of balancing all of these factors, including what patients need and the uncertainty that we're all willing to handle as a community. And that is linked to an online question from Brian C., where he was asking, what is the burden of evidence necessary regarding the performance of a biomarker that would allow using that biomarker as an alternative endpoint for registration? I touched upon a little bit about that. I know others have as well. And I think this goes back to one of the comments in a presentation where we said that we need to have a prospectively validated intermediate endpoint. And that's pretty high bar. What does that mean, and how do we get there? So I guess I wanted to ask Nicole and Kristen first, and then others for their input on, how do you think, how should we be looking at that burden of evidence for a new endpoint, and what's enough?
So I think, a lot of times when we talk about new endpoints or surrogate endpoints, we talk about it being validated at a patient level and a trial level via multiple randomized clinical trials that are then able to be analyzed through a meta-analysis. And then you come to the end with an endpoint like overall response rate in solid tumors as a quote unquote validated surrogate endpoint. I think that that's clearly not something that is going to happen in a rare disease or an osteosarcoma, nor is it something that we expect. I think we have been willing in rare diseases to consider novel endpoints or endpoints that maybe aren't novel but haven't been applied in that particular disease in the rare disease setting. That said, for something like ctDNA, I completely agree, with Martha and with Kate, that these at the minimum need to be looked at prospectively in a trial. Otherwise, it's very difficult to know whether or not a change means anything and what magnitude of change is meaningful. And I know also on the other side that asking pediatric consortia or academic investigators to do that, to say, okay, not only do you have to do all of your study procedures, but also can you do every 3-month ctDNA blood draws, adds a lot of cost to a study. And I don't have an answer for that. I'm just saying that I know that on the other side, that's what we ask for, particularly if we're going to rely on something like ctDNA. We really do need a longitudinal assessment, and ideally across all trials, most trials in osteosarcoma, in order to rely at all on the results or to use it in any way during our regulatory.
I'll just say that we're fortunate that we have several prospective phase 2 trials in our various populations that I talked about, the relapsed refractory patient populations, that have included circulating tumor DNA time points using a test actually that I think Brian C., who asked the question, has developed, and we're working on bringing into the clinic. So I hope that we will have that circulating tumor DNA data to figure out if it actually does track with response and with other endpoints. So, but it's I guess one thing I've been thinking about as I've been sitting here is the resources for the work in this disease in the academic setting are terribly limited, both grant funding and just human people. So it takes us time to get the information out there. It's just a comment that hasn't come up yet. And I just thought I would make that. So that's why we're so happy to see young people here like Dr. Marinoff, who is full of energy.
I'd like to go back to the question because I do not want to dissuade a biotech or other developer in the fact that a rare tumor population cannot be a good win. And for all of the reasons that you brought up. And I think the one thing I also want to add to your conversation is you brought up radioligands. There was mention of PET up here as a response biomarker criteria. That's data from FDG PET from, I don't know, 10 years ago or something more like that. You can find something that tracks with response. But some of the things in the rapidly developing radiopharma world, a target-specific PET for osteosarcoma would be thrilling for clinical drug development in osteosarcoma. Not only could you use that as a bit of a diagnostic and a biomarker confirmation of response, but now put an alpha emitter on it in a tumor type that has all of this messed up DNA response that we heard from Alejandro, and you're throwing in those alpha particles, it could be really a science-driven win that if we could support that kind of PET across multiple trials. The other thing that has not been said here is the people taking care of osteosarcoma are multifunctional, really dedicated teams, and the patients travel for that to have the surgeon. So when you're thinking about the radiopharma development, you have it built in at the key sites that can actually do those early trials for you as well. So I do think that is a field to keep an eye on for osteosarcoma.
I think I just want to come back to this question about the endpoint and the biomarkers and Rick talking about ctDNA, because we've been talking about ctDNA for many other diseases, not just rare, but the diseases I mentioned up top that don't lend themselves to response rate. So that's head and neck, GBM, PDAC. No approvals on ctDNA yet for any of those. Much of the data on ctDNA is in a very common disease where response rate is very viable, and that's lung cancer, and that's through Friends of Cancer Research. And so I want to say that I don't necessarily think, I think about this in a composite manner, And when companies came to me at FDA with a composite endpoint proposal, I just wanted to like put the paper down and walk away because it was just, it was going to be so complicated, but we have to do complicated, hard things for this disease area. So I would think about a composite endpoint that puts, you know, we can think about the weighting, and this is something we'd have to work through with the agency with heavy stats, but with some landmark EFS. Yes, some ctDNA. It is getting less costly to run these tests. I have these in almost every protocol I look at. I know that our lab does it very reasonably priced. So I know it's being, you know, more commonplace. I mean, that's just factual. Like it's becoming more easy to do regardless of the lab. Some imaging, you know, criteria, whether it's PET or what have you. And then of course the functional outcomes. And I think you know, I dare to say this, but I think FDA in the past, we have used almost like a poor man's composite endpoint. And what I mean by that is we've approved assets with low response rates because of various aspects of the disease, very high unmet need. The disease just doesn't lend itself. So the response rates have been admittedly low. So if you look at the numerical value, you're like, that's not a knock it out of the park approval, but it was the additional components. So it was the durability. There may be fewer responders, but that response was really lasting. We were collecting patient-reported outcomes, functional outcomes. They maybe were not alpha controlled, so we had a debate about what got into the label, but it definitely contributed to our understanding of what these assets were doing, not always in the context of a randomized controlled trial. So I think we love to say ctDNA, and I do too, and I'm ready for it. And I think we're getting really close, honestly, to being able to use it as an intermediate endpoint. And I just don't think that will be enough. So I think we have to think about a more composite kind of endpoint.
Amy saying no. Sorry. I wanted to, we're going to end in 5 minutes and break for lunch, but I wanted to just give Mackenzie and secondarily maybe Anne and then Mike and Mac. I have you've been hearing us talk a lot about endpoints, overall survival, event-free survival, PROs, patient-reported outcomes, ctDNA. What endpoints matter the most to you other than our traditional clinical trial endpoints? And by endpoints, I mean, what outcomes?
Survival. That's it. Survival. We have to do better. We have to do better. That's it.
So my question was also for Mackenzie and Mike, et cetera. Mac, you've heard a lot of talk about number of trial sites and how that's a real barrier for our industry partners, right? Every single trial site increases the amount of time the trial takes and increases the cost. And so we have some limited experience now in osteosarcoma of using sort of more limited trial sites. It is feasible, but it definitely slows accrual. I guess my question is, like, what are the factors that people think about, and what are the unique constraints in osteosarcoma when you're thinking about clinical trials and travel for care? And what sites do you tend to travel to? That kind of thing. Like, what are your thoughts about how to solve this dilemma?
Okay, so we talk about this a lot with the kids, with the young adults. We also talk about this a lot with our scientific partners. Here's the reality. We complain a lot as a community about not having enough diversity in our studies. You get white people, you get people who have money. So, you know, there's a story I can tell you that it's a quick story, but it informs so much of what we do at our organization now, which is this, we had a school counselor call our director of programs and say, hey, what do you guys do? So director of programs goes through the whole thing. This is all the stuff we do. He goes, great. Can I connect you with one of our parents? Sure. Yeah. Connect away. So the mom calls. She's a single mother with twin boys. One of the twin boys has osteosarcoma. She goes, so she calls, she says, okay, so what do I do? What's the treatment? And we say, okay, we don't give out medical advice. One thing we'll say is, please get to a sarcoma center, a high volume center that treats osteosarcoma. She goes, okay, great, sarcoma center. Where do I find one of those? So we said, oh, well, just give us your address and we'll look it up in the SARC directory, your closest sarcoma center. She goes, okay, great. So hold on a second. We look it up. She is in Jamaica, New York, and her closest sarcoma center is Memorial Sloan Kettering. She goes, Memorial Sloan Kettering, that's in Manhattan. Yes, that's correct. It's in Manhattan. She says, ma'am, I don't have the bus fare to get to Manhattan. I also am a single mom, single income. What do I do? If I can't work, like for that. And she's talking about the day. She's not talking about long-term treatment for her son. She's talking about that day that I have to go take my child to a major institution that's not that far. I don't have the bus fare for me. I don't have the bus fare for my son, and I don't have childcare for my other son. What do I do? Okay, we will never have the advantage of treating that child, and for that child to have the advantage of being treated by an expert. I don't have anything for that family. Now, I know it's difficult on many levels to open up a trial in Jamaica, New York. We can never complain about not having a complete data set of Black, brown, all the types of people that live in America if we don't start to serve all the people in America, because we don't have the kind of healthcare in America that everybody thinks we have. So that's a real problem. And if we are truly dedicated, and I know we can be dedicated and also simultaneously not have the funds, and that's where the problem lie, because we all want to help all people. We're not doing that. We're not doing that, and we need to do it. We need to have trials at multiple institutions. We often have young adults who are now 18 years old. They can make their own choices about whether they accrue for a trial or they don't. A lot of those decisions are around, I'm an adult now, my parents are working, I cannot take time or money away from my parents to show up for this clinical trial. There are so many factors here that are much needed. And I'm saying this as a person who, if I were treated at my local institution, I'm 100% sure I would not be here. But we could afford to drive 5 hours to a major institution. I got treatment there and I'm here today. And by the way, today's my 15th anniversary of diagnosis. And it's great, and thank you. Yeah. Thank you, but it's privilege. It's a privilege. It's a privilege to live this life. It's a privilege to have been treated by the excellent team that I was treated by. We need this to expand. We need to do better. And honestly, right now, it's getting worse.
Now we can't even treat vaccines.
Okay.
We're not going to solve this problem, but what we can solve is multiple sites as much as possible. Whenever possible, we need to bring along the institutions that don't have a badass PI at the place, that we have to work to bring those people along and inform them and help them inform their patient and help us inform a better database that is multinational within this country that we live in, that is multinational.
Thanks, Anne. I really just wanted to make sure everybody has the flavor of the sort of added complexity of that adolescent, young adult, family unit patient population in terms of travel for trials. You know, we're going to have to take baby steps in this area, and maybe we can think together about what's the first step that we can take to making these trials more accessible to more people, but also still not open at 100 centers, because that's really hard to do in the drug development space when you're trying to, the goal is to get FDA approval so that it's available and make it standard of care so it's available everywhere. And so how do we help the adolescent young adult family unit get to the trials? That's, I think, like a baby step that we should really start thinking about together.
We have lots of ideas on our junior
my
advisory
God.
board and next gen about how that can happen. And they're pretty easy. Those things are pretty easy. They're not always super expensive. Some of them they're already happening. People just have to know that it's available. But I just also had a question/comment. How can we make sure that the data that comes out of each trial doesn't die when the trial dies? How can we have a clearinghouse database that holds all of this information so that as funders of research, we're not funding something twice that's already been tried and putting all the time, effort, labor, money into that trial?
Absolutely.
We need a clearinghouse database.
And I think we all agree 100%, our time is up. So just right before lunch, this question has come through many times. So the slides and recording will be available after the event. The slides will be posted on the event page of the FDA TBD. And then it'll also be on the OSI YouTube channel by the end of this month. So don't worry. And lunch.
Hi, I just wanted to thank all the panelists. Thank you, Anne, for grounding us and providing the big perspective, and all of you for being here today for the first part of the workshop, which is going to be broken by lunch. I did want to, for all of you online, I just wanted to thank you for your questions, your comments. I'm sorry we weren't able to get to all of them, and we will do our very best to answer those questions that we can within the next 2 weeks. So please know that we're reading these comments and questions, we're considering them. And although we haven't been able to get to them, we will do our best to get to as many as we can within the next couple of weeks.
Okay, welcome back, everyone. This the next session that I'm really excited about. Although there is no fire. I think it's a really important topic that is not well understood by many people and needs to be. And I think it's a big part of the answer to the question about why should I bother from industry. I think that question, as others up here said, is one we should focus on throughout the rest of this workshop, because if it's not worth the bother, then we're all wasting our time. So we're going to try to keep that center of mind. But I'm really delighted that Nancy Goodman agreed to join us. Nancy's had an amazing impact on the landscape of regulation and policy incentives for pediatric cancer therapeutics development, and the economics for investors that have come out of that, and I think is in a position to really advise us on how that may be applicable to the questions that was asked. So, Nancy, thank you, and please tell us how you got here, and if you would explain to us how we all got to the state of policy matters for pediatric rare cancer and rare disease drug development that we have today.
Well, thank you so much, Mac, and thank you for inviting me. This is about the most impressive disease-specific conference I've been to in 15 years. So thank you so much for putting it together. Can you all hear me? Okay, thank you. I don't have a direct experience with osteosarcoma, but I'm here because in 2007, my 8-year-old son, Jacob, woke up one day with nausea and a headache. And the story is a little bit like Mackenzie's. 6 weeks later, he was diagnosed with a pediatric rare disease of brain cancer called medulloblastoma. Jacob had emergency surgery. He had radiation. He was really never the same after surgery. He suffered very significant neurological deficits called posterior fossa syndrome. So he never had balance to walk again. He was anorexic. He vomited. He was in a diaper. He had cognitive impairments. He lost his hair. He had unmanaged pain for 9 months, for months and months. He was in the hospital for 9 months, and then he died. And so that's my experience. That's the experience of a lot of people here in the room. Or luckily, it's not the experience of a bunch of kids here in the room who are just super awesome, like Mackenzie and Michaela. So that's how I got here. I was the kind of parent who you doctors like really just, you know, your blood curdles when you meet me because like I had 36 different second opinions with doctors all over the world, you know. And the reason I did is I just couldn't, I was so shocked that like osteosarcoma, the treatment offered to Jacob was drugs that were 40 years old. There was no theory of cure. And how could that possibly be? You know, I'm not a physician, I'm not a scientist, but I've been reading in the New York Times, you know, all of these exciting cancer drug innovations and the excitement of medical research. And how could it be that it didn't apply to kids? So after Jacob died, the next morning, I put my laptop on my kitchen table and I said, you know, I'm just going to address this issue. And that's what I've been doing for 15 years. So I'm a lawyer. And a mom, and I really leaned into my legal expertise. You know, all of us are approaching this problem from different perspectives, whether it's investor, scientist, pharmaceutical executives, regulators, you know, and I chose to do it from the perspective of legislative advocacy. And so for that, what that means to me is sort of undertaking policy analysis to understand like what the problem could be that we could address and what narrow solution could make a big impact, writing the original draft of the bill and doing the original round of stakeholder discussions. Of course, it always got out of my hands before passage, which was awesome. And then championing it and, you know, undertaking years of legislative advocacy, oftentimes with Anne Graham's kids, by the way, to try to persuade Congress to pass these bills. So that's my background. That's why I'm here, and that's really that's how I came to be sitting here in this chair today.
And so, how is it possible that government has decided to put in place incentives, and what have they put in, and how'd you talk them into doing it?
I have really, really sharp elbows. So that's part of it. So look, like there's this basic distrust that academics and pharmaceutical executives have, right? We're just so pissed off and frustrated when companies, you know, like Alejandro's slide where they don't pursue drugs. It's just so frustrating. But I think we have to step back a minute and say, you know, no drug development occurs without intellectual property rights and an opportunity to achieve return on investment. It never has, and it never will. Okay, so let's take the counterfactual as an assumption. When has drug development ever occurred for the good of society? Jonas Salk with the, you know, polio vaccination, right? He said, "Oh, I don't want there to be a patent. I want this to be for the good of the world." Yeah, but he got a Nobel Prize for it. Okay, so he got a payoff, right? Even pneumonia, not pneumonia, penicillin. Okay, so it was developed, it was like first invented by a bunch of scientists at Oxford, two of these guys, Fleury and Chain. You know, Fleury wanted it to be given to the world for the good, you know, the good of the world. And Chain said, no, I want patents, I want to make some money on it. Okay, then what happened? So World War II started. And suddenly, you know, during times of war, more soldiers die of infection than casualties. So there was going to be incredible demand for penicillin. And you know what Fleury said? Don't tell everyone about penicillin because I can't manufacture enough of it. Okay. Well, that wasn't going to be a good solution. So what did they do? They went to the US, to a USDA lab in like middle of America, Illinois or Indiana or somewhere like that. I'm from the East Coast, so it's flyover country. And scientists there, Moyer, undertook all these incredible new technologies to completely transform the production of penicillin so that there was enough for the soldiers in World War II. And by the way, Moyer went and he got patents for it, and he made a lot of money. And, is that really a problem? Because people got the penicillin. So I just want to start with that assumption. And we just have to put away our concerns that we're frustrated that companies aren't acting for the good of the world. That's not their job. And that's not how they operate. And we have to find a way for them to stay in the game, to be incentivized to stay in the game. So that's sort of the that's sort of my starting assumption, okay? Nonprofit work is awesome for funding academics. It's just like and academics are rewarded. They get like honors, prizes, tenure, articles, like that's how they're paid. But it's not adequate for commercialization of drug products that academics come up with. So I also just want to put it in another phase. This isn't about osteosarcoma drug development or even pediatric drug development. All drug development has this issue of, are we going to incentivize it? Because drug development is a public good. And by that, I mean, it's non-rivalrous and non-exclusionary. My use of it, if Katie's work results in some incredible new drug, in 50 years, my grandchild's, use of this drug does not exclude her grandchild's use of this drug. And she can't, through her development of this new innovation, exclude my grandchild's use of it. And so it's a public good, and that's why we have intellectual property. In fact, intellectual property is enshrined in the US Constitution, Article 8, Section 8. So we always had this understanding and assumption, even our founding fathers, that we need incentives for companies to come in and develop drugs. That's just the given. So the question is, are the incentives adequate? Okay, so, IP was put into the Constitution, patent law was developed through common law practices in the United States. But then what happened was, society came about, they said, this isn't adequate. What we're seeing is that it's not adequate for rare disease drug development, which they defined as under 200,000 patients. So it's like not our stuff. But, so Congress developed a new concept called market exclusivity, which is functionally like patent law, to give some more incentive for drugs that had orphan status. Okay, but then they said, well, there's not enough incentive for new chemical entities or for new methods of administration of drugs. So Hatch-Waxman was passed, and they gave some more exclusivity for that. There wasn't enough incentive for biologics. They gave more exclusivity for that. There wasn't enough incentive for antibiotics. They gave more exclusivity for that. This is how it goes. And so we as a community have to, I hope, always be thinking about, are there enough incentives for companies to stay in the game for cancer, for pediatric cancer drugs? And that's what I've been working on.
So take us through some of the ones that exist today if you're developing a drug for osteosarcoma and you successfully develop a drug for osteosarcoma, what incentives are you entitled to?
So, well, one incentive which I worked on, and I think a lot of other people in this room also worked on, is the Pediatric Priority Review Voucher Program, which I originally authored in 2010. Congress passed it first in 2012. I'm going to assume most of the people in this room know about the program? Probably. Okay, so we're not going to describe, it's an incentive program. Most of us also know it expired in December of last year, which was really terrible. It almost got passed as part of the end of year budget. And 3 hours before passage, somebody who is a very high net worth individual started texting that, it wasn't appropriate for Congress to have riders on their budgets, like the 900 pages of bipartisan healthcare riders, so that person would fund, for any Republican who voted for it, this person would fund anyone who primaried them. And it happened like 3 hours before the vote. There was mayhem, and every single page of the 900 pages of riders were struck from the bill, including ours. So we've been, we started again January 1st. It's a new Congress. And, for you companies who are developing drugs or considering it, I just want to say, I'm cautiously optimistic. That's what one of you suggested I use as my phrase, that it will be passed in December. The PRV program will be reauthorized. So where we are is, should I talk about this, Mac? Like where we are in the PRV program? Okay. So it's the fourth most co-sponsored bill in Congress out of 5,000 bills. And again, Michaela, Mackenzie, and Ann's team have been really critical to that. They led a group of 150 kids who came to Capitol Hill in September to push. It also passed the House Education, Energy and Commerce Committee unanimously in September. Nobody opposes it in Congress. It's just that we're in a historical moment where it's just really difficult for Republicans and Democrats to work together. And so for that reason, there is a chance it won't pass in December, but there's consensus across party lines that this is a priority. And so I hope that means it will pass. And so please hang in there. I really think it will pass.
And in addition to priority review voucher, you get some extra consideration on patent expiration and those kind of things?
Absolutely. So under the Orphan Drug Act, you get 7 years exclusivity, which is pretty good. And also, if you look at the data, many drugs that receive 7 years exclusivity, in fact, because they're small markets, generics don't come into those markets. So it ends up being, it shouldn't maybe, but it ends up being more than 7 years exclusivity. Orphan Drug Act provides some research and development tax credits. And company and sponsors aren't required to pay the $4 million user fee when they submit their drug approval application to the FDA. So these are some nice incentives. Another really important incentive is premium pricing. And actually, Matt, I'd love to sort of have your thoughts on that as an investor, sort of how you would think about that as you think about whether you would ever invest in a company that was developing osteosarcoma drugs.
So I have invested in some that are developing osteosarcoma drugs. And my premise is that companies developing drugs for even this size indication can make a good living off an approval. It's a more difficult argument to say they can carry all the development costs and risk of failure and so forth. That's a tougher model, except for the incentives. And so I think when an investor is making the judgment, it takes incentives to get to the point that this is worth investing in. But if a priority review voucher, which would likely accompany a approval in osteosarcoma, it's hard to say what they're worth, but people have paid over $100 million for them.
I think we can fairly say it's been about 150 for a few years now.
Yeah. So that's quite a reward. That will recoup most companies' development expenses and in one fell swoop, plus the extended patent protection, market exclusivity, and those kind of things, and premium pricing, which people may not be so familiar with, but it's kind of a two-edged sword. makes me a little queasy to think about it, but the insurance companies do pay it. I mean, I'm sure you can quote chapter and verse on that, but it is a fact that's part of the economics of this situation.
What we're seeing is that there's a very, I'm sure you're all reading the papers, the public debate about drug pricing. It usually does not apply to pediatric life-threatening diseases. And the reason is from the insurance company's perspective, they don't have very many kids with osteosarcoma, so it's really okay if they're paying a little extra. And they don't want to be on the front page of the New York Times. We, with another bunch of other pediatric cancer advocacy groups, we've done that several times. So it's just uncomfortable. It's unpleasant. It's easier just to pay for the kids' treatment. And we do that because we just, our community needs this incentive. And I think that maybe going forward, or maybe not, but maybe drug prices will decrease. But I don't think they're going to decrease very much at all for pediatric rare disease drugs for indications like osteosarcoma.
I think the theme that's come out of this session, this workshop, emerging theme anyway, is that the economics of biopharma certainly favor big indications, chronic conditions, things that are going to generate $1 billion a year revenue, blockbuster drugs. That's not the opportunity for pediatric or rare disease. Not that they can't be, but if that's what you're looking for, or if that's the returns you're competing against with investors, then a lot of things have to happen. It really means everybody that's in this workshop has to really be on their A game. And there have to be the incentives from the policy point of view, because without that, I mean, the drugs do have to work. They do have to be safe. And so the science has got to be there. The regulatory environment's got to be there. But you've got to have people to invest in getting it into the market. And so the policy provisions like you've been talking about take an outsized impact in particularly the one I know is osteosarcoma. It's a big factor in the investment decision and companies' decisions to go in it. And so the work you're doing is just super important, and it's proven itself in approval so far, and we need one in osteosarcoma.
Let's have a big party when we get one.
That would be good.
The PRV program is what does it incentivize? It incentivizes drug development for drugs for pediatric rare diseases when there is no adult market, or sometimes there might be an adult market that's a follow-on, but the initial data presented to the FDA has to be all pediatric data. But the other question is, how do we incentivize drug development? How do we incentivize pediatric studies for drugs that are already developed for adult indications? And that's going to be the vast majority of cancer drug development. And I just want to point, there's the Race for Children Act, which was passed in 2017. That was another program we focused on, that it gives FDA the authority to direct companies to undertake pediatric studies of their new cancer drugs in pediatric populations through early phase 2. This is a really new program. It only became implemented fully in 2020. And of course, then there was COVID, so we may not have noticed it. And also, so it's a really great program. In December, when I hope we get the Give Kids a Chance Act passed, This program will be enhanced as well because it will give FDA authority not only to require monotherapy studies, but to require combination studies of 2 or more targeted therapies. And that's important because we all know cures come from cocktails of cancer drugs, right? These mono drug studies that we're doing are not going to save kids permanently. They're going to extend life almost all the time. And so we really have this opportunity for cure now. One message I have for academics is I hope that we can create institutions where academics have more awareness about like what companies are working on. There's more opportunity for collaboration and exchange of ideas. And I hope when academics and companies do work together, academics understand that companies are, if they're providing investigational drugs or newly approved drugs, it's not out of the goodness of their heart, because that they are for-profit institutions created with fiduciary duty to shareholders. Why are they doing it? They're doing it because they have race obligations, or because they're going for a PRV, or because they're going for pediatric exclusivity. That's why they're doing it. They are never doing it out of the goodness of your heart. So if you want to work with a company, check the regulatory status of the drug. And if you don't know how, by the way, you can call me. I have a little side business. I charge a latte from Starbucks. Triple shot skim latte. And if you work with companies, this is my one issue that's really come to my attention that I'm so worried about, is I'm seeing academic consortiums that are so grateful to get the new drug products from companies, and they're so excited that they offer to pay for the studies. They just say, don't worry, you give us the drug for free, or we're so grateful for a little bit of support on the data collection a little bit, and we'll take care of everything else. Okay, those are required studies companies are required to do by the FDA. And if you don't pay for them, they will pay for them because the FDA is requiring them to complete them. And when you offer to pay for them, who is really paying for them? Hospitals are paying for them. Because every kid you accrue, hospitals are paying like $20,000, $30,000 in subsidized fees because you're not paying the full accrual fee to the kids. And so we are draining the pediatric cancer funds of every hospital in America when academic consortiums volunteer to pay for these Race for Children Act studies.
Yeah, that's a really important point that we'll try to solve that at the next workshop. But so from here, assume you get the PRV renewed. What's on the horizon policy-wise that you'd be interested in seeing accomplished?
Well, I think it's a great question. On the incentive question, so every 5 years, FDA renegotiates with the pharmaceutical industry what user fees sponsors will be required to pay to the FDA when they submit new drug applications. And the next one's coming up for 2027. So we should, as a community, think about what we want to ask for as our new incentive. I've got a bunch of ideas, but maybe one thing you might want to do is just run some roundtables where people have an opportunity to weigh in on what they think would be valuable.
Well, one thing that strikes me after looking at the slide that Alejandro put up earlier with the mouse results and then the word that the company wasn't interested in pursuing it is there a public policy way to encourage, to enable alternative pathways for that molecule to find out, would it work in this population where there's data?
I mean, and I guess my question back to you is, do we need public policy reform, or do we need investors like you to come in and say to the company, if the company's not developing this drug for any other indication, can we have the intellectual property, or can we buy it for a reasonable price? Like maybe it's now time for the market.
my experience is from companies going to big pharma has a lot of stuff on the shelf. And for reasons that are inscrutable to me, they're generally not interested in talking about it. They're doing nothing with it, gathering dust, but it's hard to get them to turn loose of those assets.
It is tough. I think part of the reason is because the team that does the out licensing is also responsible for the in licensing. And a company is going to make or break their future based on the in licensing choices. So maybe we do that's the problem. And so maybe we do need to think like, what could maybe we need tax breaks, for example, to incentivize that. And before we get the tax breaks, maybe as a community, we could, give companies love, give them a lot of positive press, or create, nonprofit structures, and they could put the intellectual property in not-for-profit, and they could take, a 501c3 tax write-off on a charitable contribution.
Yeah, it's, I think it's definitely worth talking about, especially if it were something that might be relevant for this 26 renewal. I have also heard, and we had a little sidebar conversation on this, that some companies will pull an asset or decline to make an asset available in pediatrics or rare disease because they've got a bigger indication and they're afraid that some adverse finding on the rarer indication will tank their chances on the larger indication. I don't know, Martha, if you want to give your thoughts on, number one, whether there's anything to that.
It won't happen. I've never heard of, I know that it's a fear. I've been hearing about this for over 15 years, but I can't think of a single case in which an adverse event in a pediatric patient with cancer has caused a lasting problem in a development program. Depending on I don't want to ever say never, but we always understand that the patient populations are different, number one, that adverse reactions can occur no matter what. And particularly in a development program for a large cancer type such as lung cancer, that's where you get the opportunity to really unearth the rare, serious adverse events. And if you're not seeing it in a large patient population in a way that's impacting the risk-benefit calculus for that disease, an adverse event in a pediatric patient, although it's horrible, it's nothing we ever want to see, is not going to materially impact a program. Depending on the event, we might have a conversation with a company and say, let's explore this. Could this have implications? But that could happen more often in your larger program. And it's not the fact that it's a child that will change anything. I've never seen this happen.
Martha,
nor do we want it to happen.
It's my guess that when companies say that, they don't even mean it. What they really mean is they're putting all their resources on getting their drug application ready for their major indication. The financial future of the company rides on this, and their perception is that they do not have the bandwidth to go off and do like a little side project that is not furthering like the future of the company. And from our perspective, we feel like, oh, they're such a resourced institution and they're doing so great. But like from their institution, like oftentimes they're publicly traded companies and they've got to hit it on this major indication. But, and I'll just turn around to Martha, what the Pediatric Research Equity Act says, upon which Race for Children Act is based. FDA has the authority to require that the pediatric study be completed by FDA approval. Now, historically, FDA has really given companies like the right to start these studies like actually sometimes years after approval, not for cancer, of course, because the Oncology Center of Excellence would never do that. And you're just in the game since 2020. But you know, I guess what I'd turn around and ask all of you is like, what, you know, now, and I know the FDA has moved towards requiring that sometimes the pediatric study be open or the, you know, which is great, but maybe we should ask for more. Maybe when there's something really exciting, why shouldn't, you know, why can't the FDA say, you know, we're going to hold you to the letter of the law, which is that you need to complete the study. It's only through early phase 2 anyway.
Well, it's a two-edged sword, I think.
Well, FDA has the authority to do that. It has never exercised the authority, but the black letter law says FDA has that authority.
I don't want to get myself in trouble. This is my personal opinion, not as an employee of the FDA. It's hard I hear you, and it's hard I think this isn't a black and white type of situation in which I can't envision a scenario where there is a product that's has the opportunity to provide a meaningful difference for an adult patient with cancer that we're going to want to hold up their approval because they haven't yet completed the pediatric study. To me personally, that doesn't feel like the right approach. I feel like it might have unintended consequences if we kind of drew a hard line in the sand like that. And so personally, that's not something that I attacked that I think I would want to take. However, it is really important to know that the trials are being conducted with due diligence within a time frame that is feasible, within a time frame that is what we owe our pediatric patients. So for products that are directed against a target that we know from existing science that has a high potential to improve a patient outcome in a pediatric cancer, we do expect and we do apply pressure for those studies to happen concurrently with the adult development program. What we need, and we look at the timeframes so that we look at, okay, when are you saying this study, this preclinical work is going to be done that will enable you to have the information needed for the pediatric study to start? When is it going to start? When is it going to end? Why do you need that long? Like, we like to understand that. Then we said, okay, then how long do you need after that reads out to start your trial? Please provide us support as to why you think it needs that amount of time. And then how long do you think accrual will take? And how many sites are you opening? Where are you opening? What are your strategies to enroll young adults and adolescents? How are you bridging that gap? Like, we do really want to see that there's that level of maturity and thoughtfulness that's being brought to bear. So I don't think there's an easy answer to that, but, you know, I think our current system isn't perfect. What we would like to hear more from is people, you know, from members of the community, when you are excited about something, when you are excited about a molecule or a drug that's being developed in an adult, send us an email. We may not be able to respond and say we're on it or something like that, but that type of information is really helpful for us to have because we review a lot of these all the time. There's a high volume of what we're doing, and we don't necessarily know everything. Well, I know we don't know everything that the community at large knows about any particular product. So we do welcome input at any time to make sure we're doing it at the right time.
You know, I said it's a double-edged sword, but I assume the pediatric requirements are for 12 and under.
No.
What is a pediatric?
Pediatric is 0, you know, from first day of life up to FDA is a little strange in their definition. Less than 17 is our definition of pediatric. I don't really understand why less than 17. It's a bit of an arbitrary number, but so the requirements under FDARA are less than 17. However, we do not at all object. And in fact, we encourage that those FDARA studies include young adults when it's appropriate. So we're not going to say, don't run your trial in an 18-year-old or 17-year-old with 6 months of age or 21-year-old even, if it's a pediatric, a disease that we know is very prevalent in pediatrics. So it's a strange, somewhat arbitrary definition. And Nicole has something to say.
Came up a little bit in the chat, why there's sort of a line at 12. And that's largely due to the pharmacokinetic processing of drugs. Yeah, it's come up in the chat about why are we making this distinction when we say 12 and up versus, and I just wanted to clarify that that's based on body weight based on, yeah.
So I just want to, Nancy, the work that you've done in pediatric drug development is amazing. It's just incredible. And it is incredibly impactful. I think a really important question that I'm not sure I fully understand is in all of these incentive programs and then the requirements, what is the definition of a pediatric study? Like what meets the criteria to be considered a pediatric study for a patent extension or for that required IPSP, for example?
So different FDA laws and regulations have different definitions of pediatric. For the voucher program, a majority of the patients shall have first presented with the disease before the age of 18. So that's in the black letter law. For PREA, I don't think it specifically says, it just says pediatric and it doesn't define it.
Osteosarcoma is a pediatric disease, median age is 16. So osteosarcoma is considered a pediatric disease, as I think you guys all know. So that's great for the PRV program because of that, prevalence median sort of incidence.
But also, like for a required study, what an FDA could say is, we're requiring these pediatric age cohorts be studied for this disease. And then they can come back to the hospital and I don't know, can they come back and sort of say, okay, we're asking for 18 to 25 or 25 to 30, Martha, and we're going to stick that on the pediatric study because.
Yes. Yeah. No, absolutely.
Because there are so many age groups below age cohorts that would be important to study below 18.
Yeah. Now you're making me question myself, Katie. I'm pretty sure we consider it a pediatric disease. Is that right in terms of FDA? I think so.
You can
I'll
also
have to double check.
But
go ahead, Nancy.
So FDA law is really different from other areas of law in the United States because there are very few regulations. And historically, there are very few regulations because it's just so burdensome for FDA to write regulations. Then you need public comment and back and forth. And so sometime historically, FDA and industry sort of came to this informal agreement that they would do what they call guidances instead, because that way FDA can write them really much more quickly than they write regulations, and they can sort of be more detailed. They don't have to worry about every single word and the regulatory implication of it. And industry sort of perceives these to be general guidances of what they're going to do. So in fact, it would sort of come out to be is it's very discretionary. FDA has a lot of discretion as to how they're going to define the requirements. And that's very different from other US agencies. And sort of historically, industry has accepted this trade-off because industry wants a lot of guidance to clarify, how the FDA is thinking about drug development on so many different issues. And it's, this way the FDA can just pop out guidances as quickly as they can. As long as they retire, 12 guidances for everyone they write now, but yeah.
I don't think draft guidances count.
Okay, good. Okay. Yeah.
Luckily.
So that's that's the trade-off FDA and industry has made. And I think everybody's pretty happy with it. So the answer is I think we're okay. We don't need a specific definition. We'll be okay.
And we do have a fluid way of sort of thinking about it for the most part. And all this is making me just want to say now, before I forget to say it, I just want to thank you, Nancy, for all of your support for the FDA and all the work that we do. And thank the whole community, actually, for their support of our work. It really means a lot to all of us. That doesn't mean that we're immune. It doesn't mean that we don't want criticism. We want criticism. We want you guys to push us. But, it means a lot to those of us that are working in the government, particularly now, to know that, to some extent, you guys recognize that the work we're doing is, that we're doing our best to try to advance our mission. So thank you all.
If I could just say one more thing to that. So when we put together our black letter law, whether it be for the Pediatric Priority Review Voucher Program or Race for Children Act or Give Kids a Chance Act, which authorizes the FDA to ask for combination studies. Our basic philosophy at Kids versus Cancer and the community of scientists we work with, which includes Katie, of course, is to give FDA maximal discretion, because when you have black letter law, like it's this rule that may not apply to whatever science is in 5 years, right? Or may not apply to all the instances we're thinking of. And so we just trust that the FDA is just going to do their best just to make the best decision they can. And so we really try to draft our bills to give FDA the flexibility and discretion to make those kinds of decisions with whatever new information comes up.
Great. Well, thank you. We are out of time and moving on to a session to try to synthesize and come up with action plans for all this. And Dr. Merchant and a big cast will come up and take us through that. Thank you very much, Nancy.
So we'll ask the whole panel. We'll ask the panel to come up and be seated. We're going to have a presentation first by our FDA colleagues, and then also by Allie, and then sort of open it up to the whole group. I'll again present. I am Melinda Merchant. I am a pediatric oncologist, actually, who trained at the NIH and Memorial Sloan Kettering. I left that out before, and I'll ask everybody on the panel to present themselves, introduce themselves, and present your disclosure of conflict of interest when it comes to your time as we talk through. So without much more ado, I think, Katie, you're ready?
Yes.
And
All right. I think this is on. Yeah. Yeah. All right. Well, thank you and good afternoon. That was a really great last session. It's hard to follow. My name is Katie Barnett. I am a pediatric oncologist and a medical officer in the Division of Clinical Evaluation Oncology in the Center for Biologics Evaluation and Research, or CBER, at the FDA. My work at the FDA and CBER focuses on evaluation of cell and gene therapies, therapeutic vaccines, and other biologics for the treatment of pediatric and malignant solid tumors. So today I'll be discussing FDA expedited programs, so I'll move the slide, and other efforts to advance product development for rare cancers. I have no financial conflicts to disclose. Today, we've discussed many challenges with developing drugs and biologics for rare cancers, such as osteosarcoma. This session will focus on the path forward, and I will provide a brief overview of several FDA programs that may support sponsors developing treatments for rare cancers, including expedited programs, orphan drug designation, as well as other initiatives. First, I'll discuss the expedited development programs. So these programs aim to facilitate and expedite the development and review of new drugs and biologics that address unmet medical need in the treatment of serious or life-threatening conditions. Dr. Jasmine Smith discussed accelerated approval in her presentation earlier, and I will cover the other four in bold here. Each program applies slightly different selection criteria and focuses on different stages of product development. So starting with a fast track, breakthrough, and regenerative medicine advanced therapy, or RMAT designation. These are requested at the IND stage, typically prior to an end of phase 2 meeting. There's important differences in the criteria and the level of evidence required for each of these designations. All three require that the product is intended to treat a serious condition, which is typically met by any cancer development program. Fast track designation also requires that available data demonstrates that the product has the potential to address an unmet medical need. And for fast track, this can be demonstrated by either preclinical or clinical data. Breakthrough therapy designation has a higher bar. This designation requires preliminary clinical evidence indicating that the product may demonstrate substantial improvement over available therapy on clinically significant endpoints. Finally, regenerative medicine advanced therapy, or RMAT, applies specifically to regenerative medicine therapies, as the name says. These are products that are regulated by CBER, the Center for Biologics. This includes certain cell therapies, gene therapies, and therapeutic tissue engineering products. This designation also requires clinical evidence indicating the potential to address an unmet medical need. So as opposed to breakthrough therapy designation, RMAT does not require evidence indicating that the drug may offer a substantial improvement over available therapies. But it does require clinical evidence, unlike a fast track designation. So what are the benefits? The benefits of all of these designations include more frequent meetings and written communication with the FDA to discuss development plans. In addition, products with these designations can be eligible for accelerated approval and priority review if supported by the clinical data at the time of the marketing application. They may also be eligible for something called rolling review, where we review portions of a marketing application before the complete application is submitted. In addition to those features, breakthrough designation includes more intensive guidance on efficient drug development. That includes more involvement of senior managers. RMAT designation, which is that CBER expedited program, encompasses all of the benefits of fast track and breakthrough designation, including intensive guidance and opportunities to discuss ways of supporting accelerated approval and post-approval requirements. And there's guidance is available online with a lot more detailed information on all of these programs. Priority review is another expedited program that's requested later in product development at the time of submission of the marketing application. To qualify the product, again, must treat a serious condition, and then if approved, would provide significant improvement in safety or effectiveness. The benefit of priority review is a shorter clock for review of the marketing application. The standard review of a marketing application is 10 months following the filing review period, whereas priority review is only a 6-month review clock. So those were expedited programs. Those are not necessarily specific to supporting programs for rare cancers or diseases. Now I'll shift to an incentives program that was mentioned earlier and I think in the last session called Orphan Drug Designation, which is specifically to support development of products to treat rare diseases. The Orphan Drug Act defines a rare disease as one that affects less than 200,000 people in the United States. This designation qualifies sponsors for various incentives, including tax credits, waiver of that marketing application user fee, and then potential for 7 years of market exclusivity after approval. And these requests can be made or can be submitted at any time before the marketing application. The Orphan Drug Act also established the Orphan Drug Grants Program to provide funding for developing products for rare diseases. So this includes the Clinical Trials Grants Program, which is a competitive program for monetary grants that are given to fund trials for rare diseases. In 2024, this program awarded more than $17 million to help support conduct of 7 clinical trials over the next 4 years. And some examples in the last several years, some of the recent grants were awarded to clinical trials in rare sarcomas, such as liposarcoma, also pediatric brain tumors, and others. I've listed some on the slide here. There's also a natural history grants program that funds well-designed, protocol-driven natural history studies in rare diseases. So those are some of the FDA-wide programs. Now I'll shift to more oncology-specific initiatives. For rare cancers like osteosarcoma, as previously mentioned in other presentations, we recognize that a one-size-fits-all approach to product development is not realistic. The Oncology Center of Excellence has several programs several of them are shown here to help address the unique needs of specific patient populations and product development programs. And I'll highlight just a few of these programs here. So the Rare Cancers Program works with program leads for many of these other OCE programs, and also the review divisions in oncology to help address the needs for rare cancer product development. And this includes interaction with the patient-focused drug development program, Project Catalyst, which was mentioned in an earlier presentation and works primarily with small companies and academic incubators to foster early stage product innovation, and Project Endpoint, which aims to engage with the community to enhance development of endpoints in oncology drug development. So this slide doesn't represent all of the OCE programs that can help address challenges in rare cancer development. On the OCE website, you can find links to all of these programs as well as others with a lot more information. I also wanted to highlight there are product-specific programs as well, including the oncology cell and gene therapy program. And as a reviewer in the Center for Biologics, I wanted to just take a minute to highlight cell and gene therapies that we review in CBER. These products are often developed to treat rare diseases. So we do have a lot of experience thinking about product development for rare diseases in CBER. In addition to the challenges that we've kind of already discussed today, as far as developing products for rare diseases, there are also unique product-specific challenges that I just wanted to highlight for developing cell and gene therapies. Some of the unique aspects, and this isn't all of them, but these therapies often have complex manufacturing issues. They can involve lymphodepletion and invasive procedures with associated safety risks. They have distinct pharmacodynamics and pharmacokinetics and safety profiles with the potential for extended persistence and late effects. And so these differences can require elements of trial design that are distinct from maybe other types of drugs, including specific safety monitoring and long-term follow-up requirements. And so we encourage sponsors developing these products for rare cancers to engage with us in CBER early to discuss your development plan and trial design. And finally, I just wanted to highlight at the FDA, we do support the use of innovative trial designs, which can be valuable for improving efficiency and reduce development timelines for drug development in rare cancers. Dr. Wessel mentioned a few examples of innovative trial designs earlier, such as seamless designs and platform designs. Some other examples are master protocols with basket trial designs, where one product is evaluated across multiple cancer types, and umbrella trials, where multiple different products are evaluated within one tumor type. Another type of umbrella trial that's specific to CBER regulated products is where multiple versions of a cell or gene therapy product are studied in a single disease under a primary and secondary IND framework. So instead of an iterative approach to clinical studies, multiple versions of a cell or gene therapy can be studied in parallel, which may streamline clinical development. And we have a lot of guidances available with more detailed information on these trial designs. So in conclusion, we've discussed today the challenges of developing novel therapies for osteosarcoma. We understand that this area requires more than a one-size-fits-all regulatory approach. I hope today we've highlighted some of the ways that FDA can support sponsors developing new treatments for rare cancers, including our expedited programs, incentives, OCE programs. Most importantly, as emphasized today, collaboration amongst all stakeholders is really key to advancing development and expediting access of promising therapies to patients with osteosarcoma. And I just wanted to, last slide, we have a number of other guidances that are relevant to drug and biologic development for rare cancers, and we really encourage stakeholders to use these resources as they consider their product development approach. And with that, I would just like to thank my colleagues and the OCE, as well as the Osteosarcoma Institute, and everyone for listening. And I think now I'll hand it over to Ali Murdoff. Is that right?
Thank you. I'm Ali Murdoff. I'm a co-founder of an organization called Battle Osteosarcoma that raises money for osteosarcoma research, and I'm a trustee at the OSI. My daughter Charlotte, so beautiful. She lost her life to osteosarcoma in 2021 when she was 18. I thought she would be a survivor. She had localized disease at diagnosis. She had a really high necrosis score. She didn't have any hiccups with MAP. She rehabbed very easily until you have one of those routine scans and a tiny met shows up. And the rest of her life was horrific. My husband and I searched the globe for answers and knocked on a lot of doors, turned over as many stones as we could, but there just aren't any. And I don't think there still are today. We spoke to many doctors. Lots of you are in this room today. And, you know, we were offered chemo that has limited success and can cause secondary cancers, or worse, or taken into a room and told there are no more options. So I'm here to just say we can't keep doing the same thing and expect different results.
Thanks, Ali. It's powerful. As you're using Charlotte's voice here to tell us a lot of the why, and we've heard that this morning, and I think actually Dr. Burnett too, for your framework of the path forward is what we've sort of described this. And the path forward has to be a road that takes into account many of the gates that are required, has to take into account the end game, which is something more effective, less toxic for some very special people. And so that they don't have to go through the things that you've gone through and some of the other things we've seen. So what we wanted to do as we were putting this together is actually, these are two very important stakeholder representatives here. They're representing many of the stakeholders in the room. And within industry and the development pathway, we do a lot of discussing about stakeholders, stakeholder management, who it is, how to influence, how to advocate. Ali wears an advocate name tag essentially in how she comes sitting in the seat, but all of us become internal advocates within our programs for our science, for our patients, for the goal and the vision that you're seeing. And what we wanted to do was take a couple of instances that are examples of success and things that have met similar challenges to osteosarcoma, and also how there is development going on in osteosarcoma and what were some of those key interactions. We're not asking everybody to tell us exactly about the program. If you want to hear more about that, the things that aren't public, that can be in separate conversations. This is to help map out that road, to give some lights for where are some of those key interactions, where are some of those key stakeholder conversations that need to be happening. Mentioned it earlier today. I think each one of you guys has mentioned it too. Early is important. And so those conversations, those relationships, they grow with time. That trust is a really big category, from investment to the challenging path that clinical development is. So we're going to start with a couple of case studies. And Davey, I'm going to pass it to you first to introduce maybe yourself and the day one mission that you took on to get to what we would call success for another challenging pediatric tumor.
Thanks, Melinda. Can you hear me okay? Thank you. Well, thanks, Melinda, and thanks, everyone. It's an honor to be here with parents, patients, and everyone else in the room to try to find a path forward together. So my name is David Curtin. I'm the Chief Development Officer at Day One Bio. Day One is actually a company that was started in 2018 to address one of the issues that we've described and discussed earlier. Is basically convert what Nancy described as a side project in a big company as the main project for a company. And that's actually what we did with a drug that was at the time called TAC-580, now tovorafenib, that the company actually purchased from Takeda to repurpose it beyond the GI solid tumors that Takeda was looking into to develop it in pediatric low-grade glioma. So that was back in 2018, 2019. The drug is now on the market in the United States since last year. And I'm happy to go over some of the steps that we've taken to get there. But it took a lot of, to your point about stakeholders, a lot of interventions from all of the parties here in the room today to actually make it happen. Started with advocacy in the patient experience.
So in our pre-discussions, we also discussed a separate study in selumetinib, which was in peripheral neuroblastoma. And Dr. Brigitte Wiederman was going to come and tell that story about some of the key collaboration in the private public partnership, actually government partnership from NIH. She's unable to be here today. We met last night to get a little bit of key things to come across. So I'll try to bring across some of her lessons learned to apply this that she would have been saying in here. But Fernanda and I will try to give a little bit of perspectives. The reason why I mentioned I had trained at NIH, who Fernanda did too, is we both enrolled patients on this study, right? Plenty of patients on this study as the docs in the clinic. And then to watch it go through the pathway of an investigator-initiated study, which is what we've been talking about for a lot of things in the concepts of what we heard from our clinical and academic colleagues this morning. An investigator-initiated study at a big company. It was an AZ drug that had lots of other sponsored trials that were attempting to find a signal in larger populations. As time went on, it became clear the credibility, the incredible amounts of natural history, experience, and clean data that Dr. Wiedemann had from that study that had been, she had 10 years of natural history, multiple studies that didn't quite hit, that didn't meet a response curve in a tumor that, quite frankly, we consider benign. And it was hard to get people's attention for. Plexiform neurofibroma happens in NF1 patients. And for a long time, they didn't have anything for these big disfiguring issues. And so here is the essence of a clinical scientist, in my opinion, in Brigitte, who says, this is a problem that's happening. Here's the science behind it. We're going to hit each one of these nodes in science on the way. There was rationale for lots of things before there became a success. And then there was a success. And I think what I want to change that broader story into is to sort of say some of the things that were key challenges that then Davey and others can talk about, key challenges, how they reflect in those other environments too. And then lessons learned for how we can apply that to development here. So let me just put on my eyes a little bit more so I can say exactly what Brigitte had said, which is that really there was an inability to meaningfully measure response. Sound familiar? There was no validated PRO. It happened in young children, and you really needed to make the difference in young children, not necessarily in the older adults first and then come down to the kids. So that took a little bit of the pediatric. And it was really 10 to 20 years worth of work. in that area before you started seeing these kids remarkably. And there's other details to it. It is a wonderful story to go and look further into. There's videos about it. There's kids out there that it's really worthwhile. But what led to success? Collaboration and persistence. Longitudinal natural history study, MRI analysis to characterize growth, interventional trials with a new way of looking at tumor response. They did volumetric MRI in this case, and incorporation of the PROs. And all those things lending credence to the fact that what you were seeing was a result of that test article, that investigational drug, and able then to sit with the FDA. And I think, actually, I'm going to ask you to speak to that too a little bit, Dr. Barnett, or maybe even Harpreet, in that collaboration and having that academic speaker at the same time that you're having the sponsor coming in is a very, very important collaboration. Do you agree?
Yes, yeah, no, it's a great point. I think it's so important that we're kind of aligned between academia and pharma and biotech when these complicated submissions come in. I think it takes it takes a whole team. So I don't know if Dr. Dresner has anything more specific about that program to say, but
So yeah, I mean, it is important to have the academic investigators and even as thought leaders in the meeting. We do depend on their assessment of the activity of a drug and what endpoints are meaningful and what magnitude of change in an endpoint is meaningful because they're the ones in the clinic seeing the patients. And so in many of our successful programs, including selumetinib, tovorafenib, the companies included the investigators in the room. I will say that happens probably still in a minority of scenarios. And it's something that we usually ask for. We want to hear from the academic investigators. And I do think that helps move the needle forward and definitely makes the conversations more fruitful. And I think Martha wanted to add.
One quick thing. I just want to say the vignette with selumetinib, because I was working in the division when this kind of started, but even before that, before there was a drug, or maybe before FDA talked to Dr. Wiedemann et al. About the drug, she brought members of the FDA, including myself, Dr. Diana Bradford, and others, to their RAINS, the Response Evaluation and Neurofibromatosis meetings, to learn. It was two-way learning, to learn about the disease, to understand the challenges, as well as for them to learn about sort of how we look at response and things. So the only reason I mention that is because this happened, the seeds were planted before we had formal meetings regarding selumetinib. And that's exactly what we're doing today. So I just wanted to mention that.
And for tovorafenib as well, we had a lot of discussions on imaging and other outcomes, ophthalmologic exams that could be leveraged to utilize an overall response rate endpoint that included minor responses. And those discussions all started very early.
Early, it can't be said enough in this meeting. Dr. Arnaldez, can you introduce yourself and then add to the conversation?
I'm delighted to be here. I'm Fernanda Arnaldez. I am a pediatric oncologist. I'm a sarcoma doctor, now turned into a drug developer with many years of developing drugs, now at AstraZeneca as part of research and development, thanks to OSI and to my colleagues. The key thing that I see as an emerging theme from different perspectives is education, but education in which practical, tactical way. Melinda said it very clearly. We all have, in big pharma, there is a really clear need of being strong advocates internally, and not because we are good people and there are other people who are bad. It's because diseases like osteosarcoma are rare. The fact that it's rare is a really good thing. I don't want more kids with osteosarcoma. But then the knowledge about endpoints, management of the disease, regulatory adventures related to that is not widely known. So I would strongly, strongly support what was said, but also advocate for really not being afraid of doing what Dr. Wiedemann did, which is really involving other stakeholders, the agency, her own colleagues, pharma, involve them early, involve them often, and do not assume that everything else is known to the degree that was known inside the NCI, where I was at the time, by the way. So I think that is a very, very important aspect to highlight. We're still trying to fight ORR as not an ideal endpoint in early development trials in osteosarcoma, as Dr. Janeway very eloquently demonstrated.
Hey, Melinda?
Yes.
When are we allowed to comment? Anytime?
Go ahead and comment.
I always like to ask for permission. Thank you. I just wanted to actually violently agree, if I may, with that point. Some of us who had AstraZeneca actually crossed paths at AstraZeneca when selumetinib was in the process of being reviewed, developed by the FDA. Reviewed by the FDA, developed by AstraZeneca, not the other way around. It really comes down to a handful of individuals really on the team internally in pharma as well to actually push things through and convince and educate at the highest levels of management. So you do need champions, whether it's on the cross-functional team or at the review board within the company to actually help you do this. So as a community, being able to identify who those people are actually really does matter. And also what those companies are, because different companies will have a different appetite for some of that work as well.
That is very true. And I didn't disclose in the former session that I was at AstraZeneca. I was not part of selumetinib's approval there. But like Davey, I can point out exactly who were the internal advocates that were really useful and necessary. Honestly, without them, that wouldn't happen either. And so I think what you see represented up here are people who have been part of that being internal advocates. And again, this collaboration, this ongoing interaction. And Martha, I'm glad you brought up RAINS because that was the next point that Brigitte had on the list for me, which is understanding how do you actually measure, follow, call a response? And I think we've seen that done in other diseases as well. I'm thinking about Raynaud category. And Davey, you had to deal with a version of Raynaud that maybe wasn't there when you first started, but was by the time,
Did you say a version of Raynaud?
A version?
Multiple versions.
Multiple versions of Raynaud?
Well, I think the interesting case study when it comes to tovorafenib and how to measure response in low-grade, pediatric low-grade glioma, if you look at the time where the company acquired the molecule from Takeda, which was December 2019, to the time we reached out to the FDA for the first time, which was February 2020, to the time the trial was designed and endorsed by the agency in 2020 as well, the time the drug was submitted for NDA review and the time it was approved, the endpoint actually changed 3 times in terms of its definition. So the dialogue is key to that success and being able to go through that successfully. So yes, the short answer was yes. Thank you.
Were you going to say one more thing, Fernanda? Yes. Go ahead.
I completely agree with the flexibility and the ability to learn as we go. And as we were listening to in prior sessions, real world evidence, synthetic control arms are things that used to be more sophisticated and unreachable. But many of the companies represented here are really invested in improving AI to apply to the development of drugs in all different indications. So I think that we are in an unprecedented moment to leverage all those resources together as a community and continue to find the endpoints that really matter.
And by the way, too, like we talk about the FDA and the sponsor, but like if you take the case of Raynaud-Rapnol, in the case of torapatinib in low grade, being able to better understand it is actually through the investigators and also what does clinical benefit looks like for the patient. So like literally every single stakeholder in that room actually matters in that conversation equally.
So I'm keeping track of time. We're about halfway through. We're going to switch from our vignettes of things that weren't osteosarcoma to a little bit of ongoing osteosarcoma work and lessons learned for some of the pathways that we've heard from the regulatory presentations. And also, again, this idea of collaboration. How do you define success? And so I'll pass it over to you, Teddy, to introduce yourself and then talk a little bit about those concepts.
Great, thanks so much. Hopefully you can hear me okay. So my name is Teddy Soul. I'm one of the global medical affairs leads at GSK, and I've spent the past 2 decades or so in pharma, working to develop drugs for patients with unmet need, and also in medical affairs, really bringing that external voice into the organization to really drive and influence change internally. I'd like to say it's a privilege and a responsibility to be a part of a dedicated team at GSK that's working to evaluate an investigational treatment for relapsed or refractory osteosarcoma. And our journey at GSK has been defined by collaboration, both collaboration with another pharmaceutical company, collaboration with the osteosarcoma community, and also an internal collaboration amongst our teams to really band together with this shared determination to address unmet need for these patients. So to start with the external collaboration piece, our journey began with an alliance collaboration with a Chinese company called Hansoh Pharmaceuticals. And a couple of years ago, GSK partnered to globally develop an investigational B7-H3 targeted antibody drug conjugate. And this was one that was mentioned by Dr. Marinoff earlier in her talk about the ongoing clinical trials. Hansoh is conducting studies in China in patients with relapsed refractory osteosarcoma. And last year at the ASCO meeting, they presented some early clinical data that showed encouraging clinical signals in a cohort of 42 patients in China. And so this was kind of that start, again, like the early conversations that we talked about, the collaboration. We met with external experts. We started engaging internally to understand, what path forward could we have to consider development for this asset in osteosarcoma based on these early clinical signals. But we also realized that to bring this drug to market, it would require a global development pathway. It would require, a path to determine the most effective, and safest dose, and make sure that that activity is validated so it can be, brought to market for patients successfully. So we explored the different regulatory paths, and breakthrough therapy designation was one that we thought we perhaps fit into, given that we did have clinical data, albeit in a Chinese population. And we did disclose at the beginning of this year that we did receive the breakthrough therapy designation from FDA. So we were really excited about that opportunity to have those discussions with FDA, which are very much ongoing. I think a lot of that has come from, again, the strong science, but not only the science, working with academics, some of whom are in this audience, to really understand the potential that this data is showing, to understand what types of trials should we be designing. How can we effectively develop a strategy to bring this drug to market? And it was really also bringing those insights into the company to our senior leaders to really share the potential of this asset and to really push for that development for this patient population who's historically underserved. And so again, it's a lot of collaboration. We've been really grateful. The osteosarcoma community has just been so arms welcome and willing to talk with us, to brainstorm with us. I also want to thank MAC and the Osteosarcoma Institute. I think the OSI has been really key in connecting us with key experts in the field, as well as deepening our understanding of the patients, right? What are the patient's needs, the impact on patients, and again, really reinforcing, what we're trying to do to address this unmet need within GSK. I think one thing to emphasize, and this was mentioned earlier, that our journey is really just beginning. We do have to undergo that clinical testing, rigorous evaluation to make sure that it does meet the standards of safety and effectiveness. But we're fortunate to have that breakthrough therapy designation so we can have those ongoing conversations and hopefully deliver a treatment that is both safe and effective for patients. So hopefully that shares a little bit about the path that we took. External collaboration, internal collaboration, and early discussions, I think, are key themes that we've heard throughout today that really were part of our story as well.
Thanks, Teddy. And I think also one of the things that we had touched a little bit on is that definition of success and that signal finding. So as we talked through this, we talked about some science-driven, somewhat of a repurposing for a direct target. There is a signal. So you can imagine there are lots of things that we've talked about today in studies going on that could be that path and could be moving forward. Jose, we're going to switch to you and talk a little bit about the repurposing also in that after you see some signal, and how do you keep that energy moving along?
Well, I'm Jose Perez. I work in Exelixis, and my role there is as the lead in the GU, which probably makes not much sense here today, portfolio. But also, I have been carrying with me some interest for sarcoma and some other rare diseases from my previous jobs. I'm a clinical oncologist by training, and I work in medical affairs, which we are not directly involved in clinical development. So we don't do the clinical trials that are discussed with the regulatory agencies to get an approval. So we have probably we are the eyes and the ears of the company in the external world. And we get some key information of what really matters. What is the science looking for? What are the patient's needs in different diseases? So we get all that information, and we have in medical affairs different programs. One of them is supporting investigator-initiated trials. And using that interactions with the external world, with the key investigators, and also using the science that we know from the diseases, we need to receive concepts. And the investigator-initiated trials is a very delicate area that we may have some, let's say, ideas, but we need to be hands-off. And it's the investigator who provides the concept and manages the concept from A to Z. So through supporting investigator-initiated trials in Exelixis, we have the blessing, the beautifulness, I don't know, I would like to find a shining word to say now, of getting data that have proven of benefit to patients with rare diseases and osteosarcoma in particular. We saw the table that was presented by Amanda earlier today, where we have data coming from cabozantinib investigator-initiated trials showing some results. And also we saw in her presentation ongoing clinical trials that are going hopefully to prove that treating osteosarcoma patients earlier in the disease is even more beneficial than later. So as you said more than once, the earlier, the better. So also, we have a new drug that is, I would say, a family of cabozantinib. It's called sansalitinib. And we have recently activated an investigator-initiated trial program as well. And we have some approved concepts already that have not started yet. So, but there will be some additional trials in sarcoma and osteosarcoma in particular. So it's a different way of, we have a drug that works in kidney cancer and in liver cancer and neuroendocrine tumors, and that's what we do in a regular day in Exelixis. But recognizing the value of these drugs in rare diseases and the interaction with external customers, we have built this investigator-initiated program that has delivered data that shown efficacy in patients with osteosarcoma. And in the US, we have the endorsements of NCCN for some of these data, and that would allow patients to be treated with the drug. Because of the amount of data that we have was enough to be recognized by NCCN. And through that process, patients are treated with the drug in the US.
Fabulous. I like how you described your medical affairs role down there. And maybe I'll ask both you and Teddy about this as the eyes and ears of the company bringing back things that you hear. And when I think medical affairs, I first think about the PIs, the investigators, the ones who are treating the patients out there, past approval, the docs who are treating patients. How are you the eyes and the ears also for advocates to get some of those answers and comments back through you guys?
Yes, we also have interaction with patients and with patient associations, and we get what they believe is more relevant in clinical trials, what are the type of information that we need to collect in the trial, how we should discuss the design of the trials with the investigators based on the patient's perspective.
Yeah, and I think to piggyback on that, so I love the eyes and ears because that's exactly what we are. And at GSK, I have the great opportunity to be a part of the medicine development team. So I've worked, in late stage, but having the opportunity to be involved early on to bring those insights into the organization to connect not only with academics, but also patient advocacy groups, as Jose mentioned. Those insights are so important because we can be very siloed and not understand what patients need, what clinicians are looking for, and having that medical affairs function to be that voice and bring that back to be the internal advocate, I think has been so important, particularly in this space.
And so, Allie, I'm wondering if any of that resonates with you or some of the things that we've talked about today that are really important for defining success or getting news back about what makes a better trial. Can we use a little bit of the panel time to talk about that?
Well, I would say from a non-scientific viewpoint, success to a family, I think, is time. I've told Katie this, it's time to get another opinion, time to find out results of a trial, time to get into a trial, time to find an experimental drug. As Mike said this morning, you sort of become Dr. Google when you get your diagnosis, and it's not difficult to see how grim the statistics are. So, as a parent, I'm not looking for a grand slam. I'm not even expecting a home run. Just, I will take singles and doubles all day long if that means my child can live longer. So to me, that's what success means.
So as we think about that as success, we have about 15 minutes left in the panel. And I asked all the panelists this question. I'm going to ask it of the room too. And you can think about coming to a microphone and saying it. So in this cross-stakeholder panel dialogue, as we work towards developing more effective therapies for osteosarcoma, what is the most critical support or contribution you would need from another stakeholder? Honestly, it can be up here on the panel. It can be out there. It can be listening. It cannot be in the room that we need to get in the room for our next iteration of connections with the key stakeholders. What is the most critical support or contribution you would need from another stakeholder? Davey.
Okay. Thank you. Thank you for the opportunity. I have a lot of ideas, but if I'll have to pick one, it'll be for my industry colleagues actually, because the point was made earlier on the fact that when a drug is failing in a big pharma company, there's not a huge amount of time spent on trying to explore where else it might work, right? If you're developing a drug that might be a $3 billion net product value product in non-small cell lung cancer or colorectal cancer, the team is going to be, and it's not working out, the team is going to be redeployed to the next cool ADC. There's not a huge amount of time spent and not a huge amount of credit also given to the team actually going through that data and mine it. And this community, as well as some of the team members internally, might be aware of some of the potential that drug might have in osteosarcoma, for example, or other pediatric diseases. On a side note, by the way, approaching things as a target relevance rather than a single indication matters a lot too, because that's where you dilute the risk and actually have more opportunities to make something work financially. But yeah, my ask is, as a team member in Big Pharma, especially, be curious about where you could shop that around. And not as an individual, not to make, not for your bonus to be driven by that, because it won't, but really for the greater good, right? There are companies that are going to find this interesting, that are going to put the effort in developing it, put the effort at assessing it further, and making an opportunity for the patients, because the definition of worth it is going to really depend on the company that we're talking about. What's worth it for a company of 200 people with one asset on the market is different to a GSK or an AstraZeneca. So it's be curious, reach out. TAC 580, tovorafenib, was actually got to day one because of a phone call that our co-founder received from Takeda. It's like, hey, I've heard you were starting that company to address the fact that kids with cancer were not getting access to drugs. We have that thing, and we have that trial running at Dana-Farber called Pinnacle 1-4 in low-grade glioma. It looks very interesting. Let's talk. And that's how Sam got this started. So it really can take one phone call. So pick up the phone and call. I mean, you can call me, but call each other and use the network. I think that can make a huge difference. Thank you.
I couldn't agree more. Many of the things that David highlighted are absolutely true. The elephant in the room is that in general, big corporations are risk averse, and that's the way reality is. And also that since we are really fortunate to study a disease where the numbers are small, then we get into paralysis by analysis in terms of returns of investment, where private and public partnerships are a critical element of making things work. Because if we take a step back, we all want more time, good time for patients. And it takes really a village.
Absolutely. And in that village, I want to take today, like in the midst of this time where we had to represent Dr. Wiedemann from NIH, to really give a big shout out to our government public servants. So much of what we have talked about today would not be possible without the selfless work that each and every one of them bring with integrity. And I mean, like your whole hearts. And you can tell you're trying to do what's right every day. And I wish we could shout that from the mountaintop. And I think it needs to be heard. I would love us to have a better PR sense. Not that you're doing what we need for our day jobs. You are doing, and the NIH is doing what we need that you can't get investment for, that you need to have some boundaries for safety, protecting all of us. And so huge shout out. And with that, I will hand to Dr. Dresner or Barnett to say, what do you need from stakeholders to help do this process and get more things for osteosarcoma?
Start. I think, I think we've discussed a lot today, and not to sound like a broken record, but we, from sponsors, I think the most critical contribution is early and open communication. Is particularly if you're thinking about some kind of innovative trial design that can pose additional challenges, and it might require, extra careful and rigorous planning, which can take time. And so I think the earlier you meet with us to discuss those plans, even when it's still like very exploratory thinking, that's probably the biggest contribution, at least from the sponsor point of view. I don't know if you wanted to add to that too.
Yeah, I think we need to pool our resources. Our information resources, we need to know from academia what you're working on and what you think is interesting. Our data resources, we know that patients want to share their data, and they don't care which company owns it or which clinical trial it was gotten from. They want their data to be useful, and they have provided it to our institutions and to drug companies, and we need to pool that so that we know how frequent a biomarker occurs, whether that biomarker has anything to do with prognosis, and help us design trials through that. And then finally, we need to pool our patient resources. They are our most precious, our best resource, and we, in a rare disease, whether it's reaching out across the ocean to have more international trials through Project Orbis at the FDA, whether it's by platform trials that are funded by different companies who will provide their drugs, so that we do not have to randomize a one-to-one study to a control arm and an experimental arm. We need to know that we have this rare patient population to do better for, and the more that we are able to work together in all of those things, information, data, and trial opportunities, I think the easier it will be to move the bar.
And Nicole, you're not meaning just positive data, right? You're meaning
yes, exactly.
negative stuff
we need to
too,
know
right?
if something didn't work out, we want to know if there's 4 different drugs being developed for one very rare biomarker. Maybe we don't need all 4. Maybe 1 is okay. Maybe 2 are okay. And if that didn't work, then maybe we should all know about that so that other companies aren't picking up drugs to target a particular biomarker when that maybe same approach didn't work once. So yes, positive and negative.
Thank you. And from the Medicare end of the table, what do you guys need from your stakeholders?
Yeah, so I think we're also dot connectors from a medical affairs perspective. And I think we're also trying to assimilate the insights that we're hearing from clinicians, from clinical practice, from patients, and the regulatory requirements as well to help inform future study design. So the more insights that we can get and the more that we can assimilate that information in a way that makes sense, that will really help inform future studies and also avoid pitfalls, right? That learning from other experiences.
I would say that probably communication is extremely important and will apply in each of all of these interactions. So in our interactions with the investigators, we want to know how trials are going, what they are seeing in real life with the evolution of the patients, how things are doing good or bad, and is there any new biomarker that needs to be included, or are we going to be able to collect samples so that not only the clinical data will be relevant, but some scientific information that will be relevant as well. So in interactions with, from the regulatory agencies, of course, and I think that we heard today, what would be like the ideal designs for rare diseases. So I didn't say that before, sorry. But I think that rare diseases are a particular area that is even more interesting because every patient that you make a difference, you're having a huge impact in the overall situation of rare diseases, opposite than the most common diseases that you may need thousands of patients to prove something. Rare diseases is something that needs to be approached and seen differently in a very, very, very more productive way. So what is the right trial for these rare diseases like osteosarcoma? Maybe we need some innovative approach, as has been mentioned before more than once. With other companies, also a good communication that makes us, are we working good with a single agent, or we need to combine this drug with other, and you have that asset. Can we work together so we can do the best trial to improve the life of patients? And of course, the Osteosarcoma Society Community Institute, it's doing more frequently this interaction. This moment of having the opportunity to talk all each other and share what we are going through is fantastic. And so I hope we don't need to wait 4 more years for the next meeting. And of course, it seems that each of us has a piece of the puzzle. And if we want to see the whole picture, we need to put all the pieces together. And that's what we are intended to do.
Fabulous. And Allie, how can we continue to amplify Charlotte's voice as key stakeholders intentionally in this process towards success?
Charlotte, Mackenzie, Willie, Olivia, Dylan, so many kids that we've met. I think it's sort of uncanny that we have so many brilliant minds in this room today. And yet I personally think that the people who are most qualified to tell us to get going are the ones who aren't here.
That is a very deep statement, and I almost want to just end on that. And I will. Thank you, Allie. Thank you, Charlotte. Thank you to the whole panel.
Well, thank you all for some very stimulating conversations, and really appreciate the conversational style of these last two sessions. And I was sitting towards the back, and I did hear quite a bit of muttering during when you Nancy and Mac were chatting. So I would love for for some of those mutterings to become questions during this session. So this is just
I don't know about that.
to become questions. To become questions. So we would love to have folks in the room ask questions of this very, very knowledgeable and experienced group of people. And then, of course, we also have folks online who are bringing up questions.
So thank you for great discussions and really fantastic sort of presentations. One of the things that hasn't been talked about a lot is the NCI-funded cooperative groups and where they live in the drug development spectrum for osteosarcoma. And I welcome discussion about that.
I suppose I should start the answer to that question. So Katie Janeway, chair of the Children's Oncology Bone Tumor Committee, which, by the way, I inherited from Richard Gorlick, so who asked the question. This actually brings out an important question that I was kind of discussing off to the side with a couple of people, I think Melinda and Allie. Formal drug development, bringing a drug to approval or getting an indication actually can happen within the context of a cooperative group, but I would say it's the exception rather than the rule. And the reasons for that are manyfold, but they really hinge on kind of the requirements to audit, monitor, and the level of data collection that's required. So those sort of standard requirements for regulatory approval are not usually met by our standard cooperative group trial that is fully funded by the NCI. And so if you would like to conduct one of those types of registration trials within the context of a cooperative group, it is possible, but often requires supplementing the NCI funding to enable that type of trial oversight. That said, what I was discussing with Melinda and Allie was a topic that really hasn't come up in this conversation, and I'm very curious about, which is there any world for rare diseases where those the levels of sort of auditing and monitoring and data collection that we perceive to be required for regulatory approval can be modified while still maintaining the quality of data that's needed to assess the drug properly? Is that a feasible thing? And so that's one thing. And then I have other things to say too, but I think that's really important to talk about.
Maybe I'll take a little bit of that. So I think yes, depending on what type of drug product is being utilized in the trial. So if it's a clinical trial that's using a drug that has already been approved and is being repurposed for a new pediatric indication, certainly I think some of the data requirements, safety data collection, have the opportunity for flexibility there. So if we have a good sense of the safety profile of an already approved drug, we may not need you to collect adverse events to the level that they would be collected for a new molecular entity or first in human. And the other thing I think that could potentially reduce costs, although isn't leveraged often in pediatric trials, are the decentralization of some elements of the trial. And it doesn't need to be a fully decentralized, pragmatic trial. There can be elements that after discussion, with us, that we could say, like, maybe that's something that, for example, labs, maybe it's fine to have the labs be collected at the patient's local lab rather than coming to the study site. So I think there's a lot of other examples of ways where, particularly if it's a drug for which there's experience and a good safety experience, we would be really flexible in working with you as you're designing consortium trials to hopefully sort of reduce the regulatory burden and the overall trial burden.
And then I'll just say one other thing, which is I think maybe that's not the point you were getting at, but the other point that I want to make is I don't think there's any world in which all of the drugs that are of interest in osteosarcoma will have their initial studies or even their subsequent studies in the context of a cooperative group. I think there are some agents that are of such interest to the company for sort of commercial development in this space that they will want to sort of own that full process, which can happen in a cooperative group with this sort of hybrid model, but doesn't leverage the full cooperative group. So then is it like, it's only sort of a partial cooperative group trial. So I think that there are examples of us very effectively bringing forward some of these newer agents within the context of our committees, and also actually Alliance is very active in the sarcoma space. That's one of the medical oncology cooperative groups, and SWOG. And we should mention SARC, who is another cooperative group that's not federally funded, but there are actually really effective cooperative groups in this space. Did I get them all, Laura? No. I missed some. Yeah, others.
Katie, I fully agree. However, one of the elements that I cannot avoid thinking is that as we continue to work on biomarkers, we all know that the big pie has smaller and smaller and smaller slices every time. So I envision a not too distant future, provided that the databases talk to each other, in which that could happen more often.
What I'm not sure I know what you mean by that, but that where cooperative groups work together. Yeah, I do think the cooperative groups do have an effort to work together. So that's like a known thing. It is sometimes difficult to achieve just because of competing demands on the local level for limited clinical trial resources on the institutional level. So there is some prioritization of cooperative group trials against sort of industry-sponsored trials that happens on an institution level that have so far hindered sort of full, complete rollout, with the exception at Dr. Davis's site.
We have a question over there.
Hi again. I guess in the context of industry, if we're to look at achieving something like an accelerated approval on a smaller patient population, is cooperative groups, I guess, open to performing the confirmatory studies, which the FDA typically requires us to have underway by the time of approval. I guess that might be a question for the FDA as well, but yeah.
I think the answer to that question is above my pay grade. I guess I would say what I know I can say for the Children's Oncology Group on behalf of Dr. Hawkins, who runs it, is that they are always interested in speaking with industry about their collaborative models and whether there's a model that would fit your needs. That's all I can say without overstepping.
Yeah, I mean, we would be happy to consider it as, as any confirmatory trial as part of the development plan. So yeah. Yeah.
I think
Amanda, did you want to?
I just want to. Oh, I'm sorry. Go ahead.
No, it's a separate question.
You sure? I just want to comment on cooperative groups and collaboration with pharma or biotech. In rare indications like osteosarcoma and other pediatric cancer, I don't think we can do without collaborative groups, right? So whether it is a form of collaboration through an IST, an investigator-sponsored trial, or a more formal collaboration through a biotech-sponsored trial, there needs to be that close collaboration because there's just no way out. So having that continuous relationship actually before we need it really matters.
I wanted to ask a question about optimizing opportunities for drug access. Can you hear me? Yeah. My question is about optimizing opportunities for drug access. I've learned so much today about opening up pathways toward regulatory approval. And the first step there to sort of being able to ask the question about approval is getting access to a drug to actually run a trial. Nancy, I so appreciated your shedding light on opportunities to align incentives. I think I really sort of thought everyone was going to contribute out of the goodness of their heart. And it makes so much sense to say, how can we really make this a win-win for our patients, for the folks wanting to run the trials, and of course, the companies that are marketing the drugs. And so wanted to ask those from industry what you're looking for from, let's say, academia coming in with preclinical data and a desire to run a trial. What makes that attractive? And Nancy would like your input too in terms of how to best frame the goal to write a trial in a way that works for everyone.
So the full answer to the question, it's also above my pay grade, but several fold. So from a whole heart perspective, we all want the same. We're all here because we want to end childhood osteosarcoma. We want to get rid of it. There are compelling interests and compelling constraints. I was very serious when I told Melinda in the prior session that we really need the help from all the community to be able to be internal advocates and be able to educate within big corporations and sometimes smaller biotechs about all these different areas in which we can and should work together to get to the same endpoint. It has to be a win-win-win situation. There is no other way. No.
I just jump in. So, this happens, these studies happen in the oncology space pursuant to Race for Children Act now. And what happens there is before what would be the end of phase 2 meeting, sponsor meets with the FDA, and there's, as you know, better than me, I've never been to one of these, a discussion as to what the pediatric study will be. So I guess my question is, and by the way, academics are not part of this process. The law says the meetings and the negotiations are FDA and sponsor. But the experts on the disease areas might be the academics. You as the FDA, how could you possibly know for a particular targeted therapy, all the indications for which that therapy is appropriate? Of course you can't. So, I have an open question for all of you. Is there a way we could create a process where academics, know what's being developed and could advise the FDA? FDA there's some sort of constant exchange of information. Academics sign NDAs to protect industry. Also, then industry doesn't have to spend all their time trying to figure out like what pediatric indication. I mean, they don't know the pediatric indications. They don't know what the clinical development plan should look like. So is there some process where we can bring everybody together?
I was going to actually, so I think that's a great point. And the RACE Act and the FDARA required studies that you're talking about are laid out in IPSPs, which are where the sort of early development of, in pediatric cancer is described. And there's a very similar document that's issued in Europe called the PIP. And in Europe, the PIPs are all public. But in the US, the IPSPs are not public. So it is very difficult for us even reviewing, or I should say really Martha reviewing hundreds of IPSPs every week. To know, how many drugs are being developed for one particularly molecularly, relevant population, and that information is just hidden. It's all private, and so it would be. I was going to ask you, Nancy, if in any of your, legislative work, if there's been thought given to IPSPs being made public so that we could all know, what companies are, what studies are going to be happening for a molecularly targeted drug, because I think it would help sort of all of us, the academic people and FDA and companies.
We've actually talked about that for years. And the problem is, well, there are two trade associations that represent industry, bio, and pharma. And so far, they've been wildly opposed to that. But, there's certainly an opportunity to bring it back into the conversation, and maybe they would change their minds. And it matters because the way you get a bill passed on Capitol Hill is you have to have the two big stakeholders are FDA and these two trade associations. And you have to get the at minimum, you have to get one positive and one neutral. So you can't have either be negative.
I was just going to say that one approach, I know you know about this, Nancy, but one approach that's been used in Europe very effectively is the Accelerate Forum. So, this is maybe a little bit like an Accelerate Forum, a sort of similar type of model. It's multiple people from multiple different perspectives having a conversation, a structured conversation, but that is one approach, and they have not had one on osteosarcoma. They've had one on drugs that are relevant to osteosarcoma. I think you heard Dr. Marinoff talk about that, but it's one thought. I guess the only other thing I'll mention is that I do think one of the other challenges in this field is the overlap between medical oncology and pediatric oncology, and the need to bridge that group as well in the academic space and in the institutional space. And so if you're thinking about something like that, having a purely pediatric focus, like the Accelerate Forum, actually in the US at least, and also in Europe, to be honest, and in other countries, doesn't necessarily get you everyone you need at the table.
I have a couple quick questions from, well, I don't know if they're quick, from online participants. Is that okay? Okay, cool. Have had some really great questions coming through. A couple of them from Chelsea W. And it's hard for me to pick which one to choose to prioritize, but one relates to funding and sort of prioritizing the type of advocacy that, for policy changes that we're trying to do. And she said, it's commonly said in the pediatric cancer community that pediatric cancer gets 4% of federal research cancer funding. From what I can tell, it's the NCI NIH that determines what adults versus kids get in terms of funding. Why are we advocating through policy versus advocating to the NCI for a bigger share of the allotted dollars for pediatrics?
Both kinds of advocacy. Can I like this? Is this better? Okay, sorry. So both kinds of advocacy are really important. So there are And on the legislative advocacy community, we each sort of pick one issue and we stay in our own lane. That way we can do a really good job in our lane. And the lane I've defined for Kids versus Cancer is FDA law. So when we look at the Pediatric Priority Review Voucher Program, it doesn't result in additional funding through NCI to pediatric cancer. It just creates incentives for companies. When we pass Race for Children Act, it gives FDA new authorities to require pediatric studies. It doesn't say anything about NCI's budget. The Gabriella Miller Kids First Act does result in new appropriated funds. It actually goes to a fund outside of NCI, but half of it goes for pediatric cancer. So that's an appropriation. That's appropriation move. Or for example, the DoD efforts through the Congressionally Directed Medical Research Program, that is like new money that comes to pediatric cancer research. So the answer is, and both are super important. I don't do the appropriations because it requires an amazing amount of expertise that we don't have.
Thank you so much. She also asked another question related to sort of decentralization of clinical trials. And I think that's something that we, keep coming, touching upon here and there. And Chelsea also mentions the Target Cancer Foundation, which has a completely virtual clinical trial. And what are the barriers to running trials that way, or either fully decentralized trials or relying more heavily on virtual participation? And, are there any kind of low-hanging fruit that you think we can kind of work on as a community to make trials more accessible to patients where they live?
Everyone knows more about this than I do, but I'm talkative. So, I went to OHSU last month, and one thing that I didn't know, which was kind of shocking to me, is that, when there's a COG trial, hospital needs to open up. If they're opened up for the COG trial, it costs them something every year to stay on the study, even if they don't have any patients. And the reason it costs them something is every time there's an amendment to the study, they have to retrain all the nurses, and it gets quite expensive. It takes up a very large portion of a pediatric cancer department's trial funds. So, and I understand that you are experimenting with COG on some other trial designs that would, sort of open up the study as soon as you get a patient instead of having it open forever, right?
I would say that the COG trials cost the institution. We have far fewer requirements than an industry-sponsored trial. I just want to say that, just so everyone knows, our requirements for doing something that's a standard COG trial, like the amount of data that needs to be entered and the number of amendments and the number of drug safety reports you have to review, is tenfold less for a cooperative, I shouldn't say COG, for a cooperative group trial. That's part of our philosophy. And we open trials at hundreds of sites using a central IRB. So the regulatory requirement is lower, but it's not very well compensated.
But it's not compensated. Hospitals subsidize that, whereas
that
for industry studies, hospitals are fully compensated.
That is correct.
So it's
that is correct. From an investigator perspective, the barriers are, we have regulatory requirements for research that are essential to follow because they have to do with patient safety. And those regulatory requirements, the way they're interpreted currently, require that much of the study conduct happen at your center, and that you have to keep up with all those amendments. You have to actually have IRB approval. You have to have eyes on the patient. You have to do it when the study tells you to do it. So those are, do we actually need to do all the things that we do in a clinical trial in order to get all the appropriate data for the objective of the study? That is another separate question that all of us need to do a better job of when we write these trials. I think people tend in medicine, at least in this country, we tend to err very much on the side of protecting patient safety. Yeah, I think I put it that way.
Which is very, very important. However, balancing, right? So it's very much balanced as a clinical research program director, making sure that your trial portfolio is balanced, right? Because our NCTN trials, cooperative group trials, are going to cost us every patient that comes on is going to cost us. There's no money coming in. Right. Right. And then, so we have to have industry trials that compensate a lot more to be able to help us pay for the cooperative group trials. That's just the way it is. Decentralization is a fantastic concept, and we would love to be able to do that more. But in order to balance having enough data on safety with that increased access is very difficult. And so for example, if I work in a very large state, giant catchment area, in order to have my patients be able to get labs on trial closer to home, we have to get their hospital approved by our IRB. So it's very expensive to do that, but it's pretty important to be able to not have to make a patient drive 6 hours for a CBC. And so when we are writing our IITs and advising on how to write a clinical trial protocol, if we don't really need to know that the CBC machine at that institution is perfect, please have them not either. Maybe they're not necessary at all and eliminate them, or have them able to be at a normal, regular old CLIA lab, and that will help.
So I'm not very talkative, but I do have one comment. To answer your question, actually, I think we don't know enough about decentralized trials as sponsors, as sites. I think there's a lot of preconceived ideas about how risky that might be, how much we might miss data. We actually are not getting curious enough about how to do this right. And by the way, I'm referring to registration trial in this case, right? And that takes me to another point that some of us talked about offline, is the cost of running a registration trial. Getting to the virtual, more of a virtual model is one of the key ways to actually reduce the cost of a registrational trial. And so we do need to do a better job at understanding what that looks like, what that may look like, what are the limitations, if any, so that we can implement that more as a standard, if you will, when we can, when it's realistic, really.
I'll just add one thing from experience when I was working with SARC. SARC spent a lot of money doing audits of trial sites, and it was not a kind of pre-existing group. It was kind of, the sites were built, the participating site list was built at every different trial. And doing the work that's required for a clinical trial is not easy. And one bad site can blow up your trial. And so it's a really attractive idea that's really hard to execute. And I don't know the solution for that.
Yeah, I think it is hard to execute. I think we sort of, just as you said, we sort of all just end up repeating the same thing. So all of the labs are done at the study site. All the visits are at the study site. But I think, as I said before, I think there's a sort of a middle ground, and Harpreet referenced one big example of a fully decentralized trial in Pragmatica Lung. And in that case, it was 2 drugs were being investigated that had tons of adult data. It was adult lung cancer, and there was a survival endpoint. So they didn't even need imaging. Or they didn't need centrally reviewed imaging. Imaging could just happen as per standard of care. But I think there's a way sort of between that, which probably isn't going to happen immediately for a pediatric cancer trial, and having everything be born, at the study site. And I think there is probably, if we were to go through it all, there's probably a lot of redundancies or things that are, required by CROs or by a company that haven't actually just been discussed with us when they're coming. And so maybe there's a place where we can sort of meet and say, yeah, like you're studying cabozantinib in your trial. We have a lot of data for cabozantinib, and we have data in adolescents, in pediatric patients. And so maybe maybe there are places where we can identify more pragmatic elements that makes the study better. So I think that there for sure is a middle ground. I think we just don't hear about it. And when we're discussing, trials with sponsors, it just sort of doesn't come up. And so we at the FDA don't hear like, hey, this is really, this is just really difficult to functionally do in a trial. And is a CBC something that, we can do at a local lab?
Nicole, I'm wondering if during the pandemic, there was any hybrid approach. We all switched to being able to do what was best for the patient in the setting of a lot of lockdown. Were there learnings from that could become a part of guidance that would allow us some of that leeway to let the home labs happen on very standard, labs, et cetera?
Yeah, I think we learned a lot from COVID. And actually, I don't know if Harpreet's still here. I'm going to call it because she did a lot of work in this space. Is she around? She had to. Okay. Well, so yeah, I think we did learn a lot from COVID. I mean, we learned that, we learned about telemedicine visits. We learned that a lot of things can be done more efficiently, more swiftly. And it seems like that's sort of like all is going away or went away. But they were good lessons learned. I don't know, Martha, if you want to comment on that also, because you probably were involved.
I think to your point, we haven't learned enough from what the benefits were. And part of that is it's really hard to pull together the data on sort of how this benefited patients. But we absolutely, so a lot of the things that prevent the type of flexibilities that happened with COVID are not FDA regulations. There are all kinds of rules relating to shipping products, investigational drugs. For instance, there are telehealth regulations that may vary from state to state. Like, I think it's a complicated web to try to tease apart. And I think that during COVID, a lot of the rules that people, were very willing to shake things up out of necessity. And I think perhaps we need to bring that level of urgency to today and, realizing like, these are the things that we think really, I think, the IRB system, seems from the outside pretty clunky sometimes and kind of makes it harder to have more widespread, consistent changes in availability for, some simple tweaks that I think could make a big difference for patients. So I think it's a big picture question. I think, FDA can certainly be part of that, but I think we need to look at every step along the way to really implement change. I think some of the low hanging fruit, really kind of bringing people to the table. And, at our meetings with sponsors, we don't talk about the granularity level of, do you really need to do these labs every 2 weeks versus that's just not the type of thing that we talk about. But I think from a disease perspective, if we can say, well, at this stage of development for a new drug, this is what we're comfortable with in terms of standard collection for safety, may be different when we have an approved drug, like coming up with some sort of guidelines and thinking about that as a community so that companies can kind of rely on that and think about that and realize, okay, this is reasonable. I think that would be really helpful to do as probably a good first step.
So I was just going to say, I agree. And as you said, like, it doesn't really come up in our meetings. I think that, as you said, some of the lessons that were, learned during COVID maybe have gone away due to, as Martha said, things that are sort of beyond FDA control. But I mean, we reviewed trials that were conducted during COVID and approved drugs based on those trials. So I think that, as I said before, I really think that it can be done. I think we just don't hear about it. It's not like there's so many other things to discuss. We don't get into the discussion of, do you guys really need CBC done at a particular lab?
The only other thing I just wanted to mention quickly is that we could certainly learn from what works well in other countries as well. And, I think one thing that comes up a lot in the rare space too is that companies and stakeholders are not just thinking about trials that are going to satisfy whatever FDA says is okay, but you also have, the rather large goal, an important one of thinking about what other regulatory authorities, what's important to them. And that's not an easy thing to tackle. So, I think, bringing in our international colleagues as well is really important to some sort of discussions like that, so that we don't say, well, we can live with this. This seems great. And then you go to another country and then you're hitting another brick wall, or vice versa.
I guess a general question for the panel, maybe a question of speculation, is what's the one thing that you think would really move the needle here, instead of sort of maybe incremental improvements? What would really, whether it's a modality or a biomarker, or and it's a speculative question, so.
Okay, I didn't get the chance to answer the similar question that was asked before, but I have my answer already prepared. So for me, a major challenge, I've alluded to this a little bit earlier, a major challenge that I face as an investigator interacting with my colleagues in pediatric oncology who specialize in drug development and leading trials at my institution, and also discussing agents of interest in osteosarcoma with industry, is that fitting trials targeting adolescent and young adult enrollment into the pediatric drug development framework can be very challenging. So for example, there are times when we begin to have discussions and they seem to be going very well. And then the next thing I hear is, well, we now have to figure out our PIP and our IPSP, which are the European and the US pediatric development plan. And then that requires discussion with multiple other diseases, sometimes preclinical work, and 10 different solid tumor disease models. And suddenly the osteosarcoma part of that, which feels relatively straightforward in terms of being able to use or borrow dosing derived from trials in adults, or maybe even being able to fit into some kind of adult drug development framework, becomes wrapped into the pediatric drug development framework. So I guess for me, I would like it if we came to a consensus on best practices for drug development in the adolescent and young adult patient population in the academic industry and regulatory settings. And maybe that lives firmly in pediatric drug development, but maybe it doesn't. And so it's not that those programs aren't important. It's just that I think there's a lack of clarity. I find a lack of clarity in my colleagues in industry. I don't know how much it comes from the regulatory space regarding the extent to which an osteosarcoma standalone trial meets those pediatric drug, those requirements for a pediatric plan.
Can I chime in a tiny bit? I think we have work to do on this, and I'm eager. Speaking for the FDA now, we are eager to help work out any of these sort of unintended consequences that we don't want to happen as a result of really great legislation. And so I think there is an educational piece, there's a communication piece that can certainly help combat that. I will say that in terms of a scenario such as you present, we would absolutely not want to delay the osteosarcoma program. And would be willing to kind of talk sponsors through that if they're concerned about how this may or may not affect an agreement. And I'm committed to working with EMA's contacts, at least in broad strokes, so that we can align on some messaging with regard to that. Right now, for the time being, most of the requirements are really coming from the US with regard to molecular targets, but that's not always going to be the case. And there's incentives in Europe that companies understandably want to be able to take advantage of for voluntary studies. So currently our requirements do not include a requirement to study every pediatric cancer. It's we want the drug to be studied in the pediatric cancer that makes the most sense for that product. We do have requirements to have all age groups studied. That doesn't necessarily mean that if there's a drug that has a target that's great for osteo, but doesn't make sense for, younger kids, that we wouldn't want to require a study in younger kids if we know it could have activity in medulloblastoma or something else. But that doesn't mean that you can't do the osteo first and then have discussions and do some background work to try to study the product in that younger population when it makes sense. And so there are times when a sequential approach really is the best approach, and it actually behooves a company to go ahead with the study that we have enough scientific data for, because that will help satisfy the larger requirement. But I think some of that is just having those discussions so that we can kind of think about in a given context what makes the most sense.
Just jumping in
We
here,
should work on that.
Martha, and as you work on it, the black letter law says, it says single or plural when it's talking about the pediatric indications. So the intent was not that if some adult targeted therapy is relevant for medulloblastoma, that there shouldn't also be a requirement for osteosarcoma. It should be, the FDA has the authority to require the studies for every pediatric indication for which the molecular target is relevant and it makes sense.
But the requirements are restricted to phase 1, early phase 2, but we do have incentives. So, building upon the incentive with the requirement in a way that kind of satisfies the needs of all parties involved is like the way for us to be thinking about it. You're going to say no.
I'm
You're
just going to
shaking
push
your
against
head
you.
no.
The law expressly says you don't have to use the incentives at that point. That
No.
FDA can require the studies for all indications. You use the incentives once the early phase 2 looks, exciting, then you use the incentives to do, a full phase 2 or maybe a pivotal study. That's what you use the incentives for, or maybe, correlative science, but you don't use it for extra indications. Or, obviously you could, but the law expressly says FDA can use the requirements for all the indications for which it's appropriate.
I need a lawyer, apparently.
I know one.
But in practice, we want to strike a balance so that, we're doing what's right for patients with the data we have in hand.
Thanks, Martha. And oh, sorry, I was going to say we're toward the end. I really want to thank the panel before we get into closing words. We started out today hearing from patients and advocates, and so we would like to end the panel also hearing from patients and advocates. And so, Mackenzie, I wanted to turn to you to see if you had anything else to say, and Anne as well.
I just wanted to end by thanking you all for being here. As patients, we rely on so many people, our doctors, our parents, and we have hope that drugs we are taking will be improved so no one else struggles with what we did. We have hope that someone somewhere is inspired and passionate enough to come to places like this, wanting to hear our stories and wanting to take action, wanting to leave making it better for kids you hear on these panels, for kids you hear in the hospitals, for kids that are no longer here, and for kids who haven't heard the words, "You have cancer." And to hope in the future that those newly diagnosed have better treatment than we did. So thank you.
Okay, I just have a quick thing, and I'm going to ask you to come back in time with me for a minute. When we were talking about having a CBC draw close to your home. So one of the questions we get regularly is, how do we make a clinical trial design that's appealing? And how do we get patients and families to stay on a clinical trial? Don't make them drive 6 hours to have a CBC. That's kind of a deal breaker, I have to tell you. That really is. And it might not be an upfront because you're ready to go on the trial and you're like, I'll do anything, I want to be on this trial. And then you find out that twice a week you've got to drive. And if you look at Michaela, for example, do we want to wrap her up, put her in the car for a blood draw or for an x-ray when she's got a place down the street where she can have that done? So accruing trials and keeping patients on the trial requires not just low-hanging fruit, but common sense. Common sense, just this is easy. This is not a hard thing. A blood draw is a blood draw. We all have cross-institutional like portals that all of our information's on. Please, this seems like a really simple win for today. If we could just make it so that you don't have to go all the way back to your major institution for a blood draw. Yes, please. Okay, apart from that's we can go back into the present time now. And I just wanted to say thank you to OSI and to FDA for convening this. This was really powerful, really impactful. I think everybody here feels pretty privileged that we got to be a part of this conversation. And I can't wait for more. Great, thank you.
So Dr. Wessel and I are going to give some closing remarks and some take-home points. So for me, the take-home points here are that there are clear approaches to signal finding studies which can support trials in the upfront setting and may contribute to various accelerated pathways in the FDA. I think there are multiple possible trial designs and endpoints. I think there are relevant incentive programs, alternative pathways, and regulatory requirements that can be applied to osteosarcoma. And I think there are invested regulatory, academic, patient advocacy, and industry community around this disease. You can just see it from everybody sitting here and from the engagement in this meeting. But there are challenges that remain, and I am optimistic and hopeful. Just as Mackenzie said, I think this outstanding meeting, the presentations, the perspectives, the participation, and the respectful dialogue definitely will help. I think this day has been incredibly productive, and I want to give a lot of credit to the OSI and to the FDA for taking this on. I think that figuring out the approach to an efficient trial design that could satisfy regulatory approval requirements while also serving a broad population of osteosarcoma patients as possible is a definite challenge to this community and something that we should really strive to achieve. And it should be doable. I think we need better data for trial relevant populations to be utilized in trial design. Some examples are event-free survival in metastatic extrapulmonary disease, overall survival for fully resected lung-only patient population. And I think these data points need to include molecular data. We need to further develop and incorporate alternative endpoints, circulating tumor DNA, PET, novel radiopharmaceuticals, and proof of mechanism. I think a clear understanding of how pediatric incentives and regulations apply to osteosarcoma and whether osteo trials alone can satisfy pediatric regulatory requirements is really important. And then I think collaboration and communication as early as possible has come up over and over in this meeting, and this meeting is a great start for that. And I think finally, just to say that I'm sure the planning committee will have some debriefing to do, and we will certainly consider what some deliverables of this meeting may be. And I just really want to thank everyone for being here, for engaging, and for bringing their A game. Thank you.
Sure. Thanks, Dr. Janeway. And I really can't express our gratitude enough for all of you who came here and participated and engaged in these really fantastic discussions today. I think from a regulatory perspective, we hope that we've imparted to you that we would like you to leverage us as much as possible. Both that goes to patient advocates, academic investigators, and drug developers to leverage our resources at every stage of development, whether that's preclinical data that you have, you want to start a first-in-human study and you want to submit for a fast track, or if you have some promising clinical data you want to review with us, if patient advocates have concerns that they want to share with us through a patient listening session, we're available to you all through the process. I know that we weren't able to get to everybody's online questions today, and we'll try our best to go through all of those questions and answer them. We are sending out a survey after this, and hopefully everyone will fill it out. We do have one question in there that I think will be great for our planning committee to discuss at our debrief, which is what is the highest priority action item you would like to see come out of this workshop. So I hope everyone takes a few minutes to answer that question in particular. If anyone wants to go back and revisit what we've discussed today, we'll have the workshop recording and slides up on OSI YouTube by Halloween, and then our FDA event webpage at some point in the near future. And then I did want to give one final shout out to those two people in the front, Amy from OSI and Erica, who just worked very meticulously to make today go smoothly. So thank you both for all of your help. Oh, I think I forgot one other important point that's kind of out of order, but I wanted to say that when you come to meetings with us, invite patient advocates with you and invite academic investigators to come with you, because we really do value the input that we get during those meetings. And then everyone sort of gets to communicate in real time instead of looking at meeting minutes afterwards. So I think that's something that some sponsors have done that we found really, really helpful. And we hope to see more of that in the osteosarcoma space as well.